Okay, looks like we're live. We are in for a treat tonight, guys. We've got Erika, who goes by Gutsick Gibbon on YouTube. Be sure to check her out. And John Maddox, who goes by Logical, Probable, and Plausible on YouTube. So make sure you check him out as well. This is the much anticipated debate on intelligent design. So it looks like we've already got a lot of viewers in the chat today. So it's a blessing because I have brother Sal here. He's no stranger, and neither is John and Erika. So we're going to have a good show tonight. Brother Sal, thanks for co-moderating with me tonight. He's going to be in charge of the Q&A. So guys, like I said, we've got a big chat tonight. So just make sure you tag Salvador Cordova. We don't want to miss your question. We'll make sure that we save it, and we'll have a good Q&A at the end. So we've got a little formal and informal debate tonight. We've got about 10-minute openings followed by a discussion and quick closings, and then a Q&A, like I said. So make sure you're tagging Sal. Before we go into the openings, though, let's kind of break the ice. Both Erika and John, no strangers to debate or this channel. But let's start off with you, John. Give us a brief introduction. How you doing? You know, how's your channel? Go ahead.
Oh, doing good. Thanks for having me on. Looking forward to this conversation. I'm a little more tired than I would have expected. Just got done with a debate over on modern day debate, but looking forward to this one. Should be a good time. My channel's kind of directed a combination of both intelligent design as well as addressing some of the issues that I think atheists suffer from in regards to their mental positions and worldview and how it leads them to irrational conclusions. But tonight we're going to be discussing intelligent design, and I think it should be a good time.
Awesome. Thanks for that, John, and much appreciated. Thanks for giving us your time, even though you're as tired as you are. So over to you, Erika.
No, yeah, absolutely. Thanks for being here, John. Let's see. My name is Erika, go by Gutsick Gibbon on YouTube. I run a channel that covers a lot of paleontology, anthropology, things like that, and a lot of creationist content that I kind of try to get a deeper look into and kind of dissect. But I'm very excited to be here. No real news on my channel, but I did get engaged in real life, so that's kind of fun and
congratulations. That's awesome.
Yeah, I'm very happy about it. So, but man, it's almost up there with having a conversation on intelligent design here on the SFT channel.
Well, it's an honor that the announcement of your engagement has occurred on SFT. So that's awesome. Congratulations. We're happy for you.
Yeah, thank you.
No problem. It looks like we already have questions coming in for you, Sal. So you're going to be busy tonight. It's why we pay you the big bucks, though.
The biggest.
Keep it up, guys, in the chat. Yeah, make sure you're tagging Sal. And my apologies. I'm not too sure who is starting. Whoever wants to volunteer, I guess, and then I can put the timer. Up to you guys.
It doesn't matter to me.
Yeah, I don't care either. It's you can go first, John. I will cede that to you if you'd like it.
Perfect. Okay, John, no rush. I know you have a presentation. We got Praise here, our wonderful producer. Of course, brother. No rush. Couple quick announcements. This Wednesday at 3, check out a Marvel Girls channel. I'll be debating conspiracy cats for round 2 on Ancestry, so that should be fun. And we've got Kent Hovind and Ron Garrett. I believe, I got to double check, but I believe it's July 9th at 9 EST. So we've got a lot of good debates this month. Working on confirming another couple. I'll announce those when they're officially confirmed. So it looks like, John, your screen is shared. So enough for me.
You can see it.
Your 10 minutes begins when you're ready.
All right, so tonight as we discuss the origins of humanity and, from my perspective, intelligent design, it is my intention to showcase that intelligent design is not only the most plausible conclusion for our existence, it is also the most logical explanation for the existence of all life as we know it. As people have heard before, I call Darwinism, the Darwinian evolution through natural selection, the greatest scam on earth for a very important reason. As we progress through the evidence against this being a logical, plausible, and probable explanation for our existence, the level to which philosophers and academic elites use it to dismiss a higher intelligence will become clear. Now, obviously, this debate will go in many directions. But before we even get to the fallacies of Darwinism, we must first address what I argue makes the entire premise of evolution fruit of a poison tree, and that is abiogenesis. My opponent will likely argue that, it was a much simpler back then than what we view as life now. And she may even attempt to explain the improbabilities away by saying life exists, therefore the probability is actually 1. However, let us consider if that position of potential explanation for the prerequisites needed for the theory she stakes her life on is remotely plausible to occur in an undirected manner, or if in order for the probability to be 1, the rational conclusion is an intelligent agent. So even if simpler parts somehow came into existence, this does not circumvent the requirement for a minimum number of these parts to all exist at the same time in the same specific location. And while atheist origin of life researchers try to avoid this conundrum, it is just not that unavoidable. So let's now consider what is recognized as the simplest form of life and how its simpleness by comparison to other forms of life does not mean it is actually simple. The simplest known organism, the Mycoplasma genitalium, has a genome of 580,000 base pairs. This astonishing number enables it to contain 470 genes that code for 470 proteins, with an average of 347 amino acids per protein. The odds against just one specified protein of that length are 1 in 10 to the 450th power. If we calculate the entire proteome, then the odds are actually 1 in 20 to the 164,090th power. And this is without accounting for the increased improbability when you consider that nature had to select only the left-handed amino acids and bifunctional ones. All of this is before considering all of the genes must have also been in existence at the same time in the same location on a chaotic prebiotic Earth in an environment that is admitted to be less advantageous for the required chemical reactions to occur. So now let's put this in context. The total number of seconds to work with in the entire history of the universe is only 10 to the 18th power. The total number of particles in the entire universe, not just here on Earth, is 10 to the 80th power. And the total number of interactions by those particles in the history of the universe is 10 to the 143rd power. And all of this is before considering the origin of the genetic code, the assignment of arbitrary values to codons to equal amino acids, and the requirement of DNA and transcription proteins and translation proteins to exist simultaneously, even for basic replication to occur. What is ironic about this is that I am barely scratching the surface regarding the incredible number of variables which must operate in unison for life to exist. So as you're considering all of these pieces of evidence, ask yourself, is it more reasonable to conclude that an external intelligent agent was needed for life to exist, or to imagine something that is obviously impossible to have occurred by chance somehow did occur by chance? We are surrounded by things which have similar improbabilities to occur through chance, such as the technology enabling us to have this debate. Yet we readily accept that the barrier can easily be overcome by an intelligent agent. Now, what would be impossible by any other standard is now 100% plausible and possible. Now, in regards to our technologies, information theory and computer science are fundamental requirements in order for any of this to be possible. Yet even though the exact same systems are required in biology, the root requirements for their origin in any other context are ignored. So let's now consider the Dna Enigma. It fulfills all the parameters of a code. It has arbitrary values, has a code table, it has translation, which equals arbitrary values, codons equaling amino acids. The codons are translated by ribosomes. Codons do not become amino acids. Codons do not chemically interact with the amino acids. It also fulfills the parameters of programming. Input, output, loop controls, codons, syntax, space and time expression, semantics, controlled variable outcomes, pragmatics, if then else function. It's even been determined to be Turing complete. So when you really start to think about this, you come to the logical argument. So therefore, if information is immaterial and non-random information requires an intelligent agent, then intelligent agents are required for all controlled process instruction. If non-random functional information requires a mind, and DNA genetic code contains non-random functional information, then a mind was required for genetic code to exist. If codes require intelligence to assign arbitrary values, and decoding requires equal arbitrarily assigned values, and the genetic code is encoded, transferred, and decoded, and genetic expression requires translation of arbitrary values, then the logical conclusion is a mind was required for the genetic code to exist. If time-based controls, which we'll cover later, are required for code execution and are executed by separate functions, and they modify outcomes based on temporal variables, and they require simultaneous execution, then foreknowledge of the outcome is required. If programming requires if, then, else logic rules, and syntax, semantics, and pragmatics are required for its execution, and biological cells contain all of these and the tools for its execution, and if no other programming would happen by undirected process, then an intelligent agent is required for the initial programming. Therefore, if in any other context it is accepted that non-random information, coding, decoding, programming, time-based controls, and nanomachines require intelligent agents to exist, and if life requires all of these to exist, and all observed organisms do, then an intelligent agent, and therefore, in my opinion, God, is the logical conclusion for our existence. Now, I must next address Neo-Darwinism directly. And here are 5 positions which challenge the premise of undirected evolution through natural selection being a logical, plausible, or probable conclusion for our existence. First, adaptation of existing functions to account for environmental condition variance is primarily executed through epigenetic factors which do not result in new core functions. This is a known fact, and the epigenome is a fundamental element which has shaken the entire premise of evolution to its core. Not only are these adaptations the result of variable gene expression caused by epigenetics, they are not the result of mutations. Rather, are controlled modifications executed through read, write, and erase nanomachines. Second, it is my position that macroevolution is not proven by speciation, nor by variance within species. While Darwinists attempt to explain this with very slow changes over time, it is now known that literally thousands of genes and gene regulatory networks working in synchronicity are required for new body plans to exist. Not only must they work in unison, it has been shown extremely minor modifications are fatal in embryo development. While I do not discount that variations can occur on a contained level, it is my strong contingency that these are the result of reorganization and adaptation of existing data and functional capacity, not the creation of new information or functions solely through random mutations and natural selection.
You've got 2 minutes, John, 2 minutes.
Almost done. Third, the variation of transcription factors and gene regulatory networks results in significant difference in gene expression. To date, the most studied of these are in relation to cognitive function and show 9 to 23% of variation between chimps and humans, for example. It has been shown these differences must work in unison, and any degradation of this, even to the level of point mutation, results in dramatic impairment or death. Not only does this showcase the extreme importance of gene regulation, it also decimates the relevance of similar coding gene sequences in relation to final outcome. Moreover, the significant differences in non-coding regions of the genome become apparent for things ranging from introns, retrotransposons, enhancers, and promoters, to name a few, almost all of which have been dismissed by those against intelligent design. However, the discoveries made in the last decade have now made their relevance unavoidable. Fourth, it has now become apparent through structural biology that not only is the prescriptive sequence information housed in the genetic code required, it must also be geographically coordinated through dynamic real-time folding to enable full gene expression. This applies to interchromosomal data transfer and gene regulation, which not only forces acceptance of physical gene locations to be vital, it also decimates the relevance of similar sequences existing in other organisms. I'm going to skip 5 and go straight to the conclusion. If I asked you for the source requirement for any piece of technology that contains coding, logic gates, programming, nanomachines, and exhibits temporal controls which defy simple cause and effect outcomes, before even accounting for the physical structures required for the execution being in a specific place at a specific time, yet insisted they'd all come into being without an intelligent agent being required, would you think I was exhibiting rational thought or could even be taken seriously? Therefore, it is my position that the existence of life by undirected physical process, without the action of intelligent agent, is not only neither logical, plausible, or probable, but impossible. As you consider these arguments and the expansion of them throughout the debate, ask yourself this: whose position is more rational? Is it those who conclude it is a more logical, plausible, and probable conclusion that these things could not occur by chance, or those who argue, no matter how counterintuitive it is, You must believe this could all happen without an intelligent agent being required.
Awesome. Thanks so much, John. Just in the nick of time. So thanks for the presentation. Clear, concise. And
I thought I had 12 minutes.
Oh, sorry about that. Yeah, sometimes it's easy to mix it up because there's like debates usually are like 7, 10, or 12. It's always one of those three. Hard to remember which one it's going to be.
It's tough. But Erika, whenever you're ready.
All right. Whenever you want to share my screen, by all means do it because I've got my PowerPoint pulled up.
Praise. Whenever you're ready, we can share Erika's screen and good job to the audience for telling Salvador. Oh, sorry. Go ahead.
It says I'm sharing the screen. There we go. Okay, cool. Okay.
Perfect.
Let's see. Can everyone see that? That looks kind of wild right there. What if I go like this? Is that better?
Looks good. Yeah, looks good.
All right, hold on. Let me get this on big boy mode. There we go. Okay, that should look pretty good for everyone out there. Yeah?
Looks good from my side.
I'm just trying to get the checks before I start. Okay, all right. So I'm going to begin now. So my name is Erika, as we went over previously, and today we're discussing intelligent design versus abiogenesis and evolution, which of course the latter I maintain to be the most parsimonious explanation for life on Earth and its myriad of forms today. So what case am I here to make for you? My case is that life and modern biodiversity is best explained by abiogenesis, that is to say, life from chemical evolution, and subsequently biological evolution by natural selection. What I am not trying to say is that this event and the mechanisms that followed were necessarily brought about by chance alone. I have nothing against that. It is very possible, or at least certainly possible in general, that something or someone spurred abiogenesis or implemented evolution as a mechanism, but I don't know what that would look like, and I don't think that we have anything to support that this is certainly or even probably the case as of now. Which is why I am going with the parsimonious explanation, as parsimony is key to science. So the data that we currently have suggests that life arose 3.8 to 4.2 billion years ago under the conditions of primordial Archean Earth. Data from every conceivable field confirms that life, once present, underwent evolution by natural selection over a vast amount of time to produce the myriad of forms that we see today. Now, while I would rather and typically do discuss the latter, I feel today we're likely going to be spending more time on the former, that is to say, abiogenesis and the constraints of it. By contrast, intelligent design under a YEC context argues that living organisms were created more or less in their present form by some sort of intelligent designer. That being said, intelligent design from a young Earth creationist perspective cannot denote creative kinds, nor can they grapple with the overwhelming evidence from an ancient Earth and universe via geology and physics. Intelligent designers also cannot clarify what makes a design a design. This means, most importantly of all, intelligent design lacks any semblance of a model or testable predictions and relies almost entirely on attempting to poke holes in evolution. As such, it has an abysmal literature base, as anyone who is given dipped a toe into the ID community would know. So what is design? Well, design is somewhat subjective. Here we have our Gibbon that says, what intelligent designs? It's incredible that humans did this. It's easy to project the idea of design onto nature. The eye is so complicated, it simply must have been designed, as humans can't necessarily make an eye. But perhaps we shouldn't be placing design upon things that maybe the design doesn't belong, as natural formations may look designed, but they were indeed crafted by nature and time. I don't think anyone in the audience would look at this beautiful arch or the Giant's Causeway and suggest that someone went out there with a hammer and chisel and made them, even though we do use arches and indeed various hexagonal and octagonal shapes in our building. Some things look designed and weren't made by humans, and humans can't make them today at all. So a mountain and a river valley. The mountain allows the river to flow and feeds a growing ecosystem in the valley. But the mountain was not intelligently designed or placed there, nor was the river, and nor was the complex ecosystem that depends on both. They were formed by nature and time. So what about life and what about genetics? So DNA, I do not holds to a coding language, and I've asked my fiance, who is a programmer. He kind of agrees with me on this. Of course, that's anecdotal, but upon a brief look on Google, you'll find that very few individuals sort of equate them as a 1-1 correlation. This is because code does not self-replicate, and there is no natural selection acting upon it. But additionally, and perhaps more importantly, the genetic composition of living things is rife with redundancy. This redundancy is not typically functional and thus does not on the whole represent a fail-safe that has been designed. It's also very random. So how do we get life then? How would I propose that we get our nucleic acids, our proteins, and our lipids? Well, getting lipids, which of course are vital for abiogenesis, they essentially self-assemble into spheres thanks to their hydrophobic and hydrophilic nature. But they tend to destabilize in the presence of salt ions, which is why for a while people were like, hey, how are we going to get lipids? The salt in the ocean is going to prevent this even in prebiotic Earth. But lipids can actually form when carbon monoxide and hydrogen are heated and exposed to various minerals, which are typically found in the Earth's crust. We also now know that amino acids stabilize their membranes. So as seen in this paper, which goes much more in depth from 2019, we now have a mechanism, a plausible mechanism that is, for lipids. What about proteins? Well, in a similar way, we need proteins for abiogenesis. And to make a protein, a cell must put a chain of amino acids together in just the right order. But in 2019, it was found that the protein amyloid, which is abundant in nature, can self-replicate in the presence of carbonyl sulfide. Short peptide sequences shaped like amyloids do have what it takes to speed up the generation of smaller amino acid sequences. So now we have proteins. This is from a paper titled The Prebiotic Template-Directed Peptide Synthesis Based on Amyloids from 2018. What about RNA? We have a sort of plausible mechanism for the previous two. What about this one? It's also vital for abiogenesis, and it's composed of nucleobases and a ribose phosphate backbone. In both 2009 and 2017, respectively, researchers found that RNA can self-assemble in the presence of clays and a wet-dry cycle. So this is a plausible mechanism for RNA. To be very clear, I'm not saying that this is what happened. I'm saying these are plausible mechanisms. The physics behind chemical evolution is also well supported. We have two papers here, one which argued mathematically that the molecules in the presence of an outside energy source may reorganize to dissipate heat easier. This sees potential termination in organisms which dissipate heat incredibly efficiently. Essentially, we're seeing the emergence of life as a means to entropy. We also have a secondary paper that suggested that RNA and DNA are the most efficient of all molecules when it comes to absorbing UV rays, which would again help in the procession towards entropy. John likes to appeal to statistics, but do they support him? I have a cartoon here that says no, and I think Bugs is right on this. The problems with creationist statistics are numerous. First of all, and I'm glad that John presented a stat in the beginning that we can kind of go off here, but first of all, they tend to calculate the probability of the formation of modern proteins and bacteriums with all their modern facets, and they do so by appealing to random events. They also neglect the consideration of natural selection. Creationists tend to assume that there's a fixed number of proteins with fixed sequences for each protein that is required for life, and they calculate the probability of sequential trials rather than simultaneous trials. John shrugs this off in some of his videos over on his channel by saying, he kind of, in my opinion, hand waves it as if it's not a big deal that we have these indeed vast amounts of trials and innumerable almost into our modern brains time span, because we know that there are around 4 × 10⁻47 molecules of water in the Earth's current oceans. This means if we had 1 amino acid per million water molecules, we would still have 1 × 10⁻41 amino acids, which have the chance to interact over millions, if not billions of years. They seriously also underestimate the number of functional enzymes and ribozymes present in a group of random sequences. So anytime I see a creationist statistic, I always wonder, all right, well, is it meeting all of the requirements seen here on this list? And the answer, to date, is no. Conventional science statistics also preclude a young Earth creationist intelligent design perspective. In this first one, we overwhelmingly rejected both species and family separate ancestry with infinitesimal p-values. And in the separate paper, it says a non-evolutionary control model shows no convergence, and only a small number of parameters are required to account for observations. It is time that researchers insisted that doubters put up testable alternatives to evolution. Because that's the problem here. What I would like to see from John is a model.
You've got 2 minutes, Erika, 2 minutes.
Sorry, you scared me and I hit the button. Spooked me.
Oh, sorry.
No, it's fine. So is abiogenesis plausible? Currently, abiogenesis is the most plausible explanation for the origin of life. We have the mechanisms for each of the major components that make up the most primitive cell. Intelligent design, on the other hand, lacks a model, it lacks support, and it lacks a methodology. It has, through the years, reduced itself to attempting to poke holes in the hypothesis of abiogenesis and the theory of evolution. Speaking of evolution, I hope we talk about that because it's wholly supported by every relevant field in my honest perspective. This is, something that I've given a fairly deep dive into, but I would love to talk about it some more. It's one of my favorite things. So what John must do to convince me and the world of conventional science to which I abide is he must suggest empirical reasons for why the current explanations for the emergence of the components necessary for the most primitive forms of life are inadequate. He also must provide criteria for design as well as a means by which we can tell it apart from non-design. And if he wishes to push the young Earth creationist view found on his channel, he must invalidate radiometric dating, propose a means for delineating the kinds and why humans should be unique from the other apes, cope with the statistics that make separate ancestry veritably impossible, mitigate the heat problem and the various astronomic snafus, and explain the numerous other facets of geology and genetics that preclude a 6,000-year-old Earth. This is what I would personally like to see. I'm sure we're going to get into some good nitty-gritty stuff here. I also would like to tack on to the end of my presentation that I'm not a programmer, nor am I a geneticist. I study mostly macroscopic organisms, but I'm certainly going to do my best. I'll be the first one to tell you if I need some explaining on my end. and that's all I have to say.
Awesome. Thank you. Perfect timing with 3 seconds to spare. So we'll toss that 3 seconds into the much anticipated discussion portion. And thanks for the presentation, Erika. Once again, clear and concise and nice visual. So great presentations to the both of you. Why don't
try to mix it up, Standing? I like to keep it fresh.
I know how much work it takes to make these and put together these presentations. So good job, guys. Yeah, let's jump right into the discussion. I guess since you just finished your opening, Erika, if John, you want to ask the first question, I'll get the clock going.
Yeah, by all means.
Yeah, sure. So, I always find it very fascinating how folks taking your position and so often lead with our computer code isn't self-replicating. To me, that is a just stunningly ignorant position to take. And I'm not really sure how your fiance can reach that conclusion when what we're trying to do with machine learning in programming right now is enabling that exact thing to happen. And obviously that's before you even consider the fact that you can write a backup script that does do a copy. So I mean that whole premise of self-replication has zero to do with whether or not the information requirement is reasonably either created and then initially created or modified through copying. That's just one component of this whole deal. So I'm not really sure how that negates the premise of, and the other point you're making, which I'm leading to, which is it's not a code, it's just an analogy, which is kind of the point you were making, which I can directly counter, but I'd love to understand why you're coming to that conclusion.
Yeah, sure. Well, first of all, my fiance is a programmer, but also I did not show him my presentation. I showed him a previous debate that you had, I believe, with RJ. So he was informing me on the arguments in that conversation, not what's here today. I don't know if he knows about machine learning or not. But more importantly, even if code replicates, which I don't know if it does. If you're saying it does, I'll take your word for it. But I I'm
saying it can. That whole replication portion is like not a valid piece of like how does it self-replicates, therefore it's not like code in terms of the requirements.
That's a core tenet of any kind of mutation, any kind of evolution. It requires replication because it is during the replication process that change takes hold. So if your code isn't replicating, it's not subject to change and it's not subject to selection.
No, I understand that. The point I'm making is that to say that the requirements in the whole self-replication portion has zero to do with how those things I just mentioned originated.
I would very much disagree. I mean, from a conventional, maybe from your perspective, but from the perspective of conventional science, without replication, you're not getting change ever, and that includes the emergence of replication mechanisms.
No, you're missing the point in terms of the.
Elucidate me. Explain it like you're talking to a fifth grader. I'm not a programmer.
So you are operating from the position that the genetic code does not fulfill the requirements of a code, correct?
Yes.
Okay. So what are the requirements to be in information theory to be considered a code?
I have no idea what the requirements are to be considered a facet of information theory. But the problem is, if you're trying to make an analogy,
I'm not making an analogy. I'm making a direct comparison. Is the point that I'm making? You are making the point that it's only an analogy,
right? But John,
I am saying it's the exact opposite.
You don't have replication and mutation. That's just a fact of biology.
No, you're missing the point in terms of the root origin of the code that now self-replicates. The root origin requires.
So the mechanism is what you're speaking about. The mechanism.
No, I'm talking about the organizational structure of how the information is contained that is now you're claiming is being modified through self-replication. In context of abiogenesis, one of the requirements is that arbitrary values must be assigned to a codon to equal an amino acid. There's no way around this.
Suggesting they're arbitrary values. They're no more arbitrary than any value for atomic number is for any element. They're laws of chemistry. That's why it's called
no. You're missing the point. This is why I made it in my opening statement. I made the clear delineation, and this is why there's been many papers written on this of why it's the DNA enigma. I use that term from a paper from a atheist origin of life researcher. Because the arbitrary value assignment means it's arbitrary because the nucleotide base pair sequences of codon do not become the amino acid. They are symbolic representations of the amino acid. There is no chemical interaction between the codon and the amino acid. It requires the translation mechanism, which is the ribosome. There is no way around this. This is like there's papers written on this whole conundrum of like, this is the single biggest question about origin of life is how could this have possibly occurred?
I don't know about that. I spent the past couple of days kind of digging through abio papers, and there didn't seem to be like the attitude that you're presenting here is that it's like, oh gosh, sheesh, we're stumped. It's just not, that's just not the reality of it. But with regard to what you're actually saying, I mean, you can laugh, but it isn't. I mean, it just isn't. I would encourage everyone out there to go check it out for yourself. Do not take my word for it. Go look and see what's actually being said. And to me, I mean, it sounds like you're dealing with a lot of vagary here. You're basically
actually, I'm being very hyper specific.
No, I don't know about that because you're essentially from what I'm picking up. It sounds like you're essentially saying, where do we get the rules of replication from?
No. I'm saying, where do we get the rules for the information that is now replicated?
What would you classify as an information, though, John? What is an information?
Okay, so in this context, what we're talking about representative code base, symbolic code base of the syntax of the genetic code. This codon equals this amino acid.
Yes, so you're asking for why do things pair the way they pair, essentially.
Okay, do you understand that, like, the whole premise of semantics?
Well, yeah, I understand it.
Okay, and so a semantic, right? And the same logic applies to the genetic code. There is semantics and pragmatics that are derived from the syntax of the genetic code.
I don't argue that there aren't semantics or pragmatics.
Okay, so the semantics is immaterial meaning of the codon. How does the immaterial meaning get applied to this that is now being translated? There is no other code on the planet where anybody would argue that those translational values could ever be initiated and assigned without an intelligent agent. There's no other concept.
Why not?
Okay. So does the letter
explain to me what the problems of that are? I mean, you understand, like, from a perspective. Before you answer, I want to quantify myself so that you know precisely what I'm now that are capable of dealing in semantics and in pragmatics. Vervet monkeys are capable of picking up new linguistic words, so to speak, from one another and applying and altering the meaning to them. I don't.
Are the monkeys already fully formed life forms with intelligence?
I mean, it would depend on what you define intelligence as. I would call them fully formed life.
Do they have the ability to do information processing?
I would say to a degree they do, yes.
Okay, so literally everything on a cellular level is information processing. This is a well-established premise. Your brain, every single brain from the simplest brain of the lowest light form to us, is doing information processing.
No, it's not. It's not though. It's not information processing. It's response stimulus.
No,
you want to refer to
okay, Erika. This is literally. There's conferences on this whole thing of the information processing in biological systems.
How many of those conferences have you been to?
I've watched quite a few of them on YouTube. They're freely available on YouTube, and this is also discussed in.
So you watched them all then. All right. Deal. I would love to have a conversation.
So hang on. Earlier you talked about redundancy, right?
Yes, but
somehow it's a negative. Somehow it's a negative.
I would very much appreciate not being condescended to. I don't want to waste my time in a conversation where I'm being condescended to.
Well, you've kind of approached it from this whole perspective.
No, I have yet to have a single person complain about my discussion style, and you just seem quite angry. I don't know why. I would love to have a.
Actually, I'm actually not angry. I'm cracking up at somebody who's done as much research as you hasn't come across the things that I'm talking about, and then is making jokes about me not having attended a conference. I've only watched the conference. I say that's condescending in and of itself.
No, it's merely a response to the vibe that I'm picking up, but I'm keeping things cool on my end. I just am a simple request.
Okay, so you mentioned redundancy as supposedly being a negative, correct?
Absolutely. No. Let me rephrase that. I didn't, because I don't believe I said it was a negative. I believe I said it was indicative of non-design.
Okay. Well,
in the context of genetics.
Okay, so. Redundancy, there's actually been papers written on this in the context of how the redundancy of the genetic code is what actually enables translational pausing, which is a vital component to protein synthesis and actually houses a subset syntax which enables dual control, temporal control of the ribosome. And it's even been, analyzed from the perspective of does it qualify as a computer language? Yes. Machine code? Yes. It's a hexanucleotide code base. I mean, there's a whole, it's been mapped out.
Yeah, redundancy is a great thing. And sometimes it protects a cell from certain bad things from happening, but other times it's not present when it should be or where it would.
You're missing the point on the design factor. Okay. So we have a top level code base, which is the code on assignment for the genetic code in general. Then built in synchronicity from the same code base, you have a secondary syntax, which is now controlling the actual execution of from a temporal factor of the ribosome for protein synthesis.
Yeah, sure.
Okay, so you're suggesting that if you're saying that this is not indicative of design.
No, I'm saying redundancy from the most minute cellular mechanisms all the way up to in looking at entire genomes. We have repeats in completely random places. We should, if design, right? As a designer, I think we could both agree, or at least most designers would agree. I'm not a designer, so I guess I can't weigh in on that. But the hallmark of design is simplicity rather, and efficiency. That's not what we see in the genetic code, and that's not what we see in pretty much any whole genome you can imagine.
So that's not what we're starting to realize in the context of structural biology.
I don't think that's true, because I've looked at papers.
Okay, well, let me finish my point.
In the past.
Let me finish my point, because this is relatively recent stuff that's starting to come to the forefront. So things like the duplicate regions that are supposedly these examples of totally, redundant
location.
I'm saying throughout the genome. Okay. So in the context of data transfer and function, we'll use a top level example of a content delivery network, CDN, in the internet, right? So every major web host, Google, everybody, they've got multiple server locations spread throughout the globe.
Gotcha.
And you have multiple copies of the same, if you're using a CDN network, you have multiple copies of the exact same program on different servers in different geographic locations around the planet, so that loading time can be accelerated versus going to a singular source. Sure. The same logic applies to genetics, because the exact same genome is in every cell, obviously, except for the egg and sperm, obviously.
Right,
but
right.
the not every cell type is using every portion of the genome. So in different cells, different portions of the overall genome are exposed, if you will.
Sure.
So if you don't have the most top, highly used components that are easily accessible from various physical locales, then you're actually slowing down you have to unwrap the histones of another portion of it to go get that specific gene.
Right. I think I get what you're saying.
So to say that those pieces, I'm just talking about there's multiple layers to this, but just that from a top level portion.
Yeah, I think I get
to just count that is like saying that it's less efficient to have a content delivery network for your web app than having a simple or only having one server when there needs to be access from multiple points.
I get what you're saying, but that's the problem is, and I would agree with you if that's what we saw, but it's not. We have entire portions of the genome, and Standing and I have had this conversation previously, but like when you look at the functionality of the human genome and the functionality of other primate genomes, there are enormous, massive portions that to our current knowledge just don't do anything. And because of that, that's what I'm meaning when I'm saying, I'm meaning this from a surface level, because again, I'm not a geneticist. So I'm meaning it from a surface level of redundancy, where it's like we see these sometimes they're telomeric, but we see these same repeats over and over and over again. They have zero impact on phenotype. They're simply redundant. And what I'm saying is, all right, if you're a designer, right, that's probably not going to be the game plan, right? If you've got like a river and this river tends to overflow, you might build like dikes or sort of flood ponds or whatever, little reserves to catch the water as the river overflows so that it doesn't damage anything. But in genetics, instead of seeing like one or two in the areas where it might swell the most and overflow and flood the local region, you might have repeats for 6 miles in one area, just dike after dike after dike after dike all the way back, and then zero in another area where it floods all the time. So I'm talking about this weird redundancy that makes no sense to me from a design standpoint. Well,
let me address that.
Yeah,
so
go ahead.
There's multiple aspects of that. So one, say we don't know what it is, therefore it must be useless. I mean, that foundational premise is being eviscerated on a daily basis, especially in medical research, less on the Evo-Devo side, but much more in the medical research side.
Second, non-functional.
Sorry, what?
I said the fact remains, like based off of a paper literally in like 2017, it's still like the functional portion of the human genome is maxing out every time at 12 to 13%. And yes, you're right. They're finding new functions all the time. I do agree with you on that. The problem is the level of functionality for what they're finding is getting smaller and smaller and smaller exponentially. So we might find something new that a new gene does, or an ERV, or whatever.
What they're discovering, they're not just finding as many coding functions. What they are finding is the regulatory components that are altering and are controlling the outcomes, and that is expanding dramatically. Basically, pretty much every medical discovery has showed, oh wait, this thing that we were told wasn't functional, even a point mutation in it causes X genetic disorder. But where I'm going with all this is things like tandem repeats, short tandem repeats and such, that are viewed as, oh, what is up with this? Just repeat, the same thing has no function. What are they finding out now from a structural biology perspective? They're actually discovering that in terms of the DNA folding components that are X actions that are required for gene regulation, that those short tandem repeats, which have been being told have no function, actually serve as literal engineering components that act like springs almost, to enable the folding process for gene regulation.
Right,
but and they are also discovering that even point mutations to those short tandem repeats, which up until very recently, like the last 2.5, 3 years, have been considered complete junk. Actually, those point mutations, even in short tandem repeats, which have no function, quote unquote, are directly inhibiting the ability of gene regulation.
Right. So the thing is again, I have no problem with them finding that new things are doing new things, like new aspects of the genome are engaging in, helping with the transcription or what have you. I don't have a problem with that. But what I think you're missing here, and what I would wager if you went back and looked at those papers, is that the impact on phenotype is impeccably minute. And all geneticists agree, at least the ones that I've read about and in undergrad and indeed in my master's, is that for something to be functional, it must impact the phenotype. So if you went back and looked at it, I bet it would be quite small, because I would be very surprised if in the past, let's say, 2 years since Gar's paper came out, that they were coming out with an entire chunk of 25 to 50% functionality for the genome. And that's my point. It's not that we're not finding solutions for itty bitty tandem repeats, or maybe this section that we thought was junk actually turns out to do something. It's that the entire trend is redundancy, and the entire trend seems non-designed.
No, the trend with like the 4D genome project and other things of that are showing that there is massive, we're actually trending in the complete opposite direction of what you're talking about. I mean, and talking about phenotypes, for example, I mean, there's quite a few papers on the incongruency of phylogenetics and other things that are being used. But where I'm going with all this in terms of phenotypes and morphology is they're now realizing that things like microRNAs have massive implications in terms of body plans. Yet from the papers, it's literally saying, oh, these completely random, hyper-specific, but the outcome being hyper-specific microRNAs, oh, these may be the explanation for the Cambrian explosion, and all of these new body plans just popped into existence. So the point I'm making is that we're looking at things that supposedly had no bearing. Now we're finding out that very hyper-specific things that we are currently being argued to be completely random are now the result, or causing the new body plan outcomes of evolutionary theory.
I wouldn't be surprised if that's what they were finding, but again, that's not my point. I would
that's not what they're
100% correct that they're finding out that new microRNAs, new aspects of epigenetics, whatever, they're showing us how certain changes can come about. In fact, if were I more well versed in genetics, I'm sure I would find them very interesting.
Right. Here's the point that I'm trying to make here, though, is that.
Hold on, let me just finish. I was just going to say that it has to do with whole genome, and it has to do with what trend are we seeing. And there's no paper that I've seen, and again, because Standing and I have had this conversation several times, I stay up to date on that. There's nothing since 2017, or might have been 2018, with Gar et al. Coming out on the functionality, the as a whole functionality, which is my point here, of the human genome. It's staying very low. And while they find new traits, they do smaller and smaller things and make up smaller and smaller portions of the genome, i.e. redundancy.
Okay, I think you've missed a huge portion of my points here, but
explain it to me then. If I'm missing it, I want to know. I want to get your point.
What I'm saying is that what we are discovering with things like inter and intra chromosomal gene regulation is that the temporal factors and the physical location of genes is extraordinarily important, which directly indicates the foreknowledge temporal values execution that had to be in account before these new things could occur. So you're having to, the entire point of evolution is that there is no foreknowledge required, and it's all the result of natural selection. Mutation, sure. Natural selection, right?
Sure.
Okay, so your model has to explain how something that has to be in place before the other thing happens, right? From a time factor.
Yeah.
How that came into existence without it being predetermined.
So are you aware of precursor mutations? They're essentially the concept that it's like if you need A, B, and C to do something, typically A and B are going to be relatively silent in phenotypic expression, and then right. You have both of them, and they're just churning out the genome.
You're thinking about this from a linear.
And it happens.
So you're thinking about this from a linear perspective. This is having to occur from both a 3D and a 4D perspective. The linear changes that you're talking about inside of a sequence, are not even, we're not even talking in the same stratosphere.
I'm not talking about that. I'm talking about, like, for instance, the classic is like Behe or Meyer with the flagella, the irreducibly complex flagella. You need all 5 parts for it to work, and if all 5 parts don't happen simultaneously, you have something that's rather useless. But what multiple, including, I want to say it was Francis Collins, but I don't think it was actually him. But work on the flagella, and I could get you some papers if you'd like.
I'm aware. Ken Miller is the person you're referring to. I'm aware. I'm aware of the whole argument of it not irreducible complexity and stuff. That's not what I'm talking about.
No. You're talking about irreducible complexity on the scale of mechanism, are you not?
I'm talking about it in both the information sequence requirement, the temporal value assignment, the geographic spatiotemporal location.
Yeah, so time, where it is on the planet, things
no, I'm talking about right now. I'm talking about inside of the genome.
Okay.
So
yeah, you're right. We were on different pages on that.
The cascade problems of frame shift readings are catastrophic in almost every instance, right? And what is being realized in the context of structural biology, specifically in this context, what I'm talking about right now, of the interchromosomal gene regulation and inter chromosomal gene regulation, is that the introns, the exons, the promoter region, all of those things have to be in a specific geographic location, even if the sequence that's actually being executed is not in a linear connection to those factors.
So I don't understand why you have a problem with the solution that numerous biochemists and sort of geneticists are proposing, which is a sort of stepwise and precursor mutation filled solution. What do you think, that those just don't happen, or do you just not find that compelling, or what? Why don't you like that solution?
The correct order inside of the sequence. They have to be done inside the sequence in the correct order, right?
Right. Yes.
There has to be no modification. Okay, so we've got base pairs 1 through 10 are in the correct order. Then as 10 through 20 are forming, number 2 changes. So we're going to say the first 10, even though there's no known function at this point that's being conserved through natural selection, somehow the first 10 say perfect until we reach the other 300 that are required before it's a functional protein. So one, just from an improbability perspective, the math on that is just insane to actually think that results in a new functional protein. But that's before you even account for, not only does all of those have to be correct, in order for that new, supposedly through natural selection, new functional protein that has come into existence from scratch, is now only executed through gene regulation factors that have to have also been forming at the same time, or come into existence at the same time in terms of this new protein that's in a different chromosome that has to be folded over physically to bind with the promoter region of this new sequence.
I see where you're coming from.
You're having to say that all of these things are happening.
I get you.
I was on the same page to ultimately reach the end conclusion.
Yeah, I wasn't with you, but I think I am now. I think that, and I'm sure you probably get this a lot with the kind of style of argument that you take with this kind of stuff, but I really do find that it's a bit of an argument from incredulity from some ends. I'm sure you get that a lot. And I don't know the details of this specific type of protein or mechanism that we're discussing. I would have to be probably a master's or PhD student in that to understand the inner mechanisms. But with a lot of this stuff, it's like I mentioned in the slide that I had on my presentation about how creationists kind of cope with statistics. I think a lot of times what tends to be missed, what tends to be lost in the ether, are the relationships between these various parts and how things are selected for. It's like if I had an alphabet of A to Z, right? And it's like I'm spinning a dial on 26 different wheels, and I want them to be alphabetically in order A to Z, and each wheel has all the letters. From a natural selection standpoint, indeed, with things like proteins, which are hyper necessary, and I suggest you take a look at that amyloid folding paper, or amyloid protein paper that I had on my presentation too, which kind of covers a little bit of what you're saying, I think. But if I spin all those wheels and I get, A in the right place and P in the right place on the first round, according to natural selection, those two get to stay. Like, that's kind of what this is a very simplistic explanation, but for things like things have to be in a specific order at a specific place at a specific time, you're not spinning all the wheels every single time you run the die. Like, the ones that were in place, they get to stay. And furthermore, you've got trillions upon trillions upon trillions of different wheels being spun all around the world, or around the location of, if we're looking at a genome in different places on the genome, each and every time. Through of course your time, whichever time span we're working with. I'm assuming we're kind of going back pretty far here. So to me, I hear that and I'm like, wow, those numbers, they sound pretty impressive. But when you consider the laws that typically govern nature, and from my perspective, from contemporary science, how much time and space we have.
I love it when evolutionists bring up the time and space variable, because number one, per your own evolutionary timeline, you have no greater than 500 million years and potentially as short as 200 million years for the first form of life to exist.
No, 3.8. 3.8 to 4.1 for the first life form. That's billion with a B.
Years ago that it came into existence, which in terms of the 4.54 billion years from the formation of the Earth per your timeline. When the form of life came into existence, we're talking about a total of 500 to 200 million years per the evolutionary timeline for the first form of life to come into existence.
Oh, from the origin of...
You don't have billions of years to work with for the first form of life to come into existence. You have no more than 500 million per your own timeline.
You're right.
So the improbabilities of all this and the complexities are not in your favor. Secondarily to all that, the point you make about, oh, it's happening all over the place. Cool. I could care less about that. Because at the end of the day, all of these variables must come into existence, be in existence at the exact same time, in the exact same physical location. It doesn't matter if one of them happened over in Australia geographically.
Oh, sure.
They all have to be, they're molecules, right? They all have to be in the exact same location.
I'm with you, but I want to insert something there, because all of the recent studies that I've seen have RNA world or metabolism first, whichever origin of life you're going with, occurring in very concentrated patches near hydrothermal vents. So the odds of getting, first of all, we're dealing with concentrations way above what we see even today. And there are some excellent papers that from 2018, 2019, very, very recent, sort of expounding upon that I probably can't do here, at least not to do it justice. But so we're not dealing with an entire open ocean of chemicals just sitting and stewing around. We're talking about vastly concentrated with minerals and amino acids areas close to these warm hydrothermal vents, where these trials are going on by the million, by the billion through time. So you were right. My math is not the best. So you were correct on that it's more like 200 million to, I think I would say, if we're going with a 4.8 billion year old planet, which some are suggesting, you can have up to about 1 billion years. So with all of that in mind, near these concentrated areas makes it more likely for these things to bump into each other. I don't get it, John. I'm not with you. I don't find that incredulous.
Do you not realize that the chirality problem?
Oh, John, come on. The chirality problem, that's, you gotta be, I'm not saying you in particular. I'm just saying I've heard that argument from a lot of creationists. And
chirality problem is a recognized one in origin of life research.
Right now, if you go to, there's a meta-analysis on all of the abio studies that was released in 2015 or 2016, maybe, along those lines. It covers the chirality problem, and it offers up 3 or 4 different solutions, all of which seem, in my opinion, again, not a geneticist and not
so you realize that
highly plausible.
That to overcome many of the issues like the biofunctionality problem, the chirality problem, all that stuff.
I don't know the biofunctionality problem. Explain that to me.
Let me finish.
Okay.
These things are overcome in all observed life by enzymes which account for these factors, like the polypeptide bond not forming correctly, and you have a 50-50 chance each time that it's going to form a correct peptide bond and in the amino acids. If they don't, you fail. You have to start again, which is why the improbability factor, which is one of them, is in the fact that the likelihood and it being a plausible conclusion that a protein that's 300 or 400 amino acids long, when literally every bond has to be correct to reach the functional protein. And every single time, this is one of among many, it not only does it have to be the correct amino acid, the bond also has to be formed correctly every single time. And so you have to start again. So your whole aspect of the linear fashion, it has to be correct, all of them incorrect at the same.
I'm with you.
There can be no errors. So you're starting from scratch every single time.
I'm with you.
You made the point of oh, we're by these alkali vents and such, right? Well, from a chemistry perspective, you have to account for the fact that there's different stages from a temperature perspective. You also have to account for the reality that without the protection mechanisms of the cell membranes and things of that nature, as well as other enzymes, both the heat and the sunlight itself would be causing the collapse of these structures on a very accelerated basis. Also, you have the lack of oxygen per the evolutionary timeline and worldview, prebiotic worldview.
Yeah, but the
oxygen. So you're also not being able to block UV radiation, which also accelerates the destruction of these.
Here's the thing, though, John. Because you've said quite a few things, I want to make sure I don't miss any of them. So first of all, I read a paper just this morning in preparation for this debate suggesting that the conditions for life to form in an abiotic fashion from chemical evolution is astronomically more likely when oxygen is low when compared to when oxygen is high, because it turns out oxygen is just kind of a nasty element to deal with when you're a budding, microorganism near a hydrothermal vent. So one, oxygen is not going to be our friend, which is why it certainly is a good thing that according to the rocks it was very anoxic condition wise. Second, your lipid cell membrane is what's protecting you from the UV radiation, and lipids, as I pointed out in my presentation, have been shown in media res, like in real life, to form naturally near hydrothermal vents. The crazy thing about lipids, as we know, they have a phospholipid bilayer, and they
let me clarify that piece on the lipid point you're making. Which the example you're using is actually the replication of a already existing one. They took I believe it was 42 amino acids in length, that particular lipid you're talking about.
I'm not talking about I'm not talking about proteins. I'm talking about lipids. They're just phosphate head and the little tail. They make up.
I said.
No, it's okay. I get it.
I've read this paper.
Yeah, I've done this before too. I get it.
The self-replication was only occurring from one that already existed in its complete state. It didn't just form and then start replicating. They took one that already existed, dropped it in that environment, and then it began to self-replicate.
I'm not talking about any, like, lipids don't, to my knowledge, self-replicate. Like, I'm talking about lipids, the little guys that form the cell membrane by coiling their tails together and their hydrophobic heads go on the outside. They're not, when I'm talking about these guys appearing near hydrothermal vents, they're not replicating, they're forming. They're found naturally by hydrothermal vents by the heating of carbon monoxide.
Okay, we must talk about different papers. Because I've read something exactly on this whole thing, and they were talking about how they had to take a one that was formed for this whole thing, this process.
No, this is naturally in hydrothermal vents today. Like that, you couldn't go, and I don't think it would be you would not want to get too close to the hydrothermal vent. But this is actually just taking samples from hydrothermal vents and seeing if there's lipids, and then there just are. So that's my point. So already in this natural environment, you're getting an encapsulation that's going to protect you from the UV rays. Check. The second weird thing about lipids is they can only maintain a stable boundary if amino acids, the basis of proteins, are present. Otherwise, they break apart in ionized water, aka salty water. So the fact of the matter is, and we know that amino acids are present near hydrothermal vents because we also find them near hydrothermal vents. So when the two are in the presence of one another, essentially what you get is a lipid membrane that surrounds amino acids, and you've got two of those first steps already there. All you need is RNA, and RNA coincidentally involves itself a lot with nucleobases, and you're going to need amino acids there too. So my
hang on. So I see where you're going with this whole deal, but the point you were making earlier about this, we're obviously going with the RNA stuff, is, we've seen it form on clay and such. This, and there's been papers on this whole thing of how cool RNA molecules will form on clay. This has zero relevance to how the syntax, semantics, and pragmatics value assignments happened, and then the components that are required for its transcription. The whole, DNA is this DNA molecule can self-replicate has zero value in relation to it resulting in a protein. I mean, there was a paper written that was published last year about how, hey, we finally have to admit that the RNA world hypothesis by itself is not valid, and we have to have proteins, transcription proteins, in existence at the same time.
But I think that's I think that's, just to intercede quickly, and I'll let you go on again. I think that's a little bit of and I don't mean this in a derogatory way, but a bit of a quote mine, because as far as I know, biochemically speaking, most people aren't gung-ho for RNA world right now. It's mostly metabolism first, which is why it would make sense to see in a paper someone being like, RNA world's just not going to cut it, because now we have a better hypothesis. I'm sorry, continue.
I mean, that switches up on a regular basis. It's gone from protein world, RNA world, metabolism world. I mean, it bounces around
as science should.
By like crazy, and all of them keep running into chicken and egg problems. All of them. And
I don't know, though.
Okay, hang on. They keep running into chicken and egg problems. This is a well-admitted premise in many of the papers. They keep running it here. Okay, this is why I think this one's the best. Here's all the problems with yours, but ultimately they all run into the same chicken and egg problems. Which at some point, all of these variables have to exist simultaneously in a structured format with value assignments to the different components that can be transcribed, translated, and executed.
For your value assignment thing, and I'm sorry, were you done? I didn't mean to interrupt. Sorry. So, did you say anything else on that?
Yeah, I mean, the point I'm making is unless you are willing to conclude that, for example purposes, in binary, the letter A equals 010001. Okay. Is there any context in which, without a intelligent agent assigning values, could the A ever be equal to 01001?
I think that here, because I heard you talk to Speed of Sound of Gravity on this, and I think we might just have like a fundamental disagreement on, because I'm assuming that from your perspective, the laws of chemistry are also intelligently placed where they are.
No, the laws of chemistry have zero relevance to what the arbitrary value is of the codon to amino acid. For case in point, in human technology, as we speak right now,
sure,
we are using DNA and the chemical process of base pairs and ribosomes and everything to both store and execute information and programming functions. Okay, I don't think there's anybody with half a brain on this planet who would argue that the processes that are being executed through chemical functions and reactions inside of DNA for the information that we're storing in it and the arbitrary value assignments of the syntax that we're using, just because the medium is DNA, does not constitute value assignment.
And I think I'm getting what you're saying, like conceptually speaking, but I guess I just don't understand why value assignment can't be assigned through the trial and error of natural selection and numerous trials along the way. And what works. I mean, I don't understand.
We have start codons, we have stop codons.
Yeah, right. And how does DNA conduct its business though?
If
chemically.
For the execution of it. Okay, so do you agree that a gene sequence and the base pair organization of which it is organized is equal to the required organization of proteins that result in or amino acids which result in a protein?
Yeah, sure.
Okay, so genetic code sequence A equals protein A.
Yeah, I would agree with that. I mean, that's pretty much what we see.
Okay, so in order for that to happen, that has to be a translation mechanism. So you have transcription from DNA into mRNA, a bunch of other stuff that goes on in that process. Then it goes to the ribosome, and then that transcript is translated to equal those proteins. Those proteins are put in the correct order, and now you have protein synthesis. And of course, there's other stuff that happens, chaperones and stuff that happens after the fact. In the context of direct comparison, we have a 3D printer that is executing a program to result in a physical object. And this is why, in the context of like translational pause, and the whole point I was making in what the significance from a design perspective of the dual syntax is that mRNA is acting as both the elements needed and the order and the time of, just like you would do on a 3D printer, of, hey, the aluminum needs to go in this angle, this time, pause, next, and then you're fast forward, you now have a three-dimensional object. That's exactly what's happening in the ribosome.
Well, it's It's not, though. It's similar. I mean, there are comparisons that you can draw. I agree. There are comparisons that we can draw from a human archway to a natural archway too. But for me, the problem that and I realize that's a rudimentary comparison, but to me, I just don't understand what is difficult to kind of not conceive, accept would be the word that I'm looking for.
Okay, so
Let me finish my point. So when we look at DNA, how does DNA conduct its business? It does so chemically. Yes, there is a spurring, there are instructions, so to speak, and I use that term colloquially, but when it gets right down to it, DNA responds to chemistry. That's why we can impact it chemically. That's why it can mutate under the influence of radiation, which can cause various changes and whatever in the genome. But my point is that it's all right, if DNA currently conducts its business through chemistry and physics, why is it so hard to conceive that initially this was also what happened under the sort of guidance? I realize that's anthropomorphizing it of natural selection. So what works sticks around, and what doesn't, well, the process up until that point collapses. You're right. You have to have a lot of things in order. In order for something to
let me directly address your point there. So I think you missed it because I've already addressed this whole chemistry is not relevant to the medium and the process. It's not relevant to information.
I get what you're saying. I just don't.
So the point I'd like a direct answer from you on this. So the first thing that was stored, one of the first things that was stored in human usage of DNA for information storage was a picture of a horse, crossing a finish line. Do you think it is remotely plausible that given an infinite amount of time, an infinite number of universes, and an infinite number of chances, that somehow the data in the correct order of genetic syntax and nucleotide order could ever result in being able to be read as a picture of a horse?
If you're selecting every time one gets it right, then yes. Why wouldn't it? With infinite time, especially. I mean, from my perspective, it's I don't understand why. I feel like we're going back and forth. I'm saying yes with selection, and you say, but how's it all going to happen at once?
No. Okay. So first, you have to account for the data, but you have to account for the syntax that can be read to equal the correct first point of the picture of the horse.
Right, but why can't syntax come about naturally? Do you think that for every organism,
again,
from the littlest ants to the termites to the vervet monkeys to the geladas, whatever, every organism that communicates with one another wasn't taught syntax. They understand things by chemistry and by vocalization. So I don't understand, and the reason partially
how are they able to do so? They have the ability for information processing, which you're claiming doesn't exist, but is exists in every single form of life.
John, I'm saying that information processing is not necessarily, nor does it actually, I wouldn't even say is not necessarily, I would say just strictly does not need an intelligent input. It's response stimulus, it's selection, and this is my thought. I'm agnostic. If you want to sit here and tell me that God spurred life, sure. Then I'm going to say, what the conversation I would love to have with you in particular is more to do with what you think after life started, but it's the whole fruit of the poison tree. I get it. You want to have this, we want to have this conversation too. I think it's an important one to have, but I just don't buy it. I don't think that it's that incredulous.
Okay, here's what I'm just really not comprehending is you think it's natural that if the genetic code and AUG equals start, right?
Sure.
That is a completely subjective and arbitrary meaning that has to now be interpreted. There's no way around this. Okay. So you're claiming, this could just happen naturally. Okay. Is there any scenario in which you think that without an intelligent agent applying the value, that the letter A being equal to 010001 in binary could ever occur naturally?
Again, I feel like, and I'm understanding where you're going. I think that we're reaching a fundamental level of what do we accept?
Do you think that could ever happen through a natural process without an intelligent agent? That's a yes.
Again, I mean, you've asked me this before. I think if we have selection as a player in the game, and again, I think that we do.
You don't have selection yet. You don't have selection
yet. Why would you not?
If
it doesn't do anything, it doesn't do anything. If it does, it's automatically selected for. That's the most basal level of evolution.
Okay, so we're We're talking about how do you get to the point of even to be able to have natural selection.
Right.
Right. So if you don't have this process yet, and in order to get to the process, you have to have this as a predecessor. You have to have a start in protein synthesis. If the stop and start codons are not in the correct spot, the protein does not form. That is a fact, right? There is different start and stop codons in different types of organisms. Unless you have those factors along with the codons which equal the specific amino acids that are going to be done, you will never get a protein being formed. Period. There's no way around this.
So your barrier to it is the fact that there are seemingly arbitrary, and I'm saying seemingly just because, again, this isn't my area. Maybe they are arbitrary. I don't know. But you're saying that these seemingly arbitrary values have appeared and they obviously cause something to happen. Since there's no conceivable way to understand why it is that they're interacting with one another, someone must have done it. Am I gathering that correctly?
I am suggesting that ultimately,
in a simplified version, though.
Ultimately, yes. But I'm suggesting that when you have arbitrarily assigned values, which must be the result of sequential codons and nucleotide base pair sequences, and a ribosome that is able to interpret them as the appropriate amino acid, and you have to have the tRNAs already in existence, and you have to have all these other proteins for transcription. That's just not remotely plausible that all those things could ultimately be derived without a external agent. But in the context of the DNA portion, RNA portion, all that stuff, when you actually look at it from a chemistry perspective, there is no chemical bond between the base pair nucleotides, in between the pairs. There's a hydrogen bond between the two bases, and there's a bond to the phosphate backbone of the double helix. There is no chemical bond between the base pairs. So you have an entirely random. There is no chemical reason for those base pairs to organize in a specific sequence. There is none.
So essentially,
you're dealing with 100% randomness. You're dealing with 100% randomness in order for the sequence, the DNA to somehow end up in the correct sequences before you even account for the process by which the start and stop codon and the amino acid codons are now delineated.
Right. So the rules
you get to the factors that are required for it.
Yes, I'm kind of gathering what getting what you're saying.
So where I'm going with this is you are coming at this from the perspective that somehow through random chance, all of these variables, we're talking about hundreds of thousands of them that must be in the correct order without flaw in order for life to exist. And there could have been if you got these certain portion correct, oh wait, they all stay, even though we're not to natural selection yet. I have to get to thousands and thousands of things all in the correct order for somehow the spontaneous generation of the proteins that are now going to be able to transcribe this and translate this into functional proteins to now expand the process. And the point I'm making is when you start to really dive down into things like, as the example I made in my opening statement, for the recognized simplest form of life, you need 580,000 nucleotide base pairs in the correct order. That's from a secular paper. That's not. But
John, you're using an extant organism there. I mean, every paper that I've ever read in regard to abiogenesis, though, has spoken on the fact that the most basal organism, the very first thing that would have appeared under this lens, and I want to touch on some of the stuff you just said in a minute, but this one was I noticed it in your presentation. It's nothing like anything we have here today. I mean, this organism, hold on, I wrote it down. Mycoplasma genitalium is just as evolved as we are. It's still here. It's still been undergoing selection and change. There's no reason to even posit that the very first organism would have been even as advanced as that. So
they're recognizing this as the minimum requirement.
No, this would not be the minimum.
No, they're postulating on some of these proteomes that they're are not the equivalent. The point I'm making.
We're postulating it to a point, though. Like we're postulating it to the point that it's like, if we had one, there are billions of, or rather millions, 2 billions of organisms alive today. Therefore, the bare minimum for the first life must have been, at least some rather large fraction of that. It's like the origin of life is something that I will readily.
So pretty much everybody agrees that you have to have a helicase.
No, I'm with you. I'm just saying.
Okay. So do you understand how big
organism when it comes to this kind of comparison? Extant organisms have almost nothing to do with origin of life.
So the self-replication factors are ultimately at the bare minimum, and this is Kunin, I believe, talked about this, and he also talked about why it's so stupidly implausible to think this could happen without or just by chance. Even he's an atheist, by the way. You have to have a helicase or a helicase. You have to have ribosome. You have to have DNA, and there's like 4 or 5 other enzymes and functional proteins. You have to have protein machines. You have to have in order for self-replication to occur. So, as an example, just the helicase is 1,000 amino acids long. 1,000. That's just for one component of the transcription factor requirement. You have to have, so that is going to equal no less than 3,000 base pairs sequences in the correct order for at the bare minimum for the helicase to come into protein to come into existence. That's before you account for the other components. So now you're easily looking at thousands and thousands and thousands of base pair nucleotides, which can have no errors. They have to all be in the correct sequence. But now you're stuck with the chicken and egg problem of, okay, wait a minute, that helicase that I need and the polymerase and the other things that are going to be used for the transcription, the sequence is in the DNA, but those things don't exist yet. So how in the heck does the transcription occur? That's why it's the ultimate conundrum, which is why it's like, okay, so I have arbitrarily assigned values, I have information, I have prescriptive information that is required to be executed in a specific order by a specific process in order to result in this functional protein. But without that prior existing functional protein, it can't occur. Therefore, I have to argue that somehow the functional protein somehow came into existence in order to enable that transcription process to occur. And
right. Again, though, like I 100% am down with the idea that it's like, all right, abiogenesis with one single organism in one single location with everything being able and ready to organize in that specific way seems unlikely. But again, I keep feeling like we're pulling back to this singular case, this one sequential event, when in reality it's occurring far more times than that.
Even
hold on.
Erika gets equal time with one.
Yeah. So again, I get what you're saying, and I'm with you on the idea that it's like in that aspect, it would seem rather astronomically implausible. But again, because of the massive amount of time and space that we're looking at, and again, I'm very clear on the specifics on how it's not simply the organization. It's not simply the right place, right time. You also have to have these applied rules to them. And I guess the thing that I'm struggling with, and perhaps it's just because I'm not a biochemist and I'm not a programmer, but I don't understand why these laws, these rules, these semiotics that are applied to non-intelligent organisms seem so far off as something that could have emerged by chance, given to create even a single organism, all of these things have to be present. Yes, they're in a concentrated area with all of many, in my opinion, if not all of the things that they need right there. It's still going to take a while. That's why it did take so long. But in real life examples, and from a sort of utilitarian standpoint that you were using earlier with the mycoplasma, there are numerous scenarios today and again, I use language as an example, where organisms that are unintelligent behave in an intelligent way and can acquire new means of behavior in unintelligent fashion. And again, it's because of natural selection. And I'm with you, selection isn't acting on something before it's a thing, but in some ways it kind of is.
So what you're talking about in terms of learning a new function, that is the direct result. It doesn't matter if it's a lower level, quote unquote, lower level life form that has some lower level of intelligence. It still already has intelligence. That's like saying that a human learning how to play a piano is not the result of intelligent application of information processing functions which are executed through the brain.
Wait, though, you're confusing, or rather conflating, intelligence with response. Like, obviously an amoeba is not, or any kind of microcell or very small organism is not going to be intelligent, yet you can train certain, I guess, even fungi to go through a maze, to grow their way through a maze, because they respond to the stimulus around them.
Right. And how do they respond? What causes the response?
Well, a lot of it's chemistry. I mean,
it's sensors. It's sensor organelles that already exist and are the result of
sensor organelles are picking up on chemistry.
Correct. That's not so the manner in which they're being able to do that is requiring nanotechnology that is housed in the instructions and the execution of which.
How is that nanotechnology, though? It's just chemistry. Chemistry occurs whether an organism is living or not.
No. Okay, so those sensors, those organelle sensors that are, if you blew those up onto the macro level, those would be incredibly complex technologies.
Well, there are many. The Giant's Causeway is a very complex geologic formation that's been subject to a lot of chemical and physical.
The rocks cannot determine and process if they should go in the direction of.
They respond to stimulus.
No.
And if there's the presence of selection.
Sorry, are you actually trying to equate life to rock?
No, what I'm saying
because it's a well-defined element that there is a tremendous difference between organic and inorganic.
Yes, no, I would agree with you. But
okay, so then don't compare it to rocks then. An organelle
very specific
to rocks.
actions. What it is it's chemistry. You just admitted that.
No, I'm actually directly
organelles, but it's still chemistry. That's what you just said.
Okay, no, I
is
said
it not? Correct me by all means.
Are you going to stop interrupting me? Okay. The organelle is a sensor. Okay, it's a technology sensor. Okay, what you're arguing about, it's just chemistry, is like saying that when you hold up your camera and it is able to sense light, that there is nothing going on in order because it accepted light. That's what you're talking about here. The organelles that we're talking about that are just sensors on the the membranes and such, they're enabling these processes. They're nanotech. They're being able to read. We have
literally just not though. Like the mitochondria is just undergoing chemical processes. Like so is any level of cellular biology.
Okay, so you don't. So you think that the ATP?
Equal time, guys. So
are you arguing that
America? And then go ahead, John.
Are you?
So we're kind of talking over each other there. So
no, I get it. It's fine. Tensions go high in these kinds of debates. Yeah, I just outside of John and. Actually, just outside of John, I've yet to see anybody in sort of not even in the bioinformatics realm as of late, like past 3 years, refer equatably to DNA and sort of a computer code, or more specifically, an organelle and nanotechnology. I think it's an original take. I just don't think it's being.
So I don't know what you're reading, because proteins are directly. Called nanomachines directly in plethoras of papers in very recent history. Like there's no argument inside of academia of whether or not
you're defining it, though.
Okay,
does it not?
So are you suggesting that ATP turbine is not a nanomachine?
It depends on how you define it, because the Golgi apparatus is technically an apparatus. Does it mean it's the same as a crane? No. Like they're weird ways of categorizing things, of course. But I bet if we sat down, the two of us, and we decided to Skype with a bioinformatics or biochem guy, and we were like, "Hey, do you think it's fair to equate an organelle to a nanobot?" They would be like, "No." Like
I will take you up on that. Let's go find somebody. I'll bet you $100 that I can go and find things on YouTube right now from conferences said by biochemists. That yes, hey, these are not just chemical reactions; these are literal nanomachines. Do
you think they're being hyperbolic?
No, because I've also read papers where they talk about how it is not hyperbolic; it is literal.
I would be really interested to see those. Again, I am open. Okay. This is a subject where I'm just open to being proved wrong, because again, this is not my wheelhouse. But I very much doubt that.
So let me ask you this: so the transport proteins, for example. That use a literal, they bind to microtubules, but they use a process by which they literally walk. And that process was actually copied in a molecular nanocar race that was done back in 2016. They actually looked at the process by which they were able to make this step process occur on the molecular level and applied it to their nanocar. And then they expanded that into creating they looked at the ATP turbine and used the exact same logic to create a axle with molecules that can turn and bind powered by photons. Exactly like what's going on with the ATP turbine.
Yeah, that's super cool. But the thing is,
hang on. So is the molecular car, which has structure, it has turbines, it has wheels that are powered in the exact same process as by the ATP turbines proteins, is the nanocar, is that nanotechnology in a machine? Yes or no?
is the nanocar in an actual machine?
Is it a nano machine?
I would say the one made by humans. Yes, it is a nano machine.
Okay, so if per the admission of the same people who are creating these kind of things, that theirs is not anywhere close to as efficient as something they were attempting to copy, and it is so much more complicated to build from a chemistry perspective that no, we don't know how to do this because there's so many steps in the process that have to be done in a controlled fashion with specific enzymes and all those kind of things in order to be able to execute what we're seeing in biology. I don't understand how you can conclude that one that has the exact same types of functions, right? This one's not technology, but this one is because it was created by a human, even though humans admit that this other one is way more high-level technology than the one they were, from a capacity perspective, than what we were able to build.
Because the fact that humans can't recreate it verbatim, to me, is almost more supportive of the fact that it is not designed. The fact that you can create something that looks we can create, macroscopic versions of, or rather large scale to scale versions of a heart, but that doesn't mean that it can do all of the same jobs a heart can do, or a liver, or lungs, or whatever. To me, that doesn't necessarily state that those things are designed, but rather they're impeccably complicated. The fact that things are complex and that we can't mimic them, to me, seems like a ridiculous comparison.
Okay, so talking about the heart. Talking about the heart.
So for a minute too. So if we were to say, all right, what's a really a perfect example would be like if we wanted to build a very high vantage point. We're like, wow, trees grow really tall. We want to be able to see really far too. And so we try to make something that's super flippin' tall so that we can see as far as we can. Well, Mount Everest is gigantic and has an enormous vantage point to it. The fact that nature made it on a massive scale and that we can't reach that scale, be it because the scale is high or the scale is low, says nothing about its ability that it's designed. The hallmark of design is simplicity. And it takes it back to my entire original point, which was the code is redundant. Now, I couldn't tell you, and I will be the first one to tell you right now, you've given me a lot of stuff to research with the evolution of actual mechanisms of replication and transcription, because I can't give you great answers because I don't know them. But what I would say is that I would be very surprised if I got off this call with you and I went off and I found a bunch of scientists with their hands up saying, must have been intelligent design. I would wager that there are mechanisms proposed for these semiotics that are out there that I don't know about because this isn't my field. But what I do know is that while I can't answer you that question, while I can't tell you how semiotics came about, that the answer is out there. I don't think you can say the same to me, about why redundancy exists in designed organisms.
Okay, so number one, the redundancy that you're referring to, I've already addressed that because we're discovering that it's not negative. So many of these aspects are not negative redundancy. They actually have direct function, number one. So you can either accept that or reject it. Doesn't change the fact that it's true.
Just the papers just don't say that. I said the same thing in my conversation with Standing. Unless you're about to tell me the strict reasons why Grahr et al. In 2017 is incorrect in his assessment of the functionality of the human genome, then I don't know what to say. Maybe there are papers out there and you have them and I haven't read them. But I've heard you say there are papers, but then you haven't given any.
Okay, so as I've told everybody, I have an open folder, and there's papers in there literally titled these things. You can go look at them.
Okay, chat, get on it. You guys got it. Find if you would be so kind.
Hey, I let you talk. My turn.
Yeah, your turn. Sorry.
Okay, so a minute ago, you tried to equate a mountain and us not being able to recreate a mountain as being equivalent to us not being able to recreate something from a biological perspective, and then you finished up with, a hallmark of design is simplicity. Well, that's a very naive perspective, especially when you think about hypercomplex functions. Just because it is complex doesn't mean it's not the most efficient model in order to reach the desired outcome. And just because you don't know how every piece of it works, doesn't mean that they are not having functional outcomes. But in terms of optimization, there's been studies on this that, like the genetic code, for example, which actually is a code. They did a whole study on the fact that it's a code, and the whole purpose of the study was to determine whether or not the genetic code was the most optimal one. Of the million different possible variations they studied, our genetic code happens to be the most efficient in data storage. And they also found out that the redundancies and the different aspects to it also made it the most robust. So we actually have one of the most efficient options. That's interesting. Beyond that, we have things like ion channels and different functions inside of the body, which they are optimal. There is no greater optimization possible.
I don't doubt it.
So we're arguing on one side, hey, this is all a bunch of crap over here, and there is no hallmark of design. Over here, we have something that cannot, literally, from a physics and chemistry perspective, cannot be better.
But why isn't it all that way, John? Why is it random what's optimal and what's not? Why? It's my example.
You're making the arbitrary. Okay.
Why do you have 80 dikes here and 0 where it floods the most? Why aren't there 100 different things that prevent us from getting tumors? Why is it the way that it is?
So again, we are literally, there were papers published. I've mentioned this in multiple debates. There were papers published in January of this year in Nature on the same day, 17th of January, I believe, all about medical research, about cancer and different genetic disorders. And in multiple of these papers, the authors were saying, okay, basically, I'm paraphrasing here, but it was pretty direct language. It was basically, hey, you evolutionary biologist idiots who are consumed with Neo-Darwinism, stop claiming that this has no function, because we're finding out that it does on a daily basis. And the reason we don't know more about it is because you guys have been arguing that there is no function. And then if we find it, it will negate your entire theories.
So let me be very clear on
I'm not done yet. Hang on. They literally are saying things such as, the reason we don't have the technology in order to be able to better document and discover is because there hasn't been the funding to do so. However, now we've made so many discoveries that there is massive amounts of money that's going into it. I think there's a project, I forget the name of it. It's like the secondary level of ENCODE. I think it's got like $4 billion to go into this, because they're realizing all this stuff.
Allow me to respond. Let me get this. I want to make sure I'm getting you 100% clear on this. Are you telling me that this paper, when I go and search it, are going to say that the functionality of the human genome is, Brar et al. In 2017 is wrong, and actually the functionality of the human genome is far greater, and when I say far greater, I don't mean a few percent, far greater than what we have always said it to be? I want to be very clear on this, because I'm about to go read this paper top to bottom, baby. You're telling me to
I am directly stating that there are numerous researchers in a variety of fields.
In this one paper?
Okay, apparently you're not actually listening to the words that are coming out of my mouth.
I'm doing my best, but a lot of it sounding like gibberish.
Well, your ignorance of some of these topics is becoming readily apparent in regards to a lot of these topics we discussed.
You got to put up or shut up on this one and show me this paper that's overturning all of genetics. I really want to see it.
So number one, I never stated it was overturning every aspect of genetics. What I stated, what I stated was that there are numerous papers who are directly, the authors of which are directly stating that there is way more functionality in the supposed non-coding regions of the genome that they're discovering on a daily basis. Number two, what I was stating is not only is that the case, and I'm forgetting the name of the, I'll have to go find it again. I'm forgetting the name off the top of my head of the secondary component of the ENCODE project, which is specifically dedicated to the non-coding regions because of all these discoveries that they're making.
So how many papers is it? How many are we dealing with? Because I want to find and read these very well so I make sure I'm educated next time I have this conversation with someone. How many people are saying that there are far more functions and they need more funding? What papers with these medical researchers? And then which one is the
go to Nature and search non-coding DNA discoveries, and you will find a ton of them.
Yeah, but I asked that. I remember I asked a question.
And I have several in my folder.
I tried that. I tried searching that last time and I didn't find it. Do you think you could do me the favor and maybe email it to me?
I will email you the link to my folder, and you can go find it in the plethora of papers I literally have documented and titled.
You don't know where it is in the folder? You couldn't just email it to me?
I do, but since you don't actually seem to want to consider the arguments other than do anything, then not really.
Oh, so, okay. All right. Well,
because you've been kind of condescending to me today.
No, I didn't turn it on until you did. You know, honestly, I do try to be pleasant in these kinds of things, but I think you're very aggressive, and that's fine. Everyone's got different styles of arguing, but you know, I guess
my aggression is coming from the fact that you are directly dismissing factors that have been well established since Watson and Crick in regards to the genetic code, number one.
I just think you don't have the papers. I think that you've been saying go look in the folder for a long time, and that I know people who have, during the middle of your debates, gone to look for these papers and not found them.
So obviously I haven't read any of the papers because there's plenty of them in there. But are you suggesting that since Watson and Crick, the premise of the genetic code, what is the origin? Hang on. What is the origin of the entire premise of the frozen accident?
You're the moderator standing. Go ahead.
So what I want to do, because this has been a very fast-paced, engaging discussion, I got to say, great job, guys. I know it can get heated and passionate at times.
I'm having a great time. I always have a good time in these.
It's been a wild ride. The audience has had a blast. We've had 90 people or so in the chats. So you guys bring the numbers, and to the chat's credit, we got a ton of good questions. But what I want to do, because it's been so fast paced, I want to give you guys a couple minutes at least just to kind of wrap things up, go over whatever you feel is necessary to sum up the discussion, and just give your final thoughts. If you want to address anything you feel you were not able to in the discussion, you can do that in kind of like a 2-minute closing if you'd like. Since John started, we can start with you, John. We can go to you, Erika, and then Brother Sal's has done a phenomenal job gathering all the questions. There's been a ton, so we're not going to be able to get to all of them, but we've got a lot, so we'll have some fun there. Go ahead, John.
Well, I mean, tonight, folks, has been yet another example of the atheist, or in this context, agnostic mindset worldview of my opponent, who apparently is incapable of recognizing, or unwilling, not necessarily incapable, unwilling to recognize the significance of the things that she is trying to explain through natural processes. There are things such as the genetic code, going back to Watson and Crick, and the whole origin of the premise of the frozen accident was to explain the arbitrary value assignment of the genetic code. This is an unavoidable component. Now, atheists and evolutionary biologists and agnostics desperately attempt to circumvent this and try and claim it's only an analogy because they recognize, or some recognize, the significance of the entire point. If it is actually genetic code, if it actually is programming language, which all of the factors show that it is, then there is no other context in which that could occur without an intelligent agent. When you factor in, from a chemistry perspective, the extraordinary improbability and the number of processes which must be happening simultaneously in order for life to ultimately come into existence, the time factor has zero benefit to the evolutionists. They think that it does, but the time factor has no bearing on whether or not it's actually plausible for these things to come into existence. And you still have to have it accounted for the first point of syntax, semantics, and pragmatics. Then you haven't accounted for the temporal values, the space and the spatiotemporal factors, which must be in existence in the genome in order for life to exist. Then you have to account for the functional proteins, some of which are so massive that you are now arguing that something like the Titan protein, for example, which is 33,000 amino acids in length, over 100,000 base pair nucleotides in a single genome, in a single gene sequence. Somehow, those all came into the correct order with no errors to result in one of the most fundamental proteins that enables life to exist. I mean, and this is just one example. You go on and on and on. The information processing components, it is completely recognized. There is massive amounts of information processing to the extent that at a top-level computer science conference, I think in November of last year, they were talking about how there was a whole keynote speaker on how not only is the genome and cellular biology Turing complete technology, it is information processing, it's not analogy, it is literal. They even went so far to argue that the information processing happening in the cell is superior to what we're trying to recreate with quantum computers. And they're arguing that we should go down that path because it is that much more efficient. So ultimately, in the context of intelligent design, I am arguing that it is not logical, plausible, or probable that information processing systems and communication systems and technologies and engineering and mechanisms and nanomachines which enable life to exist could ever come into existence. I don't care how much time you have in the works without an intelligent agent. If you are arguing that they can, then if you blew a cell up to a macro level, you are talking about something that's bigger than the city of London, with more factories than on the entire planet, and more technology infrastructure and communication systems technologies that are vastly superior to anything humans have ever done. Just because it's happening on the molecular level does not alter the fundamental components and requirements for all these things to exist. That's enough of my diatribe. Thank you for our debate. Glad we had some heated back and forth, and the floor is yours, Erika.
Thanks, John. Yeah, we get passionate, but honestly, it all boils down to a desire to better understand the other person's position and to try to get where you're coming from across to the other person. So while we do get heated, I still had a ball. So from my perspective, though, with regard to John's specific argument, I find it to be a very large argument from incredulity. I am certainly not as well versed as I could be in all of the immense literature body that involves abiogenesis and all of the components that go into it. In fact, I'm more of an evolution gal when it comes down to studying once life is already here. But like I said previously, that was never going to be the kind of conversation that we were going to have from John's perspective, because it's the fruit of a poison tree. And I'm glad we got to have this conversation. I got to get out of my comfort zone a little bit, and now I have a lot more reading that I can kind of get into and learn more about. But when it gets right down to it, you just got to look at the literature body and take the time to go through the various hypotheses, because there are quite a few. This is an area of science that I'll be the first one to tell you. We don't know as much as we could know about it. And that's part of what makes it so cool. And that's just kind of my thoughts on it, I guess. It doesn't really get too much more complicated than that. Of the papers that I've read, none of them seem to be very problematic in the light of what John is saying. I mean, I Googled just now, just for fun, is DNA like a computer code? And the very first thing that comes up is DNA is not like a computer program, and preceded by several smaller bars kind of going more in depth on that. So I do want to get more in depth into that on my own in the future and articulate my arguments a bit further on this in particular, and look into the biosemiotics of how the people who know far more than anyone in this chat and in this talk here ever could know, or maybe not ever could know presently, because they've done their doctorates and things like that on that. But yeah, I'm looking forward to learning more. I just don't find the argument that John presented for intelligent design here particularly compelling. I just think it's a lot of irreducible complexity, arguments from incredulity, and a bit of, and I don't mean this in a disparaging way, but sort of a hand wave of the factors that I find to be very important in the process. And that's pretty much all I got for you. I did have a good time. It's not very often that I get to get a little bit heated, but better to do those kinds of things when we're all just trying to get to the same place and learn more.
I agree. It was definitely a lot of fun. I'll tell you, one to remember. Thanks for making it fun, guys. So I want to thank Brother Sally. Had a tough job. We got a lot of questions, super chats flying in. So what we'll do And it was a good idea from Sal's. We're going to acknowledge the super chats first, and then we will get right into the questions that Sal has gathered. So go ahead, Sal.
All right. Well, I'd like to tell the audience, Erika and I go a long ways back. And when she was on some of the Reddit subs, including some of one of my own. But at the time, I thought she was a guy. So it was a shock to find out Gutsick Gibbon was Erika. So first super chat is from Modern Day Debate. He says, congrats, Erika. And he said, hit like, peeps. The next super chat. Yes.
Liked myself.
I'm sorry?
I said I just went and liked it myself. I'd forgotten to hit like on the debate.
The next super chat is from Bond, George Bond. And he has a question for Erika. If the RNA from clay is the most probable, why is it that they are now suggesting in a paper that it is probably both RNA and DNA come into existence at the same time? Before you answer that, why don't I just acknowledge the other super chats?
Yeah, sure.
And I'll queue up those
Good
questions
idea.
for you, just so we can recognize these fine people, since it's been a long discussion.
Yeah,
Jeff Lynn, did I mistype that? Oh, wow. $250? Maybe I mistyped that, but if you did, thank you. And we can send Erika a nice wedding present. SFT buy a coconut for raw mat. And from Smoky Saint, GBU SFT, thanks for the support today. From Coco Puffer, Erika. This was one about UB dating, but thank you, Coco Puffer. So I'm going to queue up 2 questions there. And Ian Chen says, Erika is super awesome. I already know she's one.
No,
so there you go.
Ian's a good guy.
All right. So shall I just go ahead? I'll read George Bond's question since he was in the super chat.
Yeah, absolutely.
If the RNA from clay is the most probable, why is it that they are now suggesting in a paper that it is probably both RNA and DNA come into existence at the same time?
I don't know of a paper that suggests that they come into existence at the same time. It sounds like a cool paper. Maybe it's new and I haven't seen it. As far as the clays go, to my knowledge, it isn't that the RNAs come from the clays, it's that the clays allow the RNAs to more easily self-organize. So if this second paper is the more viable hypothesis, it could still exist under the lens of clay assisting in RNA self-organization.
Thank you. And then Coco Puffer, Erika, UP/PB, that's uranium lead dating, assumes no PB, that is lead, is present at the formation of the zircons. Have you read the paper published in PNAS showing PB present in zircons devoid of uranium?
Yeah, I think I get what you're going with here. No, I haven't heard that. I haven't read that paper. It sounds like an interesting one. I would be interested to see if whichever way, like when they're looking at this, because typically the way that radiometric dating works is they corroborate numerous methods across with one another. If it's a mineral of any importance for determining the date or age of something that is vital in the geologic column, vital for the study that they're doing, typically they'll cross-corroborate with more than one method. So I would say that if in this particular case, this paper is 100% correct, and occasionally we can have trace elements of the daughter or parent where they're quote unquote not supposed to be at formation, I would be interested to see if that applies across the board with elements, because as far as I know, it doesn't. And more importantly, in this specific paper, how much was found, because I think it's very easy to point to sort of novel findings and apply them across the board. But the tricky part comes when we're analyzing it in depth and trying to figure out why it is the way that it is. Case in point being what factors could perhaps contribute to that. Anyways.
SFT, I'm just going to farm out the questions if you want to help me handle the back and forth, if there's any, that'd be appreciated.
Of course, yep. No problem.
And so from Oh, yeah. There are 31 more questions now.
Whew.
From Spart 344, open question. What kind of an off day was the designer having when he designed the human reproductive system and the wind plus food pipe? I think that's a rhetorical question. So I guess no answers. I'll answer
if I think
he's
if
having
they-
a fun day. He's pretty mischievous. That's just my opinion.
A little
Sorry,
bit.
I shouldn't be.
Personally, I think if you actually dive into the reproductive system and the processes that are occurring in order for it to happen, it's actually rather exquisite from a functionality perspective in terms of what is actually happening in the zygote itself, and then how that ultimately results in a human. It's pretty amazing.
Richard Dawkins has a fun quote. I'm not a big Dawkins fan in most ways, but he does make a good quote. And once when he was being asked how an organism how all life could evolve from a single cellular organism, he was asked by a young woman who said, "How is it possible that that could happen in billions of years?" And Dawkins looked at her and he said, well, you did it in 9 months. Which I thought was pithy and funny. I don't think he was meant to be taken literally, but I did find it to be a funny quote, somewhat relevant to the question.
Well, quick counterpunch to that. So you do realize in the mammalian or in the more advanced life forms that are sexual reproduction, which is such a foundational piece of evolutionary theory, you do recognize that the mutations have to have occurred in either the sperm or the egg, right? And it has to have occurred in the sperm or the egg that gets fertilized.
The evolution of sex and sexual reproduction is a fascinating conversation, but I fear if I answer that, I'm going to go on a bit of a tangent because once we get into macroscopic stuff, baby, I'm chomping at the bit.
I'm not talking about how did sexual reproduction come into existence. I'm talking about the actual supposed macroevolutions that you're talking about. Those mutations have to have occurred in the sperm or the egg.
Why could they not?
Because it contains 1/2 of the whole of the recombination, right?
You realize that there are mutations that occur today and only occur on the egg or the sperm. I don't understand why that's a difficult.
No, I get that. What I'm saying is every single mutation would have had to have occurred in a sperm or an egg.
I reiterate
in the outcomes. You've lowered your opportunities to such an extent that the probabilities of all this just become ridiculous.
I'm telling you, John, it's an
Go ahead. Next question.
We got a lot of questions to go through.
Okay, next question. Sorry to cut you all off. This is really more for the service of our faithful listeners and viewers. Jungle Jargon. How do you get high intelligence from no intelligence?
Oh, that's very similar to the conversation that John and I were just about to have. I know quite a bit about cognition in primates and how cognition scales across the primate order. And a lot of it comes down to how complicated are your social systems. So when you reach higher levels, it comes down to maintaining sociality and things like that. And we can track this based off of the sizes of baboon troops to a degree. As far as the first intelligence, I think it depends on where you draw the line for intelligence. Like I was saying to John earlier, certain kinds of sponge molds can navigate a maze if you give them enough time. Does that make them intelligent? Well, no, they're doing response stimulus and managing to grow their way around walls and get to the food source that they can pick up on. So it's a difficult question. It depends on where you draw it. What would be the first intelligent, quote unquote, organism? I don't think it's a line that you can draw. It's certainly not one that I can draw.
Okay, I'll go to the next question. Interrupt me if you feel you want a little bit more time. To John from Luca Medugno from Italy. I think that's one of the guys from Europe here. We have people from all over the world. And his question is, how can you tell that abiogenesis was just a random process? Maybe a chemical pathway exists, one that can explain the phenomena. And that was directed at John.
Well, at some point you have to recognize that we've been staring at this chemistry for quite some time now. And what it's boiled down to is that somehow all of these things had to have come into the correct order through an undirected process if you want abiogenesis to occur, and arbitrary values have to be assigned also through an undirected process to result in a controlled, structured outcome. And what is being argued in abiogenesis is that, hey, there is a minuscule chance that absolutely surpasses anything else from a probability perspective that would be considered remotely plausible. It surpasses Borel's law. It surpasses the law of small probability or chance probability, which is 1 in 10 to the 150th power, equaling 0 from a universal scale. 10 to the 50th is on a Earth scale, being 0. But no, due to laws of large numbers, that this could possibly happen. Well, at this point, it's like saying, oh yeah, cool. There's a chance that I could pick I could win a lottery that has 20,000 numbers that all have to be picked in the correct order. And yeah, I'm going to be able to win that. I mean, it gets so ridiculous to the point of, yeah, bro, if you win that lottery, I'm going to immediately start an investigation into how you cheated. And that number of 20,000 numbers to winning the lottery isn't anywhere close to what's actually required to have happened in order for abiogenesis to have occurred. So, I mean, just because you can say, oh, if we have enough chances this might happen, does not remotely make it a plausible scenario. As in my debate with Ron Garrett, he said, oh yeah, well, from an information theory perspective, it is actually possible to come up with a chance probability of a piece of iron turning into a sword. Now, that doesn't mean it's plausible, but you could still come up with an equation. And that's exactly we're talking about in the context of abiogenesis.
Okay. Everyone okay? Next question from James Downard. Question for both debaters. When did life first appear? What did it consist of? And what happened to it next? That's for both of you.
I just ranted. Go ahead, Erika.
Oh, I can go ahead. I mean, I kind of said it in my earlier. I'm trying to switch back. There we go. I said it earlier in my conversation with John. I mean, from my perspective, according to the current literature, which might change as science does and should, I think, if new information comes along, you definitely want to change your ideas on it. I think that's the hallmark of a good field. But from my perspective, we're probably dealing with something in the hydrothermal vents approximately 3.8 to 4.1 billion years ago. We see probably the formation of lipid encapsulations forming first, followed perhaps either by proteins or sort of our nucleic acids. I'm not sure. Again, I cannot emphasize enough how this is not my field. I do my best when the conversation goes in an area or is planning on going in an area that I tend not to know about, but I think you would have to have a pretty good sized degree to understand the inner mechanisms of the current literature on this in its entirety. But I think what happened to it next, well, replication is the hallmark of life, one of the hallmarks of life, along with like responding to a stimulus and doing all of those things. So I would wager it started reproducing, it started dividing, or it started consuming, depending on what kind of organism it is, if it's like a methanophile or whatever. But I think it's going to be interesting to find out more.
Okay, the next. Go on.
Sorry, to me too. I mean, the entire premise of Downard's question operates on the idea that somehow life could come into existence in a non fully formed manner. So my argument is that the entire point of the intelligent agent being required is that a fully formed model must come into existence simultaneously. All the factors must come into existence simultaneously. And then when you look at this from a bigger perspective, and you look at the elements of design structure across all forms and the barriers to macroevolution being even remotely plausible, fully formed structures of the different lines of animals is the logical conclusion.
Okay, the next question I think is more or less rhetorical. Gavin Herleman. Why do atheists like Tom Jump insist that DNA is created by RNA? Okay, I guess we don't have to answer that. Okay.
Well, probably because there used to be origin of life researchers that tried to argue that, and then they came to the realization from a chemistry perspective that was not remotely plausible and had to give that one up. But people like T-Jump still cling to some of those models.
Okay. I'll make no. I'm a co-moderator. I can't make snide comments. Sorry. Bill Morgan. Question for Erika. Do you think crop circles are most likely the result of design or chance? And what criteria do you use to determine?
I think that crop circles are probably the product of design. I think that when it comes to determining a design from a non-design, a lot of it comes down to efficiency and simplicity versus redundancy. And a lot of it comes down to, is it something that can be selected for and can reproduce, or is it not? Which is fundamentally why, when in sort of John's example, when he was talking about making a nanomachine that mimics cellular structure, to me, that doesn't really say anything other than we copied something that self-replicates. I mean, and sort of made a human version of that. The parts of our nanomachine, nor can our nanomachine itself replicate on its own. So for me, those would be the hallmarks. That's why I want criteria for design. Like, so very frequently these conversations, and I fell into the same trap today, fall into critiquing the evolutionary model rather than saying, well, what's the creation model? What would the first design look like? How does one determine design from non-design? Because we know that creationists accept sort of evolution to a degree, as long as it's within the kinds. So how do we determine what nature makes versus what God made or what humans make? But I didn't push on that, and it ended up being a conversation about my model, or conventional sciences model, which, c'est la vie, maybe another time.
There are several questions from also James Downard and Bill Morgan. What I'm just out of courtesy, I'm going to actually try to get to some of the other questions first from other people. Chris Keith, ask Erika what process she believes allow for the irrefutable soft tissue we find in dinofossils. How does she think osteocytes, bone cells, survive for millions of years?
Ooh, I'm going to direct that person to my video on Mark Armitage on that. I cover extensively how plausible I think that it is that osteocytes with a half-life of approximately 22 years could survive in a dinosaur bone for several, maybe a couple of centuries, let alone millions of years, because I don't think that was one a Triceratops horn, and also I don't think that even if it was a bison horn, which I think it is, likely a result of contamination, which we see frequently in the conventional field and has to be controlled for, but we lack the methodology to understand exactly what was going on with Mark Armitage's work there. I would say, as for the soft tissue, I hear that one a lot from creationists, and they don't tend to keep up. They, meaning the ones that I'm interacting with anecdotally, maybe this person is different, but they don't tend to keep up with the work of the actual progenitor of that work, Mary Schweitzer, on her various preservation methods, nor on her colleagues' preservation methods, which propose a series of different mechanisms kind of working in tandem, and they seem to work quite well experimentally. So I would encourage them to look for that, and if they want, they can email me about it, and I will give them all the sources that I have for it. gutsickgibbon@gmail.com, all lowercase.
Thank you. And the next question is Leophilius. I have a question for John. Is all genetic material made of natural chemicals? Does it all operate under natural laws? If so, where's the implication of anything supernatural?
Yeah, it does. So does a hard drive, and magnetic hard drive. It's using the laws of physics to store information. However, nobody would argue that the process by which magnetism is used in chemistry, in some aspects, is used to store that information could ever occur without the intelligent agent. So if we're talking about and this is the point I've made many times, and apparently people who believe in so many people cannot seem to grasp this premise, that the medium which contains the information and the processes is completely irrelevant to the origin source requirement for that prescriptive information. It blows my mind how this seems so difficult for people to comprehend. I write down instructions on a piece of paper. I take a picture of it. I upload it to Google Drive or to an email, which I send to Erika. She then downloads that and prints it out onto a new piece of paper. That thing has just gone through 4 or 5 different states of the information being contained. Now it's back on a piece of paper. However, there was still information that was required. The medium which contains it has 0 relevance. There was chemical processes as a result of me taking a pen and the ink binding to the paper. That has nothing to do with the origin of the syntax, semantics, and pragmatics, which are now being delineated onto that piece of paper through a chemical process and then translated and transmitted via encoded and decoded through electronic transfer. This is exactly what's happening in all aspects of biology, and why I don't think anybody would conclude that what I just described in terms of what I did to send directions to Erika could ever be accomplished without an intelligent agent.
Okay, with 4 more questions, we'll actually get to all of the readers before we start to repeat. So let's see. The next one is from Dapper Dino. The question is for both. How does the recent discovery of persistent ribose in asteroids impact your ideas about abiogenesis/ID?
I can go first on this one. I think it's super interesting. The panspermia idea has kind of been poking around in the abiogenesis community, for a while now. And I say that as someone who's barely involved in the abio community. But I think it's very interesting. We know that ribose is it's pretty important for early life in many different capacities. Sorry. So I think it certainly says a lot about the ideas that at least some components may have arisen from the stars, which is weird and kind of woo to say, but the paper's pretty interesting. I think everyone should go and give it a read and see what they think. I don't know what I think. It could mean a lot of different things.
Assuming it's the one from last year, there's 2 things about that. One, okay, they may have seen some amino acids. They think that it might be a protein, not 100%, but what often gets glossed over is the fact this asteroid that they're referring to crashed into Africa, I think back in 1989, and they didn't, quote unquote, discover this protein until literally 30 years later. And we know full well that Erika was talking about this in terms of contamination. It's also not a completely unrealistic or implausible and improbable outcome that this is a result of contamination. So I know there's excuses on that whole deal, but just either way, it doesn't really affect the requirements for abiogenesis.
Okay, the next one is from Mitchell. A question for Erika. Which one of the 4 RNA nucleotides evolved first, A, C, G, or U?
I don't know. I think that's a really good question. I think that's one of those questions that even if you're like totally on board with the conventional science, as I am sort of, I don't think that's a question that we would be able to answer, because we're never going to know the precise conditions of the prebiotic Earth. I think you can get close, and I think you can get a pretty good idea about what's going on, but I'll be the first one to tell you we'll never know precisely with 100% certainty what happened. A lot of paleontology and a lot of sort of origin of life studies is like playing forensics. It's looking at what we know now and trying to see if we can apply it to the past and see if maybe conditions were conducive to one thing or another. So I think a lot of it is just going to depend on what we learn in the future. But I would wager that that's not something that we can know. And therefore, I have no idea. So I'll pick guanine.
Okay. From TD Lane. What does this is to you, John. What does Maddox mean by body plan?
In the general sense, the different body structures and the morphology changes that have happened, i.e., the difference between a whale body plan and a lion. It's rather obvious in terms of body plan structure. But I don't know if that answers the question. I'm getting pretty tired. I've been talking for like 5 hours straight now.
Yeah, it's a double header there. I got two more before we want to go back if we have time, but I do want to get to all the questioners. This one's really a comment. This is from our youngest participant, Guzman 1611. Excellent dialogue tonight to both. I disagree with Erika, but excellent back and forth. And our last one, John claimed that no errors are possible with helicase. However, UniProt shows greater than 50% sequence identity, produces equivalent protein function. How does he move on from being so wrong? In point of fact, I feel that such an obvious error should be seen as disqualifying in a debate. Did he research the claim before making it and lie, or not research it at all?
Who said this?
CRISPR-Cas9. I'm sorry, CRISPR. I forgot to mention your name. That wasn't good.
So he's suggesting that 50% of the amino acids can be incorrect in the helicase, and it'll still be a functional protein. Is that what he's saying?
I'll just read it verbatim. He can explain himself, I guess. John claimed that no errors are possible with helicase. However, UniProt shows greater than 50% sequence identity, produces equivalent protein function. How does he move on from being so wrong? So I'm not going to
let's have a debate on that one, because I'm pretty sure you're full of shit on terms of what I'm actually talking about in terms of the error requirement or possibilities. So let's have a debate, bro.
Well, I was talking to CRISPR in the chat, so that he's happy. I've discussed many times with CRISPR. I think that'd be a good debate discussion. So that's something we can work at setting up in the future. So, sorry about that. Go ahead, Sal.
Oh, well, probably a number of requests by James Doward for documentation, and he said, make sure John specifies the paper he's alluding to in Nature. And
yeah, Downard, as I've told you many times in our debates, and I literally put it in the preface of my openings usually, if you want to find the technical literature, to use your jargon, that I refer to, go to my folder that I post on a regular basis. It's actually on my About section on my YouTube channel. It's under the title of For the Annoying People. And go read through the plethora of papers from which all of my arguments have been derived.
Okay, let me just appraise you, SFT. If I went back and started going through the other questions now on the second round, it'd be about another 10, 15 questions.
No, it's been we could probably go for 10 hours, but it's been a really, really fun 2.5 hours. But, to the debaters, you guys have been on the ball for full long. John, you've had a doubleheader, so I think we're all probably getting tired.
Actually, it's really, I've been talking for 5.5 hours, actually.
You're a champ. Well, to the both of you, thanks so much for giving us your time. And
absolutely,
it was a good debate.
Champ, he's been to at least three debates and discussions today, including mine.
It's been a busy day.
Yeah, I for John, if you would like, I have another friend who would love to chat with you about Origin of Life. I think Dan Sterndale. He's in the chat right now as Creation Myths. He's issuing a challenge. If you would be so inclined.
Creation Myths. Isn't that the professor that you debated, Sal?
Yes, and we're still trying to. Short advertisement, and we'd like to thank Erika for giving us advertisement, because I think a lot of the people that came out to our first debate actually came out to see her, and
wow.
And I saw some Twitter feeds, and a guy said, "I'm so heartbroken, Erika is only going to be the moderator." So
you can only imagine how heartbroken
Erika.
Well, that person's going to be crushed now that if he finds out that Erika's now engaged. That's
So
his heart is now broken. She's not showing up and is taken off the market. Oh my
I
God.
know.
That's what you were doing.
I'm excited though. I'm going to be. I would love to. I'll shoot another tweet out too when you guys are getting ready to have that debate, because I know a ton of people would want to be aware of it just for the content. Sure. I appreciate the flattery, but genuinely, I think most everyone's just there to hear the good talking points.
So just a little bit of free advertising. We've tried to contact James. We haven't been given a slot yet, a date. Just my speculation, I think the whole Matt Dillahunty and Ray Comfort thing probably just overwhelmed James, and that's overshadowing everything. So that's this Wednesday. But Professor Cardinal and I are still waiting to be scheduled, and we're hoping we would really be honored if Erika would be our moderator.
I would love to be there. I want to have it on Modern Day Debate though, because you'll definitely pull more viewers there. James's channel is awesome, and I'd love to help get as many people watching as possible. I think it's going to be a really fun time. And
if you haven't seen round 1, go check it out. That was a great debate.
Yeah,
really good dialogue.
Oh, right. You can actually. You can also go to www.evidenceandreasons.org, which is my new website, and there are links to it. And I'm building out that website. And I'm not much into debate. I prefer lecturing. And I'm hoping to put more stuff on there. So
yeah, absolutely.
Well, whatever we can do to get your name.
Thanks for letting me make the plug there, SFT.
Well, for everybody who's considering,
anybody who wants to plug away.
For everybody who's considering, the watching round 1 from Sal and creation myths, you have to just remember that you've got 2 hyper nerds that are being all nice and dialogue-y back and forth to each other, and I just agree with your position. Here's why. Me and Erika are here talking trash to each other.
We're talking trash.
It's a totally different dynamic of street fight versus academic interaction.
Tonight was more like a Rocky Balboa versus Ivan Drago. So it was
oh my goodness, I got to tell you about that. There was one guy in the YouTube comment. He said, "I was in nerd heaven." He was hearing us talk, and then people kept talking about Nate Thompson. Then I saw him debate. He's arguing for flat Earth. I said, "I think that guy would crush me." Debates are not all about facts. It's also about style. I think that guy would just totally bulldoze me. So.
Well, and the irony, though, of the flat earther community. So SFT and myself were all lined up to debate Team Skeptic and Fight the Flat Earth. They canceled the last second. I think they bowed out, but we'll see what happens. Anyway, we were not going to be debating the flat Earth because I'm not a flat earther, right? However,
you're not a flat earther. I am.
However,
flat Earth reveal.
However, the ultimate irony was that I did a live stream. SFT hopped on. We kind of bitched and moaned about the fact that they backed out of the debate a couple hours prior to the launch.
I was up all night preparing. So
SFT is coffeeed out of his mind and ready to rumble. Anyway, so we do a little live stream. We talk some trash about it. All these flat earthers come and comment about how we're denialists. And I'm like, are you idiots not paying attention? We're not going to be debating the flat Earth. We're debating intelligent design. Idiots. Then all the anti-flat earthers came and started accusing us of being flat earthers. I mean, I'm like, what is going on? Neither one of these sides, the Globers and the flat earthers, neither one of these people actually used their brains and actually listen. They didn't watch the video.
And we were
literally talking about, we're not debating flat Earth. It's cracking me up.
We can all agree here, and hopefully in this conversation, that the Earth is indeed a globe. Can we?
Yeah, I mean, I got no problem with
that. I don't know, Sal. Are you
a spheroid
flat earther?
Oh my God, a spheroid.
I'm sorry, that's nerd heaven. I worked in the aviation industry, and they said, no, it's not a sphere, it's a spheroid.
A spheroid. Okay, I stand corrected.
Well, there's nothing to fear but the sphere itself.
One of the things on my bucket list is to recreate the Red Bull challenge, where the guy went up in the helium balloon, set the record for the height, and then jumped off literally from space.
Oh, yeah.
I'm like, man, if you're gonna die, talk about if it fails, talk about the coolest way to go out. You jump off. It's like a scene from the original Superman movies. You're now, Christopher Reeve in the Superman costume, got the Superman theme song playing as you drop off and observe the curve of the Earth. Man, that'd be awesome. Let's see. That'd be cool. But
I think that's what the flat earthers are doing. They're trying to get someone to put them up to prove it's a globe. It's just a ruse so they can get a free trip.
Well, there was a flat earther last year who had built a rocket, and he died trying to ride his rocket off to prove that exact point. It exploded.
Sorry.
Yeah, I remember that. That was a bummer. It was a bummer. It was sad, but at the same time, like, you know, it's kind of a weird situation. Like, building your own rocket just seems like a bad move.
I did that as a 16-year-old. It exploded in the backyard. I was in the hospital 2 days. I still have a scar on my chest for it. So that's my little mischief. So.
Well, not too many people can say that they've attempted to build a rocket. I can barely change a tire. I'm not building a rocket anytime soon. Okay, so we could probably go at this all night, guys, but anyone else want to plug away anything we can do at Standing for Truth to, help you guys out? If the debaters wanted to plug their channels real quick before we call it a night. And Brother Sal, great job with the questions. You did a really good job with that question and answer period, so I appreciate that.
Yeah, absolutely. Thank you, Sal.
You're welcome.
So if you the debaters had a couple final words, wanted to plug in their channels, and we will call it a night.
Oh, plugs. Okay, yeah, Gutsick Gibbon. That's just it. That's the whole deal. I don't know who's or if we or you guys or who is running an after show, but I think there might be an after show on my end. It'll probably be on Dapper's channel. If it is, that's my usual guess. He's the usual suspect. So.
On that note, I'll hand it over to Praise, the amazing producer. Praise, thanks for sticking with us this entire time. You can shut her down.
Ladies and gentlemen, thrilled to have you here for another epic debate, as today we have Erika and Standing for Truth joining us for round 2. It's going to be an epic one, folks, and want to say, if it's your first time here, consider hitting that subscribe button, as we've got a lot more debates coming up. In fact, you'll see at the bottom right of your screen, Endgame, the final chapter between Mark Drisdale and Kent Hovind, will be this coming Tuesday. So our last debate between those two gentlemen, as it's always been fun to watch them, and so that's one you don't want to miss. You can set a reminder. So it's going to be a fun one tonight, folks. We are really looking forward to it, and I want to let you know what we are going to do for format. I'm going to pull up on email because I'm trying to remember, unless one of you remembers. But
yeah, we're doing 5 and 5 short intros and then open discussion. Yeah, Standing?
Very good memory, Erika.
Hey, that's what I do.
Gotcha. Perfect. So it'll be, if I understand right, 5 rounds of 5 minutes. Is that what you're saying?
I mean, I thought it was openers and then open discussion.
Right. Yeah. No. It's
gotcha.
5-minute introductions and then just open discussion and then Q and A. I'm guessing.
Thanks so much. Appreciate it. Long day already.
That's the fun way to do it, anyways.
That's what we're here for, James.
Stoked for it. Thanks for that refresher. And want to let you know, folks, if you have any questions, fire them into the old live chat. I will pull those questions out of the live chat, and we'll ask as many as we can at the end of that open discussion section when we get into Q&A. If you want to do super chat, that's an option as well, in which case it'll push your question to the top of the list for the Q&A, and it allows you to make a comment toward one of the speakers. Of course, they'd get to respond to it, and we ask you to be your regular friendly selves for that. And so with that, very excited, folks, I am going to set the timer for 5 minutes. And my presumption, because usually it's just the habit, we always have the creation person go first. Standing for Truth, do you want to go first, or did you already decided that, did you guys already decide together that Erika would go first?
I don't think, we didn't have a choice, but I don't care.
Yeah, it doesn't matter to me. I mean, to be fair to Erika, she started first last time, so I don't mind starting first this time, but ultimately it's up to you.
Deal.
Yeah, sure. You can start. Yeah, I always start. You start.
You bet.
Only fair.
Okay, let's timer set. Standing for Truth.
Awesome. Well, thanks so much. Thanks so much for doing this, Erika. The first debate was a blast, so I'm really excited for this one.
And
James, as always, brother, thanks for setting it up. So before we get started, we all must realize that it's genetics. It's in our DNA where the question regarding ancestry is answered. Genes, traits, genetics is what's inherited sperm and egg. Sperm and egg don't pass on a fossil or a bone. They don't pass on geography or a rock. They pass on genetics and traits. And so if we want to find out the history of humanity, this is where we must look. We have clocks in our DNA, for example, that go back just 6,000 years to Adam and Eve. Let's discuss some reasons why, based on genetic data, the Bible's account of human history is true. When we focus on the variation, for example, in the Y chromosome in the world today, we see that Y chromosomes have very little variation. This indicates that we came from a common ancestor in the not so distant past. Interestingly, the Y chromosomes are geographically specific. There exists African Y chromosomes, British Y chromosomes, Chinese Y chromosomes. Why is this? This is because mutations have occurred in the lines after Babel, since God divided the nations based on their Y chromosome. The family lines are based on the male ancestry. These nations were divided across the planet. Is there good evidence for limited ancestry? Or does the evidence tell us that humans did not go back to Adam and Eve, but are actually related to apes, dogs, and banana plants? Well, the evidence for the existence of Adam and Eve and limited ancestry is you and me and the genetics of the people in the world today. The genetic structure in humans today speaks to us of a literal Adam and Eve. We have a small DNA compartment called the mitochondrial DNA that we only inherit from our mothers. Everybody on this planet got their mitochondrial DNA from a single woman, who is exactly what we would expect from a biblical Eve. She is quite literally the mother of us all. In a nutshell, we have nearly identical mitochondrial DNA, and the very few mutations that differ from Eve's sequence are exactly what would be expected if Eve arose just several thousands of years ago. As I briefly touched on earlier, the same thing goes for the male side. Science tells us that all men have similar Y chromosomes, which is passed down from their fathers. And all of male humanity traces back to a single man. This single male ancestor of humanity has all the qualities expected if the Bible were true. The main argument offered, though, by the evolutionist community, especially biologos, against a literal Adam and Eve and ultimately limited ancestry has to do with genetic diversity. They will say there exists no real scientific way for two people to generate all the genetic diversity we see in humans today. This main argument is easily refuted and actually reflects a poor understanding of even basic genetics. A man and a woman today could have thousands, even millions of children, and no two children would be 100% identical. This is because they are heterozygous. There exists built-in diversity in millions of sites within the genome of the average person. In the limited time that I have here, the best way I can explain it is if God created Adam and Eve to be heterozygous, which in fact would be optimal design, you could have as much human diversity and more than we have today in their first family. Time is not required for the diversity today because diversity can be designed and built into Adam and Eve. This argument is not even scientifically reasonable. One more line of evidence I do want to point out before we go into the discussion has to do with the HapMap data. The HapMap project is what I am referring to, and this very important project was intended to compile a significant fraction of human genetic diversity. They examined millions of variants in thousands of people from around the world and made the data easily available. Dr. Robert Carter puts it perfectly when he states, and I quote, "I am quite happy with what we learned from the project." What have we learned? The human genome is young. Shared blocks of DNA are large, and there has not been enough time to scramble them to randomness. The human population came from a single source. Most blocks are shared among all world populations. The human genome is falling apart. Deletions tend to not be shared among populations, but are unique to subpopulations. This is further evidence for the youth of the genome and that we came from a single source population in the recent past. I've got 10 seconds here. Genetic entropy, orphan genes, the chimp Y chromosome, endogenous retroviruses, linkage blocks, for example. There's many different lines of evidence we can look to that point to limited ancestry, and I'd be more than happy to discuss any one of these topics with Erika and show her why limited ancestry is true and universal common ancestry is false. Thanks so much.
Thank you very much. Standing for truth, just several seconds over. We'll give Erika the same leniency. So with that, we will jump over to Erika's opening statement. Thanks again for being here, Erika, especially given how late it is. It's a pleasure to have you.
Please don't worry about it. I will, if that's okay, share my screen. I know I normally have my nice monitor up, but, and then, can you guys see that?
It's now showing. So let me just squeeze it down to make it fit.
All right. So, biology supports a universal common ancestry. This is the argument that I am going to make today. Now, generally speaking, when we look at a universal common ancestor, what we're kind of referring to is that all life on Earth shares a common ancestor in the form of a single-celled organism some 3.8 billion years ago. Now, as Standing mentioned previously, his argument is somewhat different in that he's sort of going for the creation orchard, so to speak. So if you can see in this picture here, this would be Standing's creation orchard on the left, where we have several sort of nodes that are acting as creation origin kinds, right? And the diversity within those kinds is stringent and kept separate from one another. Now, of course, my position would be the conventional tree of life, which is here on the right. So the question is, why do all of the fields in biology agree that universal common ancestry is indeed sort of the reality that we live in? And the reason is because, at least in my opinion, in my humble opinion, it is the case. This is, in fact, our reality. We can look to genetic support, we can look to paleontologic support, and we can look to morphologic support. Now, as Standing said, we're probably going to be talking mostly about genetics today. So while I included some slides here on paleontology and morphology, which I will skim if I have the time, we're mostly going to be considering genetics. So I've harped on this many times before in other debates and in other conversations, but genetics by definition dictates relatedness. This is simply the case. The same sequencing that links humans to all other humans links us to the other apes, all the mammals, all the chordates, etc. So when you look in this picture, again, as I've said multiple times before, the same logic, the same genetic sequencing, the same means by which we develop phylogenetic trees, Standing would agree within sort of the kinds, like they, creationists tend to use kinds, sort of structure their kinds with genetics as well. The question is, can the lines cross? And my argument here is that there is no empirical reason to suggest that they would. There just isn't. It's a smooth gradient. And I've said this before too, but the sort of the same process that we use to allow paternity tests to be considered admissible in court. We used to reach the 98.8% similarity between us and the members of genus Pan, and then indeed 92 with the macaques and so on and so forth. So in addition, we could talk about endogenous retroviruses. Now here I've linked a couple of nice papers that cover the similarities of the ERVs between us and the chimps, and we have this nice little quote on the side that you can read if you like, which, to summarize it, 99.9% of ERVs in humans are at the identical location and are structured identically between us and the chimps. And even when we look at these specific ERV insertions, we find that the mutations that those ERVs experience in and of themselves are also identical, which means either we're dealing with a completely separate event for every single ERV in every single primate lineage, both within and without or outside of the human and the ape kind, and I'm the monkey kind, I'm not sure how Standing structures it, or we're dealing with a common ancestor. So my question would be, why can creationists not draw these lines between the kinds? Why do we see this smooth gradient that I've been sort of alluding to? You can sort of see the same thing when we look into deep homology, which would be kind of the argument that we're dealing with the same building blocks. So geology and paleontology, of course, continued to confirm a common ancestry, and not just that, they preclude a 6,000 to 10,000 year old Earth. Now, I know it might seem sort of tangential to bring this up in a conversation about genetics, but in genetics, we get sort of divergence times using molecular clocks. And interestingly enough, radiometric dating, a completely isolated physics-based sort of just equation that we use to measure radiometric or radioactive decay, confirms this. They corroborate one another. And not only that, but paleontological evidence can also be corroborated with other methods like ice cores or dendrochronology or natural nuclear reactors like the Oklo reactor. Then, of course, there's morphology, atavisms, vestigial structures, deep homology. Here's some nice pictures. Sometimes we see horses, display these additional toes that they used to have when back with Eohippus and the like. Vestigial pelvises, which I know some people say have a function in copulation, but not so. Ask any of the whales that require a third party to mate. We see deep homology in the form of the PAX6I gene. Erector pili with our hair standing on end, and of course our wisdom teeth. None of these make sense when we're sort of taking this idea of a designer into our mind. And I know that genetic entropy is going to be brought up, and that's the reason why we have these sort of degenerate traits, but I'm ready to have a conversation about that. So my question for standing is sort of threefold. Well, I think there might be more than three, but where and more specifically, why do you draw the lines between the kinds, and what separates the humankind from the ape kind? You can do this genetically or otherwise, I don't care. Another one is, how can limited ancestry account for the gradient of forms that bridge the kinds in the fossil record? So here we have an amphicyanid, and we have a wolf, and we have a grizzly bear. Now, morphologically, it essentially looks like you took 50% of the traits from the amphicyanid and then sort of parceled them out into these two very closely related animals that we have today. And lastly, why do we see atavisms and vestiges in nearly every animal? And why do these vestiges seem to indicate and suggest that they're from our genetically derived ancestors? Why do they match evolutionary theory? What's the deal with a whale having a pelvis and then also us finding similarly structured organisms living in the same location with the involucrum, which is an inner ear structure unique exclusively to the cetaceans, and through geologic time? So these are sort of my questions for the conversation, and that's it.
Thank you very much, Erika. So now we will switch back over to, or I should say we'll switch into open discussion section. So I will close your share screen.
Thank you. I know how to do it.
Gotcha. Okay, thank you. Very sassy. I appreciate that. Next, we will go into the open discussion section. So very exciting, folks. And the floor is yours. Thanks for being here again.
Yeah.
Awesome.
All right, Stevie, where do you want to start? You get to pick because that's how we did it last time.
Sure. Awesome. Well, I appreciate your opening. Thanks for the visuals. Definitely lots of good questions, lots of good topics. Since you started off with ancestry anyways and where we can draw the line and kind of how there's a perfect gradient, a perfect nested hierarchy of life, I guess you can put it. My question to you then would be regarding certain taxonomically restricted genes that we can look to separate ancestry, right? So if we're going to look at limited ancestry, for example, if we look at orphan genes, and orphan genes, they lack similar counterparts in other organisms, as in like there's no evidence for common ancestry with, say, humans and chimps because of these taxonomically restricted genes that are specific to humans and then also very specific to, say, chimpanzees or like the cat family. Or
well, sure, of course there would be, because the divergence time between humans and chimpanzees, the divergent time would be approximately 13-ish million years ago, with the speciation time being 6 to 7, give or take. So that's plenty of time for you to get emergence. That's of sort of genetic novelties. That's why we have ERVs that are also unique to humans. But you can point to those, and the exception does not make the rule. You can't point to the exception and say, oh, look, there are individual ERVs or orphan genes, et cetera. I don't know nearly as much about orphan genes as I do about ERVs. But then I will simply turn and point to the 99.9% of ERVs that we share that are identical and in identical locations. So are you suggesting, just to kind of turn that into a question, are you suggesting to me that there have been separate genetic events regarding sort of these ancestral viruses for every single primate lineage, and then also humans, who I'm assuming you wouldn't accept our primates?
So good questions. I definitely want to talk about the endogenous retroviruses, but going back to the orphan genes first, as a counter to that, the orphan genes, for example, they're fully functional gene sequences, and what they do is they code for both a functional messenger RNA and a protein, okay? These orphan genes, though, Erika, they don't fit the hypothetical fairy tale of evolutionary theory. And our prediction based on ancestry and proving limited ancestry would be function, okay? So if these orphan genes, which according to our model are needed for the uniqueness that's associated with each created kind, as say described in the book of Genesis, which obviously we look to as a starting point, the fact that this amazing category of genes are taxonomically restricted and extremely functional and unique to each type of organism would suggest a limited ancestry for the main fact that they are fully functional. I mean, how did these come about in separate ancestries according to the evolutionary theory? I want to just make an analogy real quick just for the audience. It's like looking at 3 different books and then finding specific words that have important meanings in the different books that aren't found in all 3 books, right? There's no evidence for any type of ancestry. That's why they call it orphan genes, right? There's no actual evidence for the ancestral genes leading to these genes. So I mean, how would those pop up in evolutionary theory?
Well, I'm asking though, why are you saying, yes, things can be taxonomically restricted. I mean, obviously there are, as I said previously, there are sort of unique ERVs to humans. There's also unique breakages like that are just in the primate order. There's a breakage that does the same thing, the gulo breakage, that also exists in guinea pigs, but it's very different. It's in a different location, and it lacks some of the sort of regulators that ours has. So I'm kind of wondering why it is that you're suggesting that in any shape, form, or fashion that these orphan genes cannot emerge after splits, after divergence points.
Well, I mean, it can be suggested that they do pop up. Like, I believe that the evolutionist explanation is de novo gene synthesis, that they popped up through the non-coding regions of our genome. But the question is there any empirical observational data for that occurring? And the fact that they've popped up to be extremely functional in the genome seems to be more ad hoc, post hoc reasoning. But if I'm arguing for limited ancestry and these types of taxonomically restricted genes that are extremely important in the genome, or what are expected to be found, and this is exactly what is found.
Right, but what you're doing,
would suggest limited ancestry. It's exactly what we expect to find. Go ahead.
Right, but you've got to. I think a clear distinction has to be made. So when we're looking at data in general and we're trying to support a hypothesis or see if the data does support a particular hypothesis, it can. Obviously, as we know in the nature of science, data can support multiple hypotheses. But the sort of the issue with this is you're suggesting the orphan genes. Again, that's kind of why I wanted to make it more about biology in general, because I'm not a geneticist. But I know enough about it. Yeah, but I know enough about genetics to know that these kinds of sort of emergent genes, ERVs, etc., things like that, they don't preclude evolutionary theory in any way. You're arguing that it supports limited ancestry, and again, I don't know enough about it. It may support limited ancestry, but it also doesn't preclude evolutionary theory and universal common ancestry. And in light of pretty much, in my opinion, everything else that sort of I brought up in my presentation that I'm sure we'll touch on later, which not only does it support universal common ancestry, but it also precludes limited common ancestry. You see what I'm saying? Because there can be data that's like, yeah, it fits both frames. Like I know Jensen likes to do that with nested hierarchies, which I disagree. But the fact of the matter is then he turns, and I'm sure we'll do the same thing, and covers other subjects that he believes supports limited ancestry by precluding universal ancestry. Well, right, so it sounds to me, you get what I'm saying. Sorry. Right, yeah. And if we look to homology, right, engineers build in a homologous type pattern, or we can look at DNA level nested hierarchies. We can look at nested hierarchies on an amino acid level, right? I know Dr. Herman Mays likes to look at neutral variation, for example. But when there are competing hypotheses, we need to look at lines of evidence that can differentiate between the two. Now, I disagree. Absolutely. Right, but I disagree with your point in that orphan genes can be explained
through fish to fisherman evolution because of the important functional roles that have been discovered in them. They are taxonomically restricted, and I've yet to hear an explanation from you as to how these orphan genes can pop up, can be specific to lineages, for example, humans to the great ape family, to the cat family, to the dog family. How are these popping up and having very important functional roles? I mean, that doesn't just happen. We also don't see that empirically in the lab. That's the thing. There has to be some type of explanation in order to have that competing hypotheses, but I don't see no explanation for these. That's why they call them orphan genes. That's it. You know, they belong in specific taxons, and that's it.
Sure, but you're suggesting that the existence of, and I know that you mentioned that sort of the conventional science side of it would say, okay, well, you know, it's kind of this de novo emergence, but that does happen. I mean, I know you're frequently a fan of using ERVs that play roles in embryologic development because they've sort of been retrofitted. That's like a huge tenet of evolution, the atavisms, right? Like this idea of taking, you know, something that used to do something else and, you know, or not atavism, sorry, in some cases of vestiges, and using it for something new. I mean, because you're right, most ERVs though have zero function, but some of them do. And they did pop up, right? At least according to
real quick before you guys, I understand that you want to move on to the endogenous retroviruses. Just to kind of end the part on orphan genes, we can do that. I don't want to beat a dead horse. Let's move on to endogenous retroviruses and retrotransposons, for example. I would just disagree that orphan genes aren't a line of evidence that can differentiate between the two hypotheses, right? Universal common ancestry and limited ancestry. We can just agree to disagree.
And we
can. If you want to look to the de novo gene synthesis, we definitely need to see empirical data, whether it's in the lab or some type of technical paper that can actually show a non-coding region of our genome suddenly pop up and become functional as a protein coding gene.
So I kind of know where this is going. So I am going to openly in front of everyone make sure that this is clear. I don't know very much. In fact, I know very little about orphan genes. What I do know quite a bit about, or a decent amount about, is endogenous retroviruses. And the reason why I'm making the connection is because it sounds like conceptually there are quite a few similarities. So I'm not saying that I know enough about orphan genes to say this is certainly the case. I probably need to look into it, but I don't particularly find presenting a piece of evidence that, knowing absolutely nothing about it except for the blurb that you've given me here, could go either way in light of everything else that we're getting ready to discuss, convincing as a gotcha. You know what I mean? Because it sounds very similar.
There's many lines. Yeah, no, I agree.
It sounds similar to the gotchas that creationists use with ERVs. That's why I keep making the comparison. Not because I'm trying to dodge away, I don't want anybody to know I don't know very much about orphan genes. I don't. I'm not a geneticist, but that's the way the cookie crumbles sometimes. So I'm just saying I don't find that convincing. I mean, if there are, geneticists out there who are saying, yes, de novo orphan genes can happen, then the fact that we have de novo ERVs, emerging from ancient viruses and such things like this, and of course, new information through various mutations, I don't see a problem. It's completely in line with other conventional arguments that we're going to talk about.
Right. If I could just jump in real quick, because I definitely want to jump into the ERVs and the retrotransposons, and I'll give an answer to that. Just on the orphan genes and in the, it's not necessarily just a gotcha type argument. I will go on record that, the evolutionist community, they are somewhat baffled, and that is why they look to de novo gene synthesis. But to me, that's more of a circular argument, because the evolutionists know that orphan genes exist. Therefore, they need to hypothesize, why do they exist? So they resort to de novo gene synthesis. Well, why do they resort to de novo gene synthesis? Well, because orphan genes exist. So I'm just saying it's more of a circular based argument. Now, when it comes to the co-option in the functions found in endogenous retroviruses, I agree that these endogenous retroviruses, or the retrotransposons, for example, Erika, they are shared among humans, among some of the apes, of course. You can find nested hierarchies in the placement and positions of the ERV. So I agree with that. But the question I think we need to ask ourselves, Erika, is what are these endogenous retroviruses? What are these retrotransposons? Are they actually the leftover remnants of viral infections in our DNA, like you're suggesting, right? Or are they, say, created units of DNA function? Because for years, Erika, it has been assumed that they were the former, right? The leftover remnants of viral infections. And that is because, like you iterated in your opening, they bear strong similarities to actual viruses. And this predicts that these sequences are non-functional. And I know you look to co-option as an answer to the function, but this is a testable prediction based on the design model, and we are beginning to accumulate data. So what we do know at this point in time, Erika, is the trajectory of experimental results, and the trajectory points to increasing and surprising levels of function. So to me, it looks as if these are not remnants of viral infections, but actual functional DNA units. Now, what's their role in ancestry and in speciation? But this specific prediction, it does differ from creation and evolution expectations. Go ahead, take as much time as you need.
Sure. So I'm going to kind of respond to that in twofold. One, I'm not kidding here. We do know of a lot of endogenous retroviruses. We're not completely in the dark about this, and I know of like a handful that actually do something. And so I'm kind of wondering where you're coming with this sort of concept of, oh, well, the trajectory is we're going to find out that they have more function. And I think I know where you're going with it. And I think it has to do with the ENCODE project, because I've heard you speak sort of before on ENCODE. And for those of you out there who don't know, ENCODE was a sort of group that did quite a bit of genetic work on discovering the functionality of the human genome. And they came out with this quite large number, 80%, they came out to say the human genome was functional. Now, their definition of that sounds impressive until you actually read the work.
Erika, I don't want to interrupt, but I feel like ENCODE in general I just feel like ENCODE at this point is kind of a rabbit trail, because we're specifically on the endogenous retroviruses.
Sure, but you'll see the point I'm going to make. It's relevant. Okay. Because I actually watched your discussion with your series of discussions with CRISPR earlier today. And you mentioned several times this concept of, oh, well, the trajectory seems like we're going to find a lot of functions. But so I thought to myself, okay, well, time to look into that. Let's see, because I know CRISPR was saying, no, absolutely not. The vast majority of people in the field are saying, similar to ERVs, no, it's not looking like we're going to find more function, because every time they investigate, they find a function for something new. It makes up a smaller and smaller and smaller proportion of the genome. And there was a paper that came out in 2017, very recently, that put an upper cap and very unlikely upper cap of 25% of functionality, with a likely range of 10 to 12%. So that's the most recent literature on that, and it doesn't follow the trajectory that you were mentioning. So I don't think that you can say, because you find functions in one ERV, or you find a little bit of function in say, an intron or whatever, you can't just say the trajectory seems like we're going to find more and more, and then eventually we find out all ERVs are functional, or all of the human genome is functional. That's all I had to say. That was the connection.
Oh, awesome. And I appreciate that response and those points as well. And I did enjoy my discussions with CRISPR, and would definitely recommend anybody watch them. But you say that like just one endogenous retrovirus, for example, was found to be functional.
Oh, that's hyperbolic. I'm sure it's more than one, but continue.
Okay. No problem. Even in the ERVs, though, it's not just low levels of function, and that's the problem that the evolutionist faces, because the ERVs, they actually have crucial functions, Erika. For one, they help regulate genes. So they're not just functional in the embryo. They help regulate genes and even determine cell types. So I know that you're claiming that they were co-opted, but I would just say that that's mere uninformative gloss, because I would have to see empirical evidence, whether it's in the lab or some type of technical paper, that actually shows us a non-functional endogenous retrovirus going from non-functional to extremely functional in determining cell types or regulating genes. Before I continue, actually, would you be able to present something like that?
Hold on. What I want to talk about is your request there. You're saying, okay, well, I want to see.
I'm not saying you have to present it right now, but does something like that exist where it has been observed empirically that a non-functional endogenous retrovirus went from non-functional to a functional DNA element, important in the embryo, important in regulating genes, important in determining cell types? I mean, could you present that to us?
I mean, I would say you've already done so, because we have, what I mentioned earlier, right, is that we have several, and what you agreed to is that we have, so many non-functional ERVs, you can't hardly list them out on a piece of paper. And then we proceed to talk about a functional ERV. So I think the burden of proof is on you to find function for them if the vast majority is non-functional, and you're making the claim that functions do exist for all, or at least the vast majority. The burden of proof is on you to show that they have function, not on me to, because we have point A and point B.
Well, real quick.
This would be like
real quick, I don't want to, because you brought up some things about ENCODE. I was kind of just clarifying with you. And I want to kind of touch on everything, because it's not just one or two ERVs that are being found. I mean, there's accumulating paper after paper. I mean, I've got papers here from 2019 that are now showing these ERVs. They frequently act to distribute regulatory information and thus confer genes with new patterns of expression and function.
I don't doubt it. I don't doubt that there are tons of ERVs that have functions. The problem is, and I checked today, the vast majority of ERVs are functionless. And
have they functionally tested the vast majority of endogenous retroviruses, though? Because remember, real quick, evolutionists assume, and you admitted it earlier, that the vast majority of our genome is based on genomic fossils, evolutionary leftovers. But far from being junk DNA, the pervasive retrotransposons or the endogenous retroviruses, for example, that populate our genome, they have a powerful capacity to influence genes and even chromatin. I mean, the functions are endless. I can go on and on. Even the pseudogenes. We're finding. Well, I'm saying that your argument about co-option and suddenly the burden of proof is on me is more of a circular argument, just like with the orphan genes and de novo gene synthesis. So you're seeing that these real quick, I'm almost done. You're seeing that these endogenous retroviruses are playing very important functional roles in our genetics. So instead of actually giving us and providing us empirical evidence for the co-option of these endogenous retroviruses, you're just assuming that they've been co-opted because it doesn't fit the evolutionary theory, but it hasn't actually been demonstrated. And I'm saying that the trajectory of discovery does favor genome-wide functionality because every single month, more and more papers come out suggesting more and more function.
You're okay. First, I'm going to make a quick analogy. If I come out and I say, horses are the most important mode of travel, that's just what they are. And you say, well, no, that can't be so. And I say, yeah, look at the Amish. They can't get around without horses. And then you say, well, no, because mostly people get around in cars. And then I say, no, it's really, really important in this one particular case that the Amish use horses. Are you getting what I'm saying? Case. These functional ERVs have indeed been co-opted. And the way that we know that they've been co-opted, other than the fact that they look very much like ancient viruses just in structure, when we look at them genomically, is the fact that the vast majority don't do anything. And when you say things like, well, the trajectory looks like, that's a very vague statement, because the fact of the matter is your talk with CRISPR was what, at least a year ago? And I checked today and they capped it right where he said they were going to cap it. He said upper limit, I think he might have said 20%. So he was a little off. He said upper limit, no more than 20%.
Right.
2017, upper limit 25%. And real quick, I'll pull something up for you.
I'm actually happy that you've at least admitted to that, because I even debated a biologist, I think it was 2 weeks ago, and I've debated like T-jump, for example. And every time I ask that question, right, how much of our genome, both non-coding DNA and protein coding DNA, do you believe the evidence suggests is functional? And you just said it there, just like CRISPR said, it can't be any more than 20%, 25%. But the thing is, that's because any more than that makes that type of fish to fisherman evolution impossible, simply based on mutation accumulation. Well, I've got a paper here that suggests that the genome has to be under 20% functional, or else ape-to-man evolution could not. Well, actually, real quick before we go on too many rabbit trails, because I still want to touch on ENCODE, because a few things that you said I disagree with. Are you disagreeing with the fact that we do see massive amounts of literature nearly every single month that gives us unexpected functions of RNA? And then as you answer that too, how much of our genetics, of our genome, do you believe we've functionally tested according to the gold standard of genome testing? Go ahead, take your time.
So first, we can swap papers in a moment, because I've definitely, I straight up sat down today. I sat my butt down on Google Scholar and I combed for all literature that I could find that was recent on functional DNA. And the most cited paper, the most seemingly sort of groundbreaking biggie that came out this paper in 2017. Now, of course, we're always discovering new things about genetics. And of course, some of those things are new functions. But again, this conversation you had with CRISPR was at least a year ago. And right. We have discovered we haven't passed the cap. We haven't even gotten close to passing the cap that he set. These guys are saying, and I'm going to read this to you instead of sharing my screen, just because it's annoying for everybody I know, and it causes a delay.
Well, real quick before you do that, can you just answer the question as to how much of our genome has been functionally tested? Because that's important in this discussion. Because I'm going
to respond to that with the same sort of rhetoric back and forth that you and CRISPR had. So humans are mammals. And that's why we use mice and that for testing and things like that, our drugs and sort of pharmaceuticals. And that's why our pharmaceuticals tend to work so well, because we share so much in common. We have done extensive knockout tests on mice. We can knock out so much of their genome and they will be just fine. They reproduce, they have babies, their babies are just fine. So that's because that section that we're knocking out is non-functional. It doesn't harm them to knock it out. So, because I know this ENCODE conversation is going to go down maybe the non-coding or the non-functional quote unquote DNA is actually for development. That debunks it right there. The fact that these mice with enormous portions of their non-functional DNA knocked out can go and reproduce just fine. They might have a couple of weird little morphologies, but other than that, their fitness is increased.
Before you go on for too long with the mice, the problem is many non-coding RNA genes, even pseudogenes, for example, much of the non-coding regions of our genome, they're only expressed under certain conditions. And much of the genome is likely involved, for example, and used between conception and birth. And the fact that many of these non-coding sections of our genome are only expressed under certain conditions, you'd need almost unlimited sensitivity in these genetic knockout tests being done on mice to conclude with any type of certainty that those genes being knocked out really are non-functional. Because if these genes are being turned on and off, let's say after the developmental stage, or just based on environmental conditions during the life of the mice, especially because these mice, the genes are being knocked out in the laboratory. Therefore, many different environmental conditions that are outside of the laboratory, that a certain gene that isn't expressed and it's being knocked out, well, you're not going to be able to know that it's functional or non-functional. And the answer to your question, based on genetic knockout tests, especially with humans, because it's unethical, it's less than 1% of the genome that's actually been functionally tested according to the gold standard of science. So I even asked that question too. I asked that question to CRISPR, and he he went on looking for a paper to show how many of these experiments were performed based on these genes that are only expressed under certain conditions, and he never came back with the specifics on that. So you'd have to show that. I don't believe that those types of knockout experiments on mice have been performed.
To be fair, in that conversation, while he was looking, you were like, you can just get into me later. So there is always the possibility, as I frequently do, that he just forgot to send them to you. Now, I obviously
right, and that's fine. And that's fine.
Yeah, and that's fine. But I feel like you kind of obfuscated the point there, because again, for everybody who's watching, right? These knockout tests have been done on adult mice, or rather, the impacts are on adults. They reproduce. They pass those knocked out genes to their offspring, who develop just fine, and then reproduce themselves after living their merry mice life. So the point is that if you're suggesting that there is no plausible way that I can see to suggest that genes that have been knocked out, even though we've done it on enormous portions, are secretly doing some kind of function when nothing bad happens when you turn them off completely.
Well, no, I'm not saying
something. Having a bunch of already broken breakers.
Just to be sure that Erika's finished here, because you talked for a long time, and I want to just be sure Erika gets plenty. Also, you got it.
Well, I'm not saying that I'm not using a pseudoscientific argument suggesting that many non-coding RNA genes are only expressed under certain conditions. I mean, this is actually a fact. And my main point regarding that is we have only begun to discover just how functional, for one, the human genome is, and how functional the mice genome is. And I'm saying it's mind-boggling how many experiments, and I'm not saying it's impossible, but these number of experiments have not yet been done, and that's a fact.
And it's not, though, because knockouts have happened, and the mice still reproduce.
Right. But the amount of experiments that would be required to conclude, is my point, with any level of confidence that a particular set of DNA sequences has absolutely no function, is mind-boggling. And
yeah, but the amount of experiments that you would have to do to conclude anything without beyond a shadow of a doubt is astronomical. The point is that the fact that you can knock out enormous portions of a mouse, and then the mouse can reproduce, and then its offspring can also reproduce because nothing of merit has been lost, is parsimonious only with evolutionary theory, that's what I'm saying. To use your word from earlier, the trajectory indicates that enormous portions of the genome can indeed be knocked out because they don't do anything. So, and I want to segue this in because
I didn't say that they don't do anything. I'm saying a lot of those regions in the because the non-coding DNA, it functions much like a computer. Okay, it's involved in regulatory functions, it's involved in embryonic development, it's even involved in the stagings of proteins for.
But they can't regulate if they're off.
But I'm
if
saying
you knock them out, they can't do anything.
No, but if these specific genes, and we have to look at which ones, what types of developmental windows, which types of environmental conditions, because if these experiments are being done strictly in the lab on these mice, well, these non-coding RNA genes, for example, even some of the pseudo genes, yeah, you're going to knock that out, you're going to snip it out, and it might not have some overall effect on genotype or phenotype, but that's because it's just not expressed.
Yeah, so but we're not talking about.
Just real quick, if you consider that, and then you consider, if you look at comprehensive knockout experiments for humans, for example, it's unethical, obviously, at this point. We functionally tested, genetics-wise, under 1% of the genome when you consider non-coding DNA and coding protein coding DNA. But the thing is, and the question I asked you earlier regarding the RNA, we do see massive amounts of literature that gives us unexpected functions of RNA. For example, are you familiar with protein moonlighting? Because
yes,
in the past,
of course.
Right. In the past, one of the ideas, you can just pick up a textbook from 40, 50 years ago.
One gene, one function, or protein, one function. Yes, I'm with you. Continue.
Exactly. Now, if we're looking at the protein multifunctionality, or we're looking at, say, on an amino acid level, these redundant codons, and I'm sure you've heard me say this before, those were always assumed to be absolutely neutral. Now we know they don't just tolerate error in the cell. These redundant elements, they're used to slow and speed the process. So the level of information compression into every single letter boggles the mind. It's actually incredible. And even
I'm sure that is indeed the case, but that's not the problem that I have with your statement that and in this conversation and in previous ones, that the majority of the genome is functional. The fact of the matter is, if you knock it out, it doesn't work. It doesn't do anything. If it doesn't do anything before you knock it out, there's no harm, no foul, right? So the fact of the matter is, if they do something important, anything, regulation, whatever, it doesn't matter. If they have a job that's even minute, if you knock it out, that job doesn't get done.
How much of the mice genome has been knocked out, though, on a percentage-wise?
Oh, I could not give you a number on that. If you
well, I could, and I could be corrected on this, but the last time I checked, it is less than 30%. So based on the fact that these tests are being done in the lab, based on the fact that less than 30% of the mice genome has been knocked out, and the fact that, as I've said both 3 or 4 times, many non-coding RNA genes are only expressed under certain conditions, less than 1% of the mice genome, the human genome, mammals in general, has been tested for genome functionality. And all you have to look at, like I talked about earlier, you just have to look at the history of genetics. Like you said, back in the 40s, 1 gene, 1 protein, 1 function. I've said that multiple times. You can see how many things have been changed. So yeah, I agree that the results from the ENCODE project, Erika, I do agree that based on their suggestion that at least 80% of the genome is functional biochemically, right? Because it's showing that the non-coding DNA is transcribed into RNA. Those are preliminary. I agree with that, but I'm saying the trajectory is clear based on genome testing, based on the history of genetics, that the trajectory does suggest most non-coding DNA in the human genome is functional. At least it serves in a regulatory capacity. I mean, do you stand by a genome of only 20% max functionality?
Here's the thing, Standing. I go by current literature. And what I try to do when I don't know something about a topic is I check the most recent literature, I see how many times it's been cited, and I see who's on the paper and investigate their background. Because that's, it's just like what they teach you. It's appropriate sourcing. And I'm going to read you this excerpt. It's quite short from this 2017 paper, which was New Limits to Functional Portion of Human Genome Reported. An evolutionary biologist at the University of Houston has published new calculations that indicate no more than 25% of the human genome is functional. This is a stark contrast to the suggestions made by the scientist with the ENCODE project that as much as 80% of the genome was indeed functional. They took a deceptively simple approach, basically, to sort of sussing this out by looking at deleterious mutation rates and replacement fertility rate, which is quite odd, and I wouldn't have thought to do it because I'm also not a geneticist and that's not really my world. But this is the quote that I think is important. This is from the paper. It's Grauer et al. If you want to look it up.
I've got that in front of me.
Or I can send it to you, yeah, too.
I have it in front of me.
Oh, good. Okay. For 80% of the human genome to be functional, each couple in the world would have to beget an average of 15 children, and all but 2 would have to die or fail to reproduce, he wrote. If we use the upper bound for the deleterious mutation rate, which is at 2 × 10⁸ mutations per nucleotide per generation, then the number of children that each couple would have to maintain a constant population size would exceed the number of stars in the visible universe by 10 orders of magnitude. So this is the most recent literature. Not only does it say, yeah, probably no more than 25%, they're saying it's impossible. So that is evidence that precludes, it's just using math, deleterious mutations and reproductive rates, replacement rates and things like that. And it directly precludes what you just said about trajectory. Because like you said, these papers come out every day. And every time the new DNA that they find that's functional, the new portion of the genome, gets smaller and smaller and smaller. They might say, oh, wow, we found out that 0.15% of this, given portion is functional. Add it to the list, but it's sort of exponential. There's not that much more to kind of sort of complete what we know, at least about the proportion of what is and what isn't functional. I encourage you to read the paper because I'm not a genetics person, but I found it quite easy to understand. And the math is pretty straightforward.
Right. So I appreciate that response. So in that paper, what Gore says, he says that human evolution would be very problematic if the genome was 10% functional, but would be completely impossible if 25% or more was functional. So what he's demonstrating here is what results from an evolutionary mindset, because he knows, based on the possible functionality of the genome, if we look at mutations, if we look at what's inherited, mutation accumulation in general, it would make that type of ape to man evolution impossible, because of the fact that if you have a genome that is, say, 50% functional, 60% functional, well, that means these so-called neutral mutations that supposedly build up with no real effect on phenotype and genotype would actually be more so nearly neutral.
Right.
That
so
means
you're saying there's a heavy bias, is what you're saying.
I'm saying there's a heavy bias, and you can even see that with the ENCODE researchers themselves. You can see that they did their research without that evolutionary bias. And evolutionists like the author here in this paper, they would have to fight the results of ENCODE. They would have to fight any type of trajectory that suggests the genome is any more than 20 or 25% functional based on the accumulation of mutations. Because, for example, Komura's theory of neutral mutations, right? Neutral mutations build up. Those neutral mutations are used over time by organisms to change, to adapt, to modify. But
well, Komura, come on. Komura was taken greatly out of context. I looked into that as well, with Jameson or Tompkins. I can't remember which one. But I want to make a very short, brief comment.
A real quick before you do go on to that, and you take as much time as you need. The point that I'm making here is, obviously I disagree with the idea, or with the belief that the genome is 20% functional or 25% non-functional or functional max. Because, and even if I were to just say concede that, or just choose to agree to disagree, even if though 90% of the genome is perfectly neutral junk DNA, say only 10% of it is functional, that's still a huge problem, because based on what we know about the human mutation rate, right? We accumulate 100 new mutations per person per generation. That means, Erika, if I were to just agree with what you're saying, about 10 harmful mutations are still arising in every person, every generation. That means even the most fit individuals are still more mutant than their parents. And so even with intense selection, if we're looking at natural selection here, against the less fit, it cannot stop mutation accumulation. But like that paper suggests, the more functional the genome is, the more of a problem genetic degeneration becomes. Okay, take as much time as you need.
Yes, so here's the thing, and I'm going to use this to segue into genetic entropy, because I feel like that's kind of where you're pulling a little bit, which is fine, because I got my notes right here, because I don't know enough about genetics just off the top of my head, give at least numbers. But I want to make a quick comment about bias. Now, in the social sciences, anything that require, many areas of science that require an interpretation of the data, bias can be problematic. Here's where bias can't be problematic: with math. When you're just doing equations based off of either known rates or variable rates or whatever, as long as you've got your error bars, they're going to cover your bias. That's what they're there for, to say, okay, well, it's give or take, right? So you can say, yeah, he's biased to evolution, so's the vast majority of science. And I would agree with you because I am also biased towards evolutionary theory, but it doesn't matter because all he did was use math to get his point across. He plugged in equations. You can say I disagree with it, but that's the same thing as being like, I disagree with if whatever the Pythagorean theorem or something like that, just like an equation. Now, if you went in and you discovered, I guess that's a bad example. If I showed you an equation and you were like, I disagree with it. Now, if you go in there and you find an actual problem with it, that's another story. But as far as bias goes, it's not problematic when you're dealing with mathematics or physics or things like that. Or at least it's far less of a problem because there's less room for interpretation. Now, as far as genetic entropy goes, it's not real. Genetic entropy, or at least the conventional science term, is error catastrophe. And it doesn't happen for several reasons. If you've got, go ahead and give me, I want to make sure that I'm kind of addressing the proper point so I don't go on a big spiel and then we're talking about something different. When you talk about genetic entropy, are you talking about the accumulation of near neutral mutations that causes degeneration?
I would say if you look at genetic entropy or genetic degeneration, for example, it is the fact that natural selection cannot stop the accumulation of most harmful mutations. The problem is that the human mutation rate, for example, it is widely accepted to be 100 new mutations per person per generation. Now, even if I were to agree to disagree on your point or assertion that, you know, max functionality of the genome is say 20%, 25%, even though I want to make one last point about the function of the genome anyways, I would say that it is a fact that ENCODE, which you were referring to earlier, it does at least show that most of the human genome is functional, but also functional on many layers. Like we literally have layer upon layer upon layer of programming, Erika, within the genome. And no computer program has ever been seen to arise by trial and error or by chance. But genetic entropy would suggest that these mutations that evolutionists are saying drive the evolutionary process are either nearly neutral, which are slightly deleterious to the genetics, or detrimental. And that's the problem, is what type of selection is going to be able to filter out all those bad mutations, whether nearly neutral or just detrimental. Go ahead.
So the thing about ENCODE, again, just to give my, I guess, final point on it, for those of you who are watching, one, the definition of functionality is incredibly loose. And if you watch Stanning's conversation with CRISPR, who is has sort of hands in many different fields. He knows quite a few people who are, he's a cancer guy himself, but he knows quite a few people working in various areas of bioinformatics and things like that. And his conclusion was that he could maybe find 2 people out of the dozens upon dozens upon dozens that he works with who would agree that the definition of functionality was even remotely appropriate, because their definition of functionality covers everything. And the reason is because that's how you sell a project. I've been trying to sell my project for grants, because I'm doing a master's program right now, for several weeks, and you have to make it sound good or else you don't get the funding. So, and that I'm not saying that like it's anecdotal. That's how it is. That's just academia.
And just for the record, I'm not disagreeing with the definition that ENCODE used and the definition that CRISPR says that they use, right? Because ENCODE defined function as specific biochemical activity. And that's why I agree with you that the results suggesting that over 80% of the genome is actively transcribed into RNA is preliminary. That's why we've made a prediction as creationists that based on further genetic testing, knockout tests, for example, or testing mice under various types of environmental conditions where these non-coding RNA genes can be expressed in various ways is functional. That's our future prediction. And I think
yes, of course. And my answer to that would be ENCODE has already been busted by this 2017 paper. And I think there was another one in 2016 maybe, but I picked the most recent one because good practice, I suppose.
Do you know how many, I think this is an important question before you move on. Do you know how many cell types ENCODE tested?
Oh, no, I don't.
Well, here's the
I
problem.
could not tell you.
Here's the problem. So based on what ENCODE discovered regarding the function of our DNA, I understand that they defined it as specific biochemical activity. We have more than 1,000, and I'm sure you already know this, we have more than 1,000 cell types. So I'm saying, and I'm saying this to you in the audience and to James, the moderator, I'm saying that more function is going to be discovered in the future. Because if you're testing only 200 cell types and we have over 1,000 cell types in our genetics, I think it's just based on an evolutionary bias to say that max functionality of our genome is 20 or 25%. And I know that the reason why he concluded that in the paper is based on mutation accumulation. Because if it's any more than that, mutation accumulation makes ape to man evolution impossible. That's a fact.
The problem though is that I don't know excuse me, I'm not sure for certain where you're getting your mutation rates from. I have a hunch that it's GeneSense rates.
The survey said,
and or at least Kimura, the people that GeneSense sort of pulled from. And if I'm correct on that, which I don't know that I am.
Are you saying the 100? Are you saying the 100 new mutations per person per generation? I mean, you could find that all over the place.
Whichever mitochondrial rate, mutation rate, and nuclear mutation rate that he uses to make the case in replacing Darwin. Am I correct on that or no?
No, well, right now, I mean, if you want to get into replacing Darwin and the created heterozygosity hypothesis and mitochondrial DNA mutation rates and stuff, I mean, that's a totally different topic than
all I'm asking is it the same mutation rate that GeneSense uses that you're using to say it over 25%, or not over 25%, but the deleterious mutation buildup would make ape to man evolution impossible?
No, that's just secular literature. I mean, the human mutation rate is generally known to be 60 to 100 new mutations per person per generation. I believe that's on trio studies.
Yeah, I was mostly just curious because there are potentially infinite problems with the way that GeneSense did his methodology.
Right, and we can go into that, but that's a totally different topic.
That's why I was just checking. It's up to you. We can talk about genetic entropy, or we can talk about the LMN haplogroups, whatever you want to talk about. I'm good to talk about it. My closing thoughts are, I think that you got to look to the current literature, and I think that it was handily proven exclusively with math that there is a 25% cap.
We are coming. Hold on one sec. We are coming to the closing of the open discussion pretty quick here. So let's see. We had standing start, and so if you want to do one quick rebuttal, hold on 1 second.
Discussion-wise,
it's been about 50 minutes.
Well, can we bump it to 60 minutes so we can touch on just a couple more things here, or it's up to you? I know it's getting late for Erika.
I don't have a preference. It's up to you guys. I can stay up 10 minutes later.
That 50 minutes went by fast, Erika. I think we could go at this all night.
I think what we can do is, if we want to, how about we do this? Instead of an extra 10 or 15 minutes, if we want to just standing for truth, we can give you another round to kind of make your points, and then we'll give Erika one more round to make her points, and each of those being just a few minutes.
That's fine.
Okay. Should we do a couple more minutes of back and forth, and then, because that kind of sounds like it would be like almost like a concluding statement if we're just going to go a few minutes uninterrupted. I mean, whatever you guys think is best.
Yeah, so that's right. You had heard right. So we'll just have it be like a few minutes kind of making your points, and then we'll give
Sneaky
Erika a
James
chance to make her few points, and then we'll wrap it up.
Okay, who wants to start then their few minutes final points?
As I just said, given that you were about to start, like, you can now start, and then...
Okay, so I think since what we were kind of heading into there is genetic entropy, you can answer the question as to, what type of selection is going to be able to filter out all the bad mutations, because the major issue is the fact that selection it can amplify the best beneficial mutations and it can get rid of the worst detrimental mutations, but it's pretty well powerless to the nearly neutral mutations. Those are invisible to natural selection. I mean, you could think of it this way, Erika. I mean, we've got a genome of what, 3 billion letters. 3 billion are passed on from mom, 3 billion passed on from dad. But if you take out one letter, say change one letter randomly, are you going to have a huge fitness effect? No, it's going to have a tiny fitness effect. It's kind of like rust on a car. You can't see each rust event, but it's continuous and destructive. Or like a single spelling mistake in a book the size of an encyclopedia, right? Make one single spelling mistake. It's not going to have a huge overall effect, but the accumulation of them over time, yeah, that's going to destroy the message and the book. But why can't natural selection filter out all the bad mutations, Erika? There's two reasons here that you'll have to address. There are too many bad mutations continuously pouring into the population, and most bad mutations are too subtle, as I talked about earlier, to be selectively removed. And we see genetic entropy in real time. Alzheimer's disease, dementia, autism clearly has a genetic component to it. Asthma, autoimmune diseases, cancers, what is it, 1 in 3 now? Immunological disorders, diseases. I mean, there is a pandemic. It's pretty well entropic degeneration on all levels. And the 100 mutations per person per generation is a fact. And you're going to have to address how that accumulation of mutations is going to be rid of in our genetics, because I mean that's what we see. We see genes are going down and not up. And if fish to fisherman is true, I mean, you're going to have to explain the major problem, which is a net gain versus a net loss of information. So, I mean, if you want to kind of address those points and give me your thoughts on genetic entropy and what your best argument is against genetic entropy, I guess that would be a good way to proceed.
Okay. So genetic entropy does not happen. The ecological term, again, is error catastrophe. And the reason why it doesn't happen, even though ecologists who have no affiliation with creation, evolution, anything along those lines, have tried to induce error catastrophe and have failed time and time again. They've done it with E. coli, they've done it with phages. It doesn't work. And the reason they do it with those is because they accumulate mutations way faster than sort of macroscopic life. So they can see if deleterious mutations are going to build up in a rather short amount of time. And here's the reason why they don't. Near neutral mutations, if they're near neutral and sort of beneficial, then they're going to be slightly selected for. Selection isn't an all or nothing thing, right? Let's say you have a near neutral mutation that is sort of a negative, right? It might not kill you, but it might decrease your fitness, because if it is a negative trait, right? If it is a bad mutation, by definition, it's decreasing your fitness. So you're going to reproduce less than your peers, and whatever offspring that you do manage to produce will reproduce less than their peers, and they will continue to pass on that sort of slightly bad mutation until eventually it is eradicated from the population by one reason or another. It might simply be missed reproductive opportunity. So if it is even remotely bad, selection acts against it. That's just how it is. If it's neutral, and when I say neutral, I mean truly neutral, they can accumulate all they want. Nothing's going to happen. There's nothing to select against. That's the definition of something that is neutral. There is no good or bad kind of impact to it. Now, what you might have in sort of your best case scenario is if an entire group of organisms all get bad mutations at the same time. And as they build up upon one another, they sort of have I think it's epistatic, sort of an impact on where they build up, and it makes them subsequently worse when they combine with one another. But the problem with that is the definition of genetic entropy and of error catastrophe means that it has to accumulate across generations. Regardless of whether it's epistatic or not, the negative mutation will hit the second, and negative selection against sort of this trait will happen the second fitness is reduced. One way or another, that's happening in one generation. So there is no plausible way to suggest that error catastrophe or genetic entropy can occur, because it is the accumulation of bad mutations across generations that eventually harm fitness. We don't see that. Ecologists have tried. Understanding error catastrophe, because cancers and things like that aren't error catastrophe. They're similar in ways, but again, it's not cross-generational. So understanding it would be quite helpful for fighting those diseases, but they can't do it because it's not a phenomenon that occurs. Sanford, I believe, claims it happened with H1N1, but I read that paper. Not only is it a bad paper that actually makes several mistakes, but it swaps its definition of error catastrophe around whenever convenient. So essentially, my answer to your question is there is no such thing as genetic entropy. We don't see it naturally. We don't see it in a lab, even though we've tried. At least the definition that creationists give. So it's not a problem because it's not real.
Gotcha.
That's all I got to say about it.
Thank you very much. And standing for truth, I know that you have another round in the barrel, but just for the sake of
too well, James.
We can always do the tiebreaker debate. We can do an end game debate. We should call them that from now on.
That's a good idea.
The trilogy end debate where we have the final of 3. So we definitely can do that, but I do just want to make sure that Erika gets to sleep on a decent, hopefully at a decent time.
You're
so
sweet, James.
It's
I will
no
say
problem.
That conversation flew by. It was a lot of fun.
It's
I
also,
had a good time.
It's also because I'm tired. Now, next up, Steven Steen, thanks for your super chat. He says, standing for truth cannot be stopped. He's a beast. SpongeBob. You have a fan out there.
That is far too sweet of Steven to say. I obviously paid him to say that before the debate. If you could read a couple more super chats, I'm running upstairs for literally 30 seconds, James.
Deal. Steven Steen, that sicko, also says, James is the new Adam sent from God to fix us soy boys' flawed genes and restart the human race without flaw or weak jaws.
Listen, James, I have heard you are an alpha Chad. I've heard that from other people.
That's hilarious. An alpha Chad. I had never heard that before, but I like that. We'll call Steven Alpha Chad from now on. Let's see. Bent Hovend. No relation, it says in the name. Says Erika and Dapper Dino should debate standing for truth and Kent Hovend at the same time.
I'd be down for that.
That would be epic.
Be fun. Could be fun.
We actually, I forgot to mention. So we do actually, it's coming up quick. We do have, as you see in the bottom right, Kent Hovend will be returning. We got off on the wrong foot one day, and I will take responsibility for it. And long story short, for a while, it looks like we weren't going to have Kent Hovend on anymore. But Mark Drisdale convinced him. So you see Mark in the picture there. He, on the left side, he looks kind of like an action hero star, the bald head and the goatee. He has convinced Hovend to come back on. So that will be a good one. But you don't want
to get on Kent's bad side. He'll ban you from Dinosaur Adventureland.
That's true. So it's also Genesis and cosmology. A discussion will be going on this coming Monday with Skydive Phil, popular YouTuber, really, really pleasant guy, and also Dr. Jeff Zwierink. So that's going to be a really fun one. That's one that we've had planned on 2 different dates because I was just, I don't know what was wrong with me. It's the first time I had ever wondered, oh my gosh, really wondered. Is there something wrong with me? Because I really thought, so they contacted me, they called me, they're like, James, it's not today. And I was like, oh no, you're right. So long story short, glad you're back standing for truth. I could hear
You've only wondered that once. I wonder that every day.
Yes.
About myself.
Well, thank you. I appreciate that. And standing for truth, I think I could hear you cackling in the back. You must be back.
I'm back, my friend.
Gotcha. Thanks so much. So
wanted to make myself known.
You betcha. Let's see. Michael Dresden with his normal trollish super chat says, I'll save you all the time. Evolution is a myth. You can respond to that, Erika, or you can not dignify it and ignore it.
I'll respond. You're wrong.
Gotcha.
Yeah.
It's short and pithy. I like it. Speed of sound of gravity. Who says, by the way, he said that he or she said, standing for truth, you are afraid to debate them. So they said, standing for truth, buck, buck, bugguck.
Buck, bagok. Like, you know.
Okay,
yeah,
get
Speed of Sound sends his regards standing. He says he wants a rematch, so.
Okay,
Love it.
so and that's funny. Erika mentions that. So I've had 2 debates with speed of sound. So definitely over 2 hours, maybe 2.5 hours of dialogue. We debated the same topic that Erika and I just debated, and a topic specific to chromosome 2 fusion. So I recommend anybody go look at those debates. I suspect he only wants another debate because of something called damage control. He knows he lost, but you know what, Mike? Maybe one day. I'm going after the big dogs now, though.
Gotcha. Very sassy.
That's what they call me.
That's right. Jay Shy, thanks for your super chat, said, what if there were people outside the garden whom evolved, then mated with Adam's descendants, still making Adam and Eve our genealogical ancestor? Cain hints to other people who might kill him. So, standing for truth.
Right. Well, I would say that we don't even need to go that far because the Bible tells us we came from just 2 people, Adam and Eve, of course. And if you just look at my opening, I went over the fact that we've got these pieces of DNA, for example, the Y chromosome DNA, the Y chromosome we only get from our fathers. And it's passed on, unbroken from father to son. And every once in a while, a mistake happens. And every time this happens, a new branch in the family tree is formed. So all we have to do is look at all the branches in the world, James. They go back to a single person, not to a chimpanzee, okay? And that's what this debate was, limited ancestry versus universal common ancestry, but to a man who lived just a few thousand years ago. And the same thing goes with mitochondrial Eve. So we can just believe the Bible. And when we start with the Bible, as it's just plainly understood, we can make accurate predictions and retrodictions.
Gotcha. Thank you very
James,
much.
if I can make a recommendation for some reading for that individual, they should check out John Walton's series, because he actually subscribes precisely to that, what the question asker was talking about, sort of like a theistic evolution type thing. I've read some of his work. He's an academic. He seriously knows what he's talking about. I'd give it a look. It's pretty interesting stuff.
Gotcha. Thanks so much. And next up, Stupid Whore Energy, as she likes to call herself. Very sick. She says,
I haven't heard from her in a while.
That's right. She says, new genes can arise from non-coding sequences. That's why orphans are not a problem.
Right. So it's just as I iterated earlier. It's called de novo gene synthesis. And I don't want to beat a dead horse, but just go back and watch the discussion. It is a circular-based argument. It's never been observed that these non-functional, non-coding DNA regions are suddenly co-opted to have an important functional role. But orphan genes in general, they are taxonomically restricted. They are functional, and they do suggest, along with many other lines of evidence, that limited ancestry is true, because there's so many lines of evidence that the evolutionists have a major problem explaining, other than weak rescue devices, in my opinion.
Gotcha. Very sassy. Go ahead.
As a question sort of for standing, what do you make of all the astrological phenomena that we draw ties to despite never having actually laid eyes on them in person, like black hole physics or supernova or novae, I guess, neutron stars, things like that? We just get data, we just get readings, and we formulate entire hypotheses about them that then are confirmed by other data that we receive. So you don't have to be a direct observer of something to know that it happened. You can see the aftermath and get the same idea. That's where a lot of science goes. Anyways, that's just an anecdote.
I agree. I mean, you can have all types of hypotheses, but those hypotheses have to have supporting evidence. The evidence has to be demonstrable. It has to be empirical. So some of the things you just mentioned are demonstrable, and the inferences that are made are based on really tight scientific-based evidence. But when it comes to like the Y chromosome dissimilarity between chimps and humans,
we didn't get to talk about that, and I researched that this time, and I was ready for that. We didn't get to talk about it. Dang,
well, remember,
we didn't get to talk about a lot of stuff.
Well, remember, round 3, Endgame, Avengers style.
Listen, eventually I want an Age of the Earth conversation, okay? Or we can talk about anthropology again. I don't like being out of my element.
I enjoyed the human evolution debate, and now we did one on this topic. So yeah, maybe the next one can be Age of the Earth related, or looking forward to it. Put it that way.
Very juicy. Mike Billers, thanks for your super chat. They ask, can Standing for Truth re-explain Erika's explanation for orphan genes and maybe stop using it in the future since it's explained?
Well, I know the answer to orphan genes that is given, because it is a major problem to evolutionist theory, is de novo gene synthesis. I can't remember if that's the argument that Erika used. She, I guess, looked to endogenous retroviruses, for example, and the functions that are now been found in these so-called ancient viral elements have been co-opted. It just comes down to the fact that, are these anything more than just stories, rescue devices? So I understand the arguments put forth by the evolutionists. I just don't see them as being very convincing.
Gotcha. Thanks very much. Moving quickly, Josiah Hansen, thanks for your super chat, asked, Erika, protect your hand when giving such a spanking. Very nice. Gotcha. Excellent. Let's see. Next up, thanks for your super chat from Florida Man Edo Hypnotoad Evangelist. Appreciate it.
Blessings on you. Priest.
Gotcha. Thank you very much. Let's see. They're asking, give me 1 second. They asked, why can't creation answer the phylogeny challenge?
I'm guessing that question's for me.
Yep.
I would say that the phylogeny challenge is fallacious, because it all comes down to, where can we draw the line regarding ancestry? Where can we, where does that phylogeny break down? But the thing is, that entire argument misunderstands biology and genetics, because and theology, because they seem to imagine that God created humans and then all of other biological life, and that there's absolutely no similarity between humans and any other species out there. So based on the biblical model, there's really no reason for us to believe that that's true, as in, there should be no type of hierarchy in the biological world. As in, there's no reason to believe that we shouldn't be more similar to, say, apes than to another species, say a whale, right? So we too predict nested hierarchies in biology, whether it's on an anatomical level, physiological, morphological nested hierarchies. So that's why I always say we need to look to differentiating kinds of evidence to show which explanation, limited ancestry or universal ancestry, is true. And that's where we can get into function. We can get into Y chromosome, mitochondrial DNA, predictions. This is how we're going to truly answer the question of ancestry. So long story short, the phylogeny challenge it's fallacious, and it's based on a misunderstanding of science and theology.
Hey, James, can I answer this one too? But just from my side.
You bet.
I think the best question that the question that I have for creationists on this one, of course, has to do with primates, because I love primates and I am a primate. But frequently creationists lump all the apes into an ape kind. And my question kind of runs along those lines, because frequently a kind is described as things that bring forth, organisms that can bring forth with one another. But chimpanzees obviously can't reproduce with gorillas or orangs, or, well, they can with bonobos, but they're actually very recent divergence. And there's been slightly disturbing, but a bit of sort of theoretical research looking into how reproductively compatible humans would be with chimpanzees. And it's more than it is with gorillas, and it's not actually been considered to be impossible, as terrible as that sounds. It's simply a ring around the egg, the zona pellucida, that sort of gate checks any spermatozoans or sperm in general that try to come in. So reproductive isolation is kind of a bad way of drawing the line, which is why I always ask why and how we draw it. And that's why I don't think they can answer the challenge, because there is no way to draw a line. Genetics is a gradient, and it runs through all living things. And that's why paternity tests work. And that's why, genomic comparisons work.
Well, if I could make just one quick point.
Really
if you just
quick.
think about it, Erika, okay, according to our model, I'm just going to throw some, kinds out there, say humans, pigs, rats, ducks, and dogs. They're all land dwelling, okay? Even though they are separate kinds according to our model, since they are all land dwelling and share many similarities with each other, they can share similar nested hierarchies or designs. So where are we going to draw the line between those separate kinds if they already share certain levels of nested hierarchies, morphologically speaking, genetically speaking, physiologically, speaking? So that's the thing.
But that's a fancy way of saying you can't draw the line. That's a fancy way of obfuscating that the line can't be drawn. And if your model is correct, then the line should be able to be drawn, because it is, of all the things in Genesis that are like Genesis 1 that are like super clear, there's quite a bit of emphasis on the kinds. It's repeated over and over and over again. You would think it would be quite obvious.
Well, even though we predict nested hierarchies, evolutionists, when they say that there's this perfect nested hierarchy, there's this perfect phylogenetic tree of life, that's not actually true, because anytime there's an inconsistency, Erika, and say a lack of uniqueness in that phylogenetic tree, as you know, it's usually explained away by what? Convergent evolution. So that's a rescue device. There's also something called independent lineage sorting. If you look at different genes in, say, humans or apes or gorillas or orangutans, depending on the gene you're looking, can create a totally different type of phylogenetic tree. So even though you're implying that there's this perfect nested hierarchy of life, that's not true. There's many, many types of inconsistencies I can point to.
Gotcha.
Yeah, but the thing is the inconsistencies are, while present, they're present in every branch of science, from medicine to astronomy to, of course, evolutionary biology. That's the nature of the beast. The question is, how strong and how parsimonious are the lineages that we do have these straight through lines? And the answer is incredibly. I know primates the best. It's undeniable from my position. But that's just me.
We have to standing for truth. I'm sorry, but just to get through more questions, we have to jump through.
That could have been debate number 3.
Yes. Dapper Dino, thanks for your question. Asking to both, are you familiar with the observed de novo emergence in Drosophila or Saccharomyces?
Yeah, so the fruit flies, for example, read that paper. For one, it's done in the lab. It's also inferred. And what they've done in the lab is they've forced these orphan genes out of the genetics of these fruit flies. So he's trying to say that this is an example of de novo gene synthesis, when all it is an example of a loss of orphan genes, which can happen, obviously. So I want to see an example in real time, not in the lab, of de novo gene synthesis, and not just an example of an orphan gene being lost. That's just...
How would you even do that, though? How would you see the emergence of a de novo orphan gene not in the lab? You're not going to, I don't know, maybe you follow a colony of Drosophila around with a net, and you catch them every couple minutes, and then you do some genomic sequencing to find out. I don't know how you would even go about doing that. It's an impossible experiment.
So are you admitting that it's never been seen?
No, it has in the lab, but you would just ask for it outside of the lab, so you can't do it outside.
It hasn't been seen in the lab. I just told you that all that was seen was the loss of orphan genes. That's not the gain of orphan genes, Erika.
Hold on, hold that thought. I'm not sure. Let me pull up this paper. James, pull up the next question, if you would, please.
Yes. Next up, thanks for your super chat, Pax Americana, asking, standing for truth, even early church fathers such as St. Augustine knew that Genesis wasn't a literal account of creation. YEC isn't even theologically sound.
Technically, if you look at what the plain reading of the Bible is, if you look at church fathers, quote unquote, throughout history, the majority of them did believe that the geology of the earth was explained by Noah's flood, Hans come to people evolution was not heard of. But the most important thing is we can just look at a plain reading of scripture and we can see that God created Adam and Eve, and we can also make predictions and retrodictions. And the science tells us we come from a Y chromosome Adam, a mitochondrial Eve. The human species overall has low genetic diversity, which points directly to Adam and Eve. That's why the evolutionists had to resort to the out of Africa population bottleneck theory. I mean, Erika and I discussed that thoroughly in our previous debate, but a plain reading of Genesis clearly is consistent with empirical scientific data. So
look into the Toba catastrophe, if you would, Standing. That's one of the primary explanations for the bottleneck that there's quite a bit of evidence for.
Right, and I did, and it looks like there's no major consensus on whether or not this Toba volcanic eruption is resulted in the population bottlenecks. There's, I believe, some sides in science that believe there's some inconsistencies with that theory, while others hold to it. So, I mean, that just might be a discussion for another debate.
Yeah, I think that it's best explained with a combination of that and founder effect from populations leaving Africa. But that's just me.
Just to move through as fast as possible. Jay Shy, thanks for your super chat. They said, Francis Collins, a Christian, mapped out the human genome and is convinced of universal common descent. Why didn't this lead him to your conclusion? I think that he's asking Standing for Truth that.
So he looked at, actually, can you repeat that real quick?
They said, Francis Collins, a Christian, mapped out the human genome and is convinced of universal common descent. Why didn't this lead him to your conclusion, Standing for Truth?
Good question. So Francis Collins, I definitely recommend his book, The Language of God. The major arguments he looks to, for example, you'll see him speak of ancient repetitive elements, the non-functionality of our genome, shared genetic mistakes, for example. But he's had to actually go back on a lot of that because he's one of the scientists who has admitted that ENCODE has revealed that much more of the genome is functional, that the genome has many more functional DNA elements than previously thought. And even Richard Dawkins has had to go back on that. I mean, he's the one who quoted, and I don't have it right in front of me, that over 90% of our genome may as well not even be there, implying that most of it is useless. Yeah, Richard Dawkins, just go knock out that 90% of the genome. So it's just based on an evolutionary bias. But yeah, Francis Collins, I'd recommend reading his book, and all his best evidence for universal common ancestry has been completely overturned, just like the junk DNA paradigm has been overturned.
Gotcha.
Now, that 2017 paper disagrees with you, though, Standing. That one doesn't quite agree that 90% couldn't be knocked out if we're dealing with the 10 to 12 range, which is pretty typical. I'm sending you a paper, Standing. I think this is the one that was being discussed. Go ahead, James. Sorry.
Gotcha. And then, let's see. Kevin Tremblay, our friendly moderator, if you can do me a favor, and we usually only time people out or delete their messages if they are using hate speech. So even if they're abusive, like, please ask them, like, why they're being abusive towards somebody. But in other words, like, try to discourage them. But we usually try to only delete or ban people if they're engaging in hate speech. Next up, Mr. Archaeopteryx, Standing for Truth, an amoeba genome is 100 times bigger than a human's. Does an amoeba need 100 times more information, or is most of it non-functional?
Well, amoebas, looking at an amoeba genome, is a lot more different than, say, testing the genetics of mammals, humans specifically, which is where we're making the direct predictions. But organisms like amoebas, yeah, they are subject to whole genome duplications. Also redundancy, and in a lot of these organisms that do have 2 or 3 or 4 times the amount of genetic content than, say, a human, may just need more redundancy for environmental conditions. So, I mean, epigenetics, environment all play a factor in our assessment of the genomes of all forms of life. So, yeah, amoebas, they are subject to whole genome duplication. So some of it could certainly be non-functional.
Hey,
Gotcha.
James,
Yep.
real fast. For those of you who also want the paper that I just sent standing, it covers the orphan genes quite well. There's some excellent sources within it covering orphan gene emergence, which I believe, I didn't get a chance to read the entire thing, but it's sort of alluding to the fact that this has been observed in the lab. Not positive. Don't quote me on that. Side note, I also had the chance to briefly look, hold on, pause. I'm pulling it up, look at my email, and I had a message from Call Me Emo, who brings up an excellent point that I wanted to post to standing very briefly. I think it's if that's okay with you, James. It's very short.
Yes.
Emo, yes, I've seen his argument, and we've
Well,
rebutted.
hold on. I'll ask you the question because it's quite short. He brings up an excellent point that I didn't even think of, but that
So am I debating emo now too?
Well, I saw where I was sitting.
Let me just go Google some arguments against what you said. Okay, go ahead. Yeah, what's the
listen, I just think it's a good point. It's not my point, so that's by far no. But I did think it was quite good. He brings up the fact that orphan genes, if even within kinds, vary wildly from species to species, which would support de novo origin far more than sort of a taxonomically restrained, limited ancestry.
Well, he assumes that those orphan genes are found within kind. So, for example, you can find orphan genes that are taxonomically restricted to humans, gorillas, and chimps. But he's assuming that gorillas and chimps are of the same created kind. That might just indicate that they are three separate created kinds. And the paper that you posted on the fruit flies, for example, we have an article in our book, Raw Matt and myself, debunking that. What they did technically was they forced adult males to reproduce with infant females to get rid of their orphan genes. So it's unrealistic in nature and therefore irrelevant to the entire argument. Because like I said earlier,
I hate to say it, but if you think that that's unrealistic in nature, I've got some bad news for you. There is a lot of weird sort of sexual crossings across adult juveniles in various instars with arthropods. Arthropods are extra weird. But yeah, it kind of busts up the point that you made previously, though, that because they're taxonomically restrained, they are the ones that dictate what a kind is or isn't. And that
I'm saying it's a line of evidence that can dictate what's created and what's not, and that can actually dictate to us that humans, chimpanzees, and gorillas are all of separate created kinds. But the thing is, orphan genes are very specific. For example, we have noticed that all taxonomic restrictive orphan genes in, say, felines or the dog family correlate perfectly with what I'm saying. So right here, we've identified a feline kind, a dog kind, a human kind, a chimp kind, and a gorilla kind. I mean,
but that's that you want to talk about unrealistic. You're going to tell me that chimps, gorillas, and orangutans are separate kinds, but that every felid and every dog is within the same kind? That's wild, dude. That is like out of this world. The orangs and gorillas, really gorillas and chimps, diverged incredibly recently, even in evolutionary times.
Hold on one sec. We do standing for truth. It's got to be really short and pithy here. Standing for truth.
Yeah, you can finish up standing. Sorry, I keep inciting things, James. I apologize.
I like interaction during the Q&A. It's fun. It's fun.
Okay, gotcha. Thanks so much. Next up, appreciate your question. Speed of sound of gravity asks, standing for truth, what H3K4ME1 in Tompkins graphs indicate?
Histone methylation signatures. It's indicative of active gene promoters in transcription. He's trying to stump me, but I would recommend going to watch my debate. He's really good in genetics, so I've enjoyed my debates with him, but I got to be honest, he didn't do quite well against me on chromosome 2. We discussed it for an hour. It was a good debate. He's even admitted in one of his hangouts with some evolutionists that he was blindsided by some of my arguments. So, kudos for him to trying to stump me there. But sorry, Speed of Sound, you failed, man. Big time.
Next up, appreciate your feistiness. Appreciate your question from Terry James, who says, if creationism, quote unquote, predicted nested hierarchies, would disproving nested hierarchies falsify creationism, or would you take this as evidence against evolution?
Well, like I said, there are inconsistencies with the phylogenetic tree of life. That's why they need to look to convergent evolution. For example, I mean, you can see evolutionists invoking convergent evolution, which shows that this so-called phylogenetic tree of life is not as, it doesn't flow as well as they say it does. Because you can even look at an example that at the top of my head, let's say whales, dolphins, and bats, for example, okay? They're the only mammals that have echolocation systems. And maybe Erika has an answer to this, I'm not sure. But on the basis of gross anatomy, the whales and dolphins, okay, they're classified closer to the cow and other ruminants than to the bats. But on the basis of their echolocation systems, they should belong with the bats. Now, it gets even worse for them because molecular analysis of the protein prestin puts whales and dolphins squarely
70% of women stated that
for anyone in the audience who doesn't know, prestin is a sound sensitive protein and it's essential for hearing in general. So it's just one of the mechanisms, hearing mechanisms of the echolocation system. So this plus other lines of evidence I can go into is strong evidence that the phylogenetic tree does not represent objective reality and should falsify universal common descent. But we do predict both models some levels of nested hierarchy. So yeah, that's my answer.
Thank you very much. Sergio Sarabia, thanks for your question. They asked, Erika, how does a random evolution process create symbiotic associations? How does nature, quote unquote, select anything to work with another being in ecosystems?
That is a fantastic question. And I think that is quite intuitive because you would think that would be somewhat of a complicated process. But symbiosis has been happening from an evolutionary standpoint from the very beginning. I mean, we have the endosymbiote theory, or hypothesis rather, which is that quite a large cell sort of phaged another smaller cell, and that's how you get the mitochondria. But the truth is that it's close, loose associations that then have such a fitness benefit for these two organisms being in close proximity to one another that it actually becomes more beneficial for them to stick together than to stay apart. We've seen this recently in certain species of primate use, for instance, vervet monkeys. They can actually rely on the predator calls of starlings. Now, that has been a recent adaptation in many locations. Now, of course, it's location specific because symbiosis does take a long time, but that fitness benefit of being able to utilize a starling call to know whether a predator is around because they have a better vantage point has popped up somewhat, in the sort of distant past, and it's actually led to some really interesting conservation methods where they teach vervet monkeys how to respond to starling calls, and they actually spend more of their time kind of making their sleeping quarters near starling nests because of it. So that's basal symbiosis, but that's how it happens. It happens with loose associations that through time become cemented, and that's what I got to say about that.
Gotcha. Thanks so much. Appreciate it. And we do have another question from Stupid Whore Energy, who asks, she says, slightly deleterious mutations are responsible for neutral constructive evolution. I think that's for you, Standing for Truth.
Right. I'm glad she brought that up. I mean, that's exactly what I was iterating earlier in the debate. They assume these neutral mutations build up in the genome, and they're almost used as a reservoir for change in adaptation and in forward evolution. So that's based on an assumption. What we now know about the genome, and as Erika and I discussed during the debate, most random changes contradictory to the evolutionists are going to be deleterious. But even if, like I said, Erika, even if the 90% of the genome is perfectly neutral junk DNA, as Stupid Whore Energy is insinuating here, that means, and Erika didn't answer this problem, and I don't think anybody can, that means 10 harmful mutations are still arising in every person, every generation. So even the most fit individuals are still more mutant than their parents. And you can have any kind of selection all you want, and it's only going to amplify the best and get rid of the most detrimental, but we are going to degenerate.
Gotcha. Thank you very much. Just to keep moving, Mothra J Disco says, Standing for Truth, where does the simp gene come from?
the SIMP gene, if it's a like many genes, for example, if we look at allele frequencies, whether it's rare or common ones, they can arise, say, post-Babel, depending on their frequency in the. I'd have to know exactly what that SIMP gene does.
Gotcha.
I don't know. I'd have to have clarification. Do you know what that gene does, Erika?
New to me.
Again, to reiterate, genetics is not my field. I can speak with a decent amount of authority with primates and with human evolution. Genetics, that's why I had notes.
Yeah, I would say that the majority of our genome is created heterozygosity, but there are some alleles that have, there are some mutations. For example, blue eyes, that's based on a mutation, according to our model, post-Babel, post-flood. But we can determine that based on allele frequencies around the planet. So I'd have to know exactly what that does, if it's a mutation or if it's created. It's one of the two.
Gotcha. Appreciate it. Next up, appreciate your super chat from Jay Shy, who says, standing for truth forgets we have recombination.
Yep, recombination occurs, but recombination is no help to forward evolution. I mean, recombination and gene conversion, for example, those are a couple mechanisms that we look to in our created heterozygosity hypothesis. That's actually going to create diversity quickly, especially if the DNA differences are built in from the start. It's kind of like shuffling a deck of cards. I mean, you're going to have diversity in every single generation. That's why two people can technically have millions of children, and every single children are going to be different. No two children are going to be absolutely identical, and that's because they're heterozygous. And like I said in my opening, that would be optimal design. If you, that's all I have to say to that one.
Gotcha. Next up, thanks so much, Sigifredo Sarabia, who asks, Erika, is genome degeneration real? If no, what evidence can you give going against it? If yes, what evidence can you give that deterioration of ape DNA doesn't follow the same degeneration before evolving somehow?
So the problem with this concept of degeneration is that it depends entirely on what this individual is talking about. If this is a reference to degeneration, at least in the form of genetic entropy, the answer is selection always selects if it's not neutral. Even if it's partially bad or partially good, selection still occurs. It just might not occur as quickly. As far as variations in how selection works and other kind of genetic concepts like mutation and things like that, mutation is not constant. It's not constant in a single species through its lineage, and it's not constant further throughout into other species lineages either. Chimps have a 30% higher mutation rate than we do, and orangs and gorillas actually have a 40 to 50% faster mutation rate than us. So accumulations of any kind of mutation are going to vary depending on the species and depending on how far back they diverged. Now, again, I'm not 100% sure what they're asking with regard to genetic degeneration, but if it has anything to do with genetic entropy, there is no way natural selection doesn't select. It's not an entity. It's just literally response stimulus with the environment. There's no, people like to say that evolutionists use natural selection as like a god, but that has nothing to, that there's no, it doesn't follow. It's a non sequitur. Natural selection is simply how an organism interacts with its environment on a genotypic and a phenotypic level. And the changes that occur based on sort of a combination of chance and fitness.
Gotcha. Tioga.
Actually, if I could say something, I promise. I promise. Less than 5 words. Less than 5
I'll give
words.
you. Okay, less than 5 words. Go.
The problem is, that's 3. Natural selection acts on phenotype and not genotype, and I'll just leave it on that.
Tioga, thanks for your question or super chat. They said, "I stan Erika, my biology queen," all in capital letters.
Oh my God, you've made it worth staying up this late. That is a lovely compliment.
Very nice. Subtracted, thanks for your super chat as well. They say, hey, standing for truth, if Adam and Eve didn't know they were naked, how was Adam aware that Eve came from his rib? Genesis 2:23, you need to be naked to see your ribs.
I'm going to have to think about that one. Obviously, a theological based question, but how did Adam not know that Eve came from his rib? Wasn't God, didn't God put Adam to sleep when he took his rib, made Eve out of it? I don't know. I'd have to think about that one a bit.
Gotcha. Jay Shai, thanks for your super chat. They said X and Y Adam were separated between 30,000 years. So put that in your pipe and smoke it.
I was hoping, well, I figured we would get into the discussion on mitochondrial Eve and Y chromosome Adam, but based on observed mutation rates, the empirical method, trio studies, for example, we can use pedigree-based studies. Adam and Eve only lived just 6,000 years ago, but evolutionists have to counter that by using unobserved and unrealistic mutation rates. So they don't use the empirical method when it comes to mutation rates. So yeah, realistically, you can look at the studies and look at how those dates are derived, and they're really, really heavily based on evolutionary assumptions. So
gotcha. Thanks so much. Almost there. Let's see. Sigifredos Arabia, thanks for your super chat. They said, standing for truth, is a platypus a kind of mammal or bird?
Or bird?
Yes.
Because of the bill, I guess.
I would say platypus defies evolution. I mean, what did the platypus evolve from? It looks to me like the platypus is of its own separately created kind. I know I'm not sure if there's a universal consensus as to the history and the ancestry of the platypus, but yeah, definitely not a bird kind.
Gotcha. Appreciate it. Next up, appreciate your super chat from Stupid Whore Energy. Strikes again. She says, 2 transposable elements in humans and chimps at the same genomic locus means the original insertion was in the ancestor because there are tens of thousands of possibilities for a TE to insert itself within large genomes.
Right. So these TEs, which as he iterated, stands for transposable elements, but they are an important source of highly regulated, they're biochemically active. They are non-coding elements, but they are important in transcription, factor binding sites, and in non-coding RNAs. So they are pervasive in the genome, but they're also functional. So based on their position and location, that's predicted based on our nested hierarchical expectation based on similarities and dissimilarities. But the fact that they're functional DNA elements, just like the endogenous retroviruses, the ALUs, that's exactly what we've predicted all along.
Gotcha. Appreciate it. Next up, Stupid or Energy. Thanks for your super chat. She said, neither Dawkins nor Collins have commented on encoding or encode.
Yeah, Encode Project. Yeah.
Gotcha.
Well,
yeah,
neither
they
of them
have
are
both. I've got quotes
good.
from both of them that have commented on it.
You what? I didn't hear that.
I've got quotes from both Collins and Dawkins that have commented on it. Actually, you've referred to the CRISPR debates I've had many times. I've quoted Collins many times on ENCODE and junk DNA in general, and CRISPR had no objection to it. I mean, I'm not sure where.
I don't remember. I can't comment on it. I don't know.
Gotcha.
Yeah, that's. I've got a quote from Richard Dawkins here. He had a book in 2009. What he said was, "It is a remarkable fact that the greater part of the genome might as well not be there for all the difference it makes." Then, after ENCODE, in a debate with, I believe it was Jonathan Sachs is his name, he said regarding ENCODE, he said, "I know there are some creationists who have jumped on it because they think it is awkward for Darwinism. Quite the contrary, of course. It is exactly what a Darwinist would hope for, to find usefulness in a living world." So you see that he's kind of embraced the ENCODE results. So, I mean, there's a couple quotes right there for you.
I don't know that that's embracing it. That's embracing the work that they've done and perhaps being open to the idea that there's more functionality than previously thought, which you're right, there is. But the question is how much, and you gotta, I'm telling you, current literature, you gotta stick with it.
Gotcha. We appreciate it. Thanks for being here, everybody. Thanks so much for your questions. And most of all, we really appreciate Standing for Truth and Erika being here, staying up late and debating for us. We appreciate you guys being here.
Always a pleasure, James.
Always a blast, James. You know that. I'm looking forward to the next debate.
Always a true joy. So with that, keep sifting out the reasonable from the unreasonable, everybody. We hope you take care.
It's all yours, SFT.
Awesome. Thanks so much, guys. Tonight's the night. I want to thank you all in the chat for waiting patiently. This is going to be a really good discussion. I want to thank Ramat and Erika. Her YouTube channel is Gutsick Gibbon, so be sure to check that out. I want to thank them both. Enough for me, though. We're going to get right into it here. I'm going to pass it off to we can start off with Erika if you want to just kind of give a brief introduction. You can tell the audience, what's going on your channel. Maybe briefly go over your position, and then we'll hand it over to Ramat, followed by a discussion.
Sure, absolutely. All right, I'm Erika. I go by Gutsick Gibbon on YouTube. I cover videos on anthropology, paleontology, evolution, zoology, things like that. Mostly because I like it, but also because I have an education background in it. And today I believe we're just covering mutations and epigenetics. I imagine we'll cover the Italian wall lizards quite a bit in relation to which sort of model is more parsimonious with the evidence that we see, be it creation or the evolutionary theory. And I, of course, am in favor of evolutionary theory. And that's pretty much my stance.
Okay, well, I guess that's me then. Yeah, I mostly spend my entire life editing videos for Standing for Truth, so that's pretty much all I do right now.
That's why you get paid the big bucks, Matt.
That's right.
One day, hopefully.
With pretty much all the content we're putting out now, that pretty much keeps me more busy than you guys could ever imagine. I found it really fun, but now it's just, wow, it is overwhelming to make content for videos. So this is usually why I don't like to debate. I find making videos more entertaining and fun, which is good because Standing for Truth likes to debate. And sometimes I get dragged into it myself, like right now. So this will be a good one. The last one I did was on my specialty, which is gerontology and the study of aging. So this one is pretty cool because it's going to pit beneficial mutations with evolution. So this is going to be really good. And yeah, I'm ready to start whenever. Let's hit it.
Awesome. Okay. Well, thanks for the introductions, guys. Yeah, this is going to be a lot of fun. We'll keep it respectful. I think it's going to be a profitable discussion. So yeah, as Erika mentioned in her introduction, we can start off with the Italian wall lizards if you'd like to, since this whole discussion kind of sparked from that specific topic. And that might be just a good way to move forward. So whoever wants to start, go ahead.
Sure, go ahead.
No, I mean, absolutely. I think that with the Italian wall lizard, so for those of you who don't know, and I imagine that those following this channel probably have at least a cursory idea of the Italian wall lizard sort of experiment and observations that go kind of in tandem with that, as well as the results from 2008 and so on and so forth. But essentially what we have is a population of Italian wall lizards which were split between two islands. This population was initially omnivorous, primarily, usually, the diet was mostly insects, but omnivory would be probably the best way to put it, as I believe there was plant matter involved, however small. But the population that was split into a new environment after several decades ended up evolving cecal valves, as well as a different head shape and larger sizes, in addition to behavioral kind of novelties and a penchant for eating plants, heavy herbivory. And this has been deemed, at least in the most recent paper that I saw, as sort of this incredible example of phenotypic plasticity. And phenotypic plasticity is very vital for the concept of genetic accommodation and genetic assimilation, which in and of themselves lend themselves to morphologic change. And so I would say that the Italian wall lizards, and I believe many would agree with me, evolutionary biologists and biologists of varying fields, that this is an excellent example of evolution happening rather quickly. Now, it should be noted that these lizards, once you, if you kind of take them out of the evolved lizards, that is, once you take them out of their natural environment and feed them a heavy arthropod diet, they'll actually lose the cecal valves, which is, again, very interesting as it is an example of phenotypic plasticity. But this doesn't change the fact that this morphologic range is a result of genetic change in this population through time. And that's pretty much what I have to say about it, if Raw Matt just kind of wants to take it away from there.
Yeah, absolutely. What happened in that 2008 study is they only gathered a few selection of these mainland lizards. So they saw the mainland ones and said, oh, there's probably no cecal valve in these, because every one that they looked at they were eating insects. However, in a later study in 2015, when they actually went and gathered all kinds of different lizards, they actually found that 19% of the mainland lizards actually have the cecal valve already. And the most fascinating thing is, guess what? They were eating plants as well. So anytime something is going to a plant-based diet, it seems that the cecal valve is required. It's just like a muscle that atrophies in a human being, and it goes in this one. The cecal valve is actually a muscle. That's the thing. It's a new muscular valve that they found. It's not just some new anatomical feature that popped up out of nowhere. Matter of fact, if you type in vegetarian lizards and how many exist, how many species, they find out 1 to 2% exist. Well, how many lizards actually have the cecal valve? Well, 1 to 2%. It's exactly in line with what we would expect when something actually eats the plant. We believe in our model that animals were created to eat plants in the beginning. Well, this would make sense, so they were getting the anatomical ability to eat plants, and this is built into their DNA already. So it can vanish when it's not needed, and it can come back when it is needed. So that was a pretty fascinating thing, but they can actually track all the way back to 1971, finding the cecal valve and things. That was found in another study in 2008. Where I found that one, and they, like, within 15 weeks of a diet change, they completely dissipate this muscle. It atrophies up that quickly, but yet their offspring will still have it. It's just based on diet change, so it's already pre-built-in existing thing. And what I find ironic is that you guys need hundreds of examples of these kinds of things, and the cecal valve is just one. I mean, just one. So that's not looking too good. I mean, we would want hundreds of thousands of examples of these new anatomical features jumping up, and they're just not.
So the thing, though, with that, Raw Matt, is that evolutionary theory is, or evolution in general of species, is driven by environmental change and mutation. So if you have mutation, but you don't have a selection that's going to select for a novel change isn't going to happen, or at least that's not what we see. That's not what we would expect to happen. In fact, if we were seeing rapid evolutionary change in a stable environment, with no pressures to kind of drive that would run very contra to evolutionary theory as a whole. And I think I covered kind of some of the things that you said just now in your statement. So, and at least to my knowledge, you can correct me on this if you have read the 2015 paper. I don't believe I've read it. I read the 2014 one. But I don't believe the mainland lizards have any of the nematodes present in their guts.
I haven't seen the 2014. No, I'd have to look at that one.
Okay, well, it looks like, I guess we're at an impasse on that one because I haven't read your paper and you haven't read mine. But I would actually kind of like to push back a bit further on that. I mean, I understand that you are kind of in the same lane as I am, with 2% of our sort of squamates having an herbivorous diet, and cecal valves as well. But you have to take into account too, like dentition and things like that. Like, I understand the link that you're making. You're saying, okay, well, these, lizards, these animals that we typically think of as carnivores or, insectivores at the very least, but some of them eat plants. I mean, you're not going to find any animals out in the wild whose diet contradicts their teeth. You know what I mean?
Actually, you can look at some copperheads and venomous snakes that actually eat kelp. And they eat seaweed at night. And these are venomous copperhead snakes. They found that. That was interesting. They find they do it at night. So that's why they didn't actually discover this until recently. And
now, the question would be with that is, what percentage of their diet is the kelp making up?
Pretty much entirely. They believe that they're entirely misconstruing eating fish or eggs with the seaweed, because they smell like the fish. So the snakes walk around, and it's in our book. You can find it right there under all the different animals that decide to eat plants, like the shark that eats vegetables, or the sharks that eat kelp, things like that. But the snakes were specifically eating it. It goes back to quite a while ago, the first snakes that were vegetarian that they found. And now their teeth are made for injecting and killing. But these snakes in particular are eating nothing but plants.
So I would be interested in seeing two things. One, I would love to see the link for that. I tried Googling it. Are you certain they're copperheads? Because I couldn't find anything with regard to copperhead snakes eating kelp. Are you sure it's not like a crate or a variety, some kind of elapid snake? Just when you get the chance, if you want to shoot me the link sort of in chat, I would love to take a look at it. But the reason I bring up teeth sort of in relation to gut is because it's those two things in tandem that tend to elucidate what an organism eats. And when I say teeth, of course, I'm meaning, the shape and the pitting and sort of the microwear on that tooth. And with the guts, we're looking at the actual formation of the gut, what the insides of it look like, and sort of the surface area of it as well. Because organisms that have high surface area in the guts, it's usually used to like slow down plant material and things like that for fermentation. You usually see it in like ruminants, and not so much in monogastrics. But so I would be interested as well in looking at these particular snakes and looking at their guts and seeing if their stomachs actually match this diet that you're kind of referencing. As far as organisms eating like vegetables, if you feed them to them, I'm not certain about which shark incident you're referring to here.
My crew throws in the bait to begin the test. First tuna, squid, and kelp.
Okay, the 3 baits are in. Whoa! She just took a small bite out of the kelp. She's coming back around for a second pass. Wow, it went for the kelp again. The shark ignored the tuna and the squid and took the kelp.
But for instance, a great many animals will be kind of opportunistically omnivorous when given the chance. It's kind of horrifying that you can actually see videos on YouTube and things like that of horses opportunistically plucking up baby chickens and eating them. It's just kind of a very cruel side of nature, but it does happen. So I think that really what we're kind of seeing is you can kind of generally classify organisms as carnivores and omnivores, but again, as is typically true with nature and biology and indeed evolution as well, we're looking at general trends. So, if you want to shoot me that snake link, I would love to see it, or the one with the sharks or whatever.
Sure. But real quick, remember in our model, animals were created herbivore, right? So there was a natural instinct to be herbivore, and that's exactly what we see, because carnivores have to be taught how to kill. They have to be taught how to hunt from their parent. It's not instinctually ingrained inside of them. And teeth, they changed from epigenetics themselves, kind of like anytime that you change the diet. Matter of fact, when they took the Pottinger cat study and they wanted to change their diet, they actually didn't change the food. They actually started cooking it. That's all they did different. They started cooking the meat. And these cats, within generations, their teeth started changing and they had infertile offspring. It messed them up entirely. And to this very day, the zoos, it's illegal for them to cook foods and give them to animals to this very day. You can't do it at all. It messed them up genetically, and it messed their mouth and teeth up. But
so a great many predators, though, have almost zero parental care, right? And they still do indeed eat meat, right? Like there are many carnivores, obligate carnivores actually, sharks and various elapid snakes, that and lizards, in fact, that have no parental care. No one teaches them how to do this. They do it based off of instinct or perhaps observation as well.
I would say it's best explained through epigenetics again, because this is what is inherited. If they electrically shock something and then the next generation, I'm sorry, I should explain it a little better. If they electrically shock a mouse when it smells fruit, they normally love fruit. But instead, the next generation of offspring is scared of smelling a fruit because of this electrical shock. So when something does something, it's inherited by the parent, or by the offspring. So if the parent is hunting and killing, and that's what they do, therefore their offspring should do the same thing. So these are epigenetic tags. And this is what makes it so fascinating, because evolution needs beneficial mutations continuously, all the time. But every time a beneficial mutation comes up as what they thought was one, like the lactose mutation or the increased bone density or the HIV-1 or the high altitudes of the Sherpa people, you can go down the list. Every single one has turned out to not be a beneficial mutation whatsoever. And that's what's required for evolution, that every single one has been an epigenetic tag. So
two things with that. One, it should be noted that when we're referring to, I actually want to kind of pick your brain on this and see, what it is that you're referring to, but evolution does require beneficial mutations, but you have to understand what we're talking about when we say beneficial. What's beneficial in one context may not be beneficial in another, which is why environment is typically your primary selector. It's sort of the molder of evolution itself. You require mutation, that's the raw material, and then natural selection sort of shapes it. And that tends to come from the environment around. So I don't know why you would, there's not necessarily an inherently beneficial mutation, at least not one that I would know of, that couldn't essentially be turned against the organism in another context. So things are very context specific. This is why, I've had conversations with creationists a great many times who say, antibacterial resistance doesn't count as a beneficial mutation because that adapted bacteria is actually less fit than the other bacteria in the original environment. And my answer would be, well, of course it is. It's only beneficial in this new environment, this one with the antibiotics in it. So I would actually like to pick your brain. What do you define as a beneficial mutation?
What would I define as one?
Yes, in your own words, like what do you,
Sure.
Matt, think is a beneficial mutation?
Sure. Well, the first thing I would have to say would be look at fitness. Fitness can't drive something into a new species, and that's all that we actually see in these experiments. We're looking for beneficial mutations that would add anatomical features and changes, linearly driving up evolution. So, for example, we have finally the very first experiment ever done on eukaryotic organisms, not bacteria, not Linsky's bacteria, nothing like that. And it's been done. I just linked it to you on the side. It's a eukaryotic yeast, and they grew 12 replicate cultures of this pure yeast strain that they gathered for 1,120 generations. Now, they didn't coddle these yeast at all. They actually went full bore on trying to mess these things up. So what they did is in the first dish for about 224 generations, they put them in a carcinogistic dye called Congo red. Then they moved it to copper ions, then an alkaline environment of like pH 8, then hydrogen peroxide, and then antibiotics. And then at the end of all these generations, the scientists concluded that beneficial mutations are undercounted in exchange for environment, both in the lab and in nature. They found that it was nothing but a loss of function and degradation of function. These are what we are. We're supposed to be eukaryotic. So what I'm saying is that we need something driving the force of when we look at these charts and we're showing microscopic organisms becoming the tree of life, and we can't get even the single most primitive yeast organisms, which are eukaryotic, to change, or bacteria to change, or do the very first incremental stages, then that's horrible. I mean, when people say what is information, that's kind of like an umbrella term. It's referring to the entire genome, for example. I can give more of an example. I like to be more specific, but as far as what is information lost, think about it like the entire genome. One of you listening to me right now is missing over 100 million base pairs of DNA in their genome in different places. They've been deleted through time. That's what we're talking about. That's why we recommend people look into the genetic entropy model. It's fascinating because we see loss everywhere. We see degradation. We don't see these beneficial mutations driving bacteria up into new things. And that's required. And epigenetics even makes it harder because no gene is ever changed. And the fascinating thing about the genes never changing, it's because when they compared humans and chimpanzees and they found that it's in the germline, meaning that it's permanent, inheritable between differences between human apes, meaning they can never be changed. So that's the fascinating thing is epigenetically primates have a different brain structure. That epigenetics alteration is different. Their methylation is entirely different in their brain and in their body than it is in human beings. Now, if we were the same as them and we came from a common ancestor, then there would be evidence of that because epigenetics would regulate the exact same way that it would in them because it's passed on in the germline. See? So
I would absolutely love to have a conversation about human evolution at another time. But do you think it would be fair to say, sort of just to summarize your statement, do you think it would be fair to say that a beneficial mutation is beneficial because it increases the fitness of the organism?
I don't know. If it seems kind of convoluted because that's the equivocation. That's like saying, well, evolution occurs because fitness increases. Well, yes and no. It increases at a loss. I mean, they're throwing genes out. Take the seahorse, for example, right? When the flood happened, is what I believe anyway, the part of the ocean got caught and put up into Mount Titicaca, 12,500 feet. Well, guess what? All the salt water almost dissipated out entirely at this point. There's still some, but for the most part, they only survived because they threw all their genes out that were important, that would have allowed them to actually maintain their life in the ocean. Their fitness went up, but at massive loss. They're completely blind. They're albino. They've messed themselves up, but yes, they survived and their fitness went up, but you couldn't put them back in the ocean. They would die. So the same thing with the Linsky bacteria, these bacteria that they pull out of these other places. That's what we see. Yeah, fitness goes up, but it allows the offspring to survive in that new environment, but they can never be moved back to their original. They get limited.
Right. So that is though just evolution, right? Like that's what evolutionary theory has always predicted, and that is indeed what we see. I mean, to say that it's almost like I feel like creationists have this idea of like an ultimate fitness, which makes sense because in your model you would have sort of these barren original kinds that are sort of perfect and herbivorous and free of disease and things like that. But the evolutionary model doesn't have that. There is no ultimate fitness for all environments that we sort of have as like the pinnacle of all. An organism can be most fit for its environment, but because the environment is always subject to change, those seahorses would indeed be very fit for their new environment, the new environment that they're in. And because they're surviving and thriving in it, that would mean that those mutations would be considered beneficial. Now, I think that that's a common roadblock that we come across when we're sort of discussing these two worldviews, is because the definitions in conventional science are very different from the ones that I see creationists use. I pulled up this paper and I was just kind of reading it and attempting to pay attention at the same time, which, I'm not very great at multitasking. But I'm not sure, were you attempting to say that the findings were that, because I heard you use the word underestimate, did you mean overestimate? Because this last sentence in the paper says, these findings and the accompanying population genetic simulations suggest that molecular adaptation is consistently underestimated in nature due to its antagonistic fitness effects of mutations in changing environments. So is it possible that you misspoke?
I'm unsure. Perhaps. What I was talking about is that the 224 generations per condition that were used by these people purposely didn't allow enough time for beneficial mutations to rise to 100% to become fixated. They wanted the cells in the flask to become basically the investigators. They wanted to see if the mutations were lost when these conditions were changed. So they switched them time and time again, and in each for different length, and they found that many broken genes would pop up in the populations. So that's what
so you're suggesting sort of like a phenotypic plasticity? Is that why you brought it up with the Italian wall lizards? I'm sorry, I'm just trying to make sure I'm understanding you correctly.
Yeah, basically what I'd say is that the fitness is the total functionality of something. So when functionality goes down, we would say that it's a loss of fitness. It's kind of like a play on words. It's harder to define fitness because it's in our model, we go, well, look, it's losing information. It's losing genetics. Here, take extremophiles. Extremophiles live in the most extreme environments. That's why they survive there. But this is a small niche. And yes, they can live in the most abnormal environments, but they can never even move back to a normal environment. They're stuck there. And they're stuck in the niche. They've dumped out so many of their genes to allow them to survive. They're maxed out in what they can do. Kind of like Linsky's bacteria. They can never be jumping around. So if evolution were true, sure, you can change the environment again and they'll adapt even more, right? And therefore their offspring will come and therefore fitness will go up because their offspring can now live in that environment. But if that keep happening and their genome keeps shrinking and they keep throwing away genes, this is going the other direction. It's eventually they're going to run out of material like extremophiles and they're dead-ended. They're at the dead end.
Okay. I think I got where you're going with this. So you're essentially saying that because you're essentially limiting organisms to mutations that are deletion events. And you're saying that organisms can adapt to their environment, but this is going to be a deletion event, and eventually they're going to run out of genetic material in order to adapt to their environment, and then they're hosed. Is that kind of what you're saying?
Yeah.
Okay. So, but that doesn't happen because we have duplication events and duplication mutations and events, and as well as frame shift mutations and events and things like that add genetic material.
The survey said.
And I know that creationists don't typically like to think of that as new information, even though conventional science would deem that new information. Sort of in the same way that it's like I use the example of the sad cat sat, right? So if you had a word that says the cat, and then you have a duplication event, so it says the cat, and then you have another duplication, so it says the cat. Then you have a series of other mutations that change little letters here and there. You might end up with the sad cat sat, and that conveys quite a bit more information than simply the cat, right? Even though it's using essentially a very small number of letters and a very small amount of mutations. I believe it's like 4 or 5 actual changes, including those duplication events at the beginning.
Yeah, but they say, you can even research this, it says gene duplication is not a common event. Even according to evolution, it's 0.01 gene duplication per million years. And gene duplications are what? They're duplicating already pre-existing material. So if you're throwing out genes, eventually if there needs to be a duplication, it's not going to be there. It'll be kind of messed up.
But I think you're minimizing the sample size. I mean, you're basically like, I get where you're coming from, and it sounds quite intimidating when you say it that way. When you say, well, we're talking about, I forget exactly what you said. I think you said a mutation or a duplication event every million years.
Right.
But then there's also, with the vast majority of species, we're dealing with, I'm so sorry. Hold on just a second. I'm not sure where this music is coming from.
No problem. I sent you a link on that as well. You can see in the private one. It's the International Journal of Evolutionary Biology.
Hold on just a second. Can you guys hear that background music? I'm not sure where that's coming from in my house.
It's quiet though.
Okay, all right. If it's okay, I'll just talk through it because I'm in my dad's office and I'm sure he's just like turned something on in here. Yes, duplication events can be quite rare. But again, what I'm saying is it's not problematic because we're dealing with a vast sample size. I mean, if you have let's say we're dealing with the bacteria in an abiotic, abiogenesis style event, right? So it's like you're at the dawn of time. We're talking like, very, very like Archean period, sort of 3.8 billion years ago. And so you have a ton of different bacteria, and they're all attempting to undergo mutations simultaneously. I say attempting, they're all kind of going through their life cycle and things like that. It can be one mutation every million years, but it's one mutation every million years for billions upon billions of bacteria, which greatly increases your potential for sort of seeing this mutation. Even more so if it's selected for and it passes it on or divides even a single time, because that also increases the potential for this mutation to sort of proliferate out into the population. You see what I mean? So it's not like these events can indeed be rare, but I think that I see this quite frequently, like creationists kind of put it in a box almost and say, it's so unlikely that, that this could happen. Like I've heard one with sort of evolving new proteins or something along those lines, and it's like 10 to the 40th to the 20th or something like that. But that's evolving an entire lineage completely by chance, which is not what evolution suggests. Like pressure is one of the, natural selection and natural pressures is one of the most important things to evolutionary theory. It's like one of the tenets, you know what I mean? So I think that it's quite easy to make evolutionary theory sound very improbable, or even impossible, but only when you're using, half the pieces in the jigsaw puzzle.
But you did realize that you did say that requires deep time. That's it. Yeah, because
absolutely
it's required. So here, I'll give you an example. Take Neanderthal. We say they were, I'm going to say they're pre-flood people. And what's fascinating about them is that they have more haplogroups, they have more blood types, they have more genes, they have more complete base pairs in their genome size. And that is exactly what our model is showing. We show that humans today are less than what we were in the past. Now, that's fascinating because that lines up with exactly what we would expect. Now, if a primate was doing the thing, all right, take out human genes. How many do we have? We have about 19,000 genes in us. Do we know that number is going up? And, if anything, it's going down. Every time they do an investigation, it's like, we have less genes than we thought we did, and the number is plummeting each time. And now that we know how many genes humans have, we're not going to find new genes being added. So this process of gene duplication and different things that is required for evolution, we just don't see it.
Okay, sorry, I'm trying to mute myself and unmute so you guys hear as little of this music as possible. I figured it out. It's my sister upstairs. She's playing music over a loudspeaker. Yes, so with regard to mutations and sort of, I'm imagining you're kind of suggesting that we should see more duplication events than we do, just for the sheer fact that there's so many humans and we've been around for at least a decent period of time. You mentioned Neanderthals, which again, I would love to have a longer conversation about human evolution, because that's kind of my wheelhouse. You get what I'm saying?
Yeah. I noticed that you you would agree then that, or not agree, I was just saying, so you believe that a split occurred and humans evolved from a primate family. I've seen your videos and I've seen you debate on this, so you definitely agree with that. And have you seen the latest genetic study that showed that they've taken hundreds of thousands of species and they tested them and showed that they came out of a clear bottleneck that happened not too long ago, and that all species arose at the same time? Everything from anchovies and fish and primates and humans, all separate.
I have seen this study. This study was, I believe it was from about a year and a half ago, I want to say, because I used to have a coworker who was a younger creationist, and he brought this up to me, and we had a lovely conversation about it, actually. But the thing is that that's again, it's a misconstruing of the actual situation. Of course, modern species are indeed quite new. The problem with that is that, again, speciation occurs almost constantly. I mean, we see this sort of gradient of change occurring in almost every genera. So when they say, like, I encourage you to go and look at that paper, because when they're speaking about what they're actually finding, right? What the findings actually say, what the data is suggesting, isn't that all organisms emerged very recently. What they're saying is we have a very recent range for the species that we have currently emerging from previous species. And the way that they know that is because you can actually trace that back genetically, right? I mean, I understand what you're saying, right? That it sort of seems like, but again, I would encourage you to look at the study, because it's not all species, like you said, from anchovies to artichokes or whatever. It is a great many of them. It's like 90%, 92, I believe, something like that. But interestingly enough, that also ties in with a rather recent sort of worldwide event, right? We have these interglacial periods and glaciation events. So I don't see that as problematic for evolutionary theory.
Well, you're actually in luck. I know that study like the back of my hand, and it actually is going to answer your question that you always ask people: where do we draw the line in phylogeny? Because
yes, I certainly hope you'll answer my question.
Yes. Well, now, the fascinating thing about this is that study used DNA barcoding to do it, because it's down to the species level. Now, yes, I'm not saying that those species arose out of nowhere at the same time, because that's like Genesis, right? The creation of everything. No, this is the bottleneck of what we say is the flood. That's why it did actually count fish. So there is, matter of fact, the person that actually looked at the study said, I can't believe it. I can understand maybe a human and some deer being destroyed in a worldwide bottleneck, but he says, but anchovies and herring too, it didn't make any sense, because even aquatic life died. So I did read the study very in depth, and check this out. What they do is they looked inside mitochondria, and they found that there's 37 genes. And the interesting thing about it is one of them is called the CO1 gene. Now, this gene is very important to all species, so much so that it's lineage specific, and it's called what's called selective sweep, meaning it passes on to every single lineage of a species in that family down the line. That's how important it is. So it's in you, it's in me, it's in primates, it's in fish, it's in everything. So the really cool thing about this is when we stand back and we look and we want to compare something, we don't just go, well, look, everything with hands is related, or, oh, everything with mtDNA is related. No, we got to be more specific, because when they tried doing that with just mtDNA, they found that domestic dogs differed by as much as 0.2% with wolves. But when they compared wolves, they found that there was 4% difference. That means there's more differences between wolves than there is actually between wolves and dogs. So they needed something more specific. So they went directly into the DNA barcoding with the CO1 gene. And they found that it's always related to a family, and it branches out into new species. Now, why they like it is because they can identify new species when they pop up, meaning the barcode shows up as a gap between something. So what I mean by that is that there's just a little bit of variation when something pops up as a new species. And so there should be low barcoding gap differences between something that just speciated and that's related. The bigger the gap, the less they're related, or they fall out entirely of the group. So what happens is now we have a way to identify not only species, but what is a kind, because it works on a family level. So they went in and they studied Neanderthal, Denisovan, and modern humans, and they found low barcoding gap. So much so that I made you actually a video on this. It's actually going to be, I'm working on it, so it'll be out soon. But regardless, it's finally a way for us, besides orphan genes, taxonomically restrictive orphan genes. It's different than the other ways that we've been trying to identify kind. We now have like a trio. We have the heat maps, we have the DNA barcoding, things like this. So Anyway, I talked too long. Go ahead.
So I want to ask you, what you just said was that humans, Denisovans, and Neanderthals were very similar. Yes, you would indeed expect that. And the reason is because we find individuals, as I'm sure you know, there is a difference between finding a fossil that is legitimately bone and finding, mineralized bone. Actual bone you tend to get DNA from quite a bit easier, which is why we've been able to sequence the entire Neanderthal genome. And that's how we were able to know that the remains of this individual, I think this study came out, I want to say it was like the past year. But we found a hybridization event between Denisovans and Neanderthals. So, naturally, yes, I don't think you'll find an anthropologist on the planet, even prior to that study, who would say that humans could not interbreed with Denisovans, Neanderthals, even as if you wanted to take it that far with Naledi or Floresiensis, although there might be some difficulties getting there geographically. But the thing is you would expect those differences to be indeed quite close, right? I mean, they're, for one, even in conventional science, they're still members of genus Homo, along with Ergaster, if you want to count Ergaster, Erectus, and Habilis. And again, what you mentioned this, I wrote this down because I wanted to ask, and I'm a little bit sick, so I'm a little foggy-brained, but what primates did they test? That's a very, very, very important question. Because my question, to kind of reiterate, my point is that, that it kind of falls down the line. This is kind of my version of my go-to question, as you know, because you've seen my debates. But we use genomic comparison between humans to tell paternity, right? And it works, and it holds up in court. And we use a very similar process, albeit a bit souped up, to make comparisons between species. And so my question is more on a technical level, right? And it's like, all right, well, where and or why do we draw the line, or do creationists at least draw that line between species? And why is it reasonable? And that's why I asked which primates, because you're going to get a very, very different result if you use a loris or a galago or a tarsier than you are if you use an orangutan or gorilla or a member of genus Pan. So do you know off the top of your head? Or also, I would love it if you would send that study to me in the chat on the side so I can have it pulled up.
Oh, sure. Yeah, they tested the bonobo, the chimp, and the gorilla. That's I know off the top of my head.
Okay.
They like even more. Oh, so go ahead.
Yes. So I was going to say that would be perfect. That would be your perfect individual. So now we need some scale, right? We need to know if you tested the bark. I'm assuming I'm using the terminology correctly. I haven't seen the paper, so I don't know. But so now you want to establish a scale, right? You would want to say, all right, well, how do humans compare to mice? How do humans compare to cats? How do humans compare to insert mammal here? Because the general consensus, right, the trend that we see is no matter which area you compare, even if, let's say, you take a random protein and humans and chimps show a 25% similarity, that is indeed very different. But the sort of stipulation with human evolution is that similarity is still going to be the closest between those two, humans and genus Pan, than it is between humans and any other mammal, similarly with chimps. So do you know, by chance, off the top of your head, since you know the study, if they've tested other mammals and what they got?
Oh, yeah. Matter of fact, they did. Matter of fact, they tested bear, caribou, all kinds of things. Matter of fact, that same study that I was telling you about earlier, where they tested the 100,000 species, separate species, that was the one I'm talking about as well. This goes in conjunction with that. So that they've tested all kinds of things with this DNA barcoding. Tons. And you can see the visual layouts that they do for DNA sequencing, and they also test base pair combinations. So what they do is they look for sequences and insertions, and they try to combine them in multiple different ways. Then they found nucleotide sequences with the cheapest and fastest, because you can kind of stand back and it looks like a puzzle. You're like, oh, I can identify, that's all fish. And then they just throw them over there. And then all these are birds, they throw them over there. And then they go, okay, well, now we got to get into the detail. But the CO1 gene is down the line used because it's the germ line, it's passed to everything in the species, and it's handed down. So if we were going to be taking that back to a split, an ape ancestor, it would have gone to both of our lineages. So we would share a lot of the CO1 sequences, but because the sequences are so vast and the barcoding is so big, they fall into an entirely different family. That's what's fascinating about the study on my end. So take for the COI sequences contain around 650 base pairs in most groups tested. As I said earlier, the human, Denisovan, Neanderthal, all the way as far as you can go back in the human lineage, are only 1 to 4 nucleotide sequences different. And that shows that they're closely related. So when we do the same thing with chimpanzees and bonobos, which are in the same family, again, there's more genetic diversity even between them. But anything that really goes over a limit, you see where they fall out of the family line. And so it's really good. You'll be able to visually see it in the video that I made for you.
So I want to again kind of pause it. Perhaps it's just my foggy brain with this, but so are you suggesting that because this? Let me just kind of frame it the way that I'm kind of picking up on it. So you're saying that based off of this DNA barcoding, you get genus Homo, right, and all the members of genus Homo sort of in a group on their own. And then you also separately, by quite a great many differences, you get the hominoids, so your other apes. Would I be correct in saying that?
Right.
Okay. That's what's expected, though. That's why we're in a different genera than the members of genus Pan and the members of genus Gorilla and the members of genus Pongo. So, and I would say, okay, kind of, like, yes, I agree. I don't think that humans are literally chimps or that we should be in genus Pan. And in fact, again, every single member of our lineage we should be closer to than we should be to chimpanzees or bonobos, right? Because they're sort of like cousin. We're not related to them in a sort of linear fashion up and down, but rather we're at the end of a branch that's split by some 7 to 13.3 million years back. So I would say, to kind of clarify, that a great way to figure out whether or not this is going to work for creationists with sort of this idea of kinds, right, is there a way to create sort of separation lines, right? Because the problem that creationists have, and I would say that Jensen has kind of attempted to solve this with this idea of like common design, amphibious vehicles for the transitional species, like because we create amphibious vehicles, God might, and that's why we see transitional forms, etc. Jensen likes to cover his bases. But I think that you're still kind of in trouble, right? Because, and again, please do send me this paper in the chat while I'm talking, if you get a chance, because I would love to have it pulled up. But you have to be able to have something to compare it to, something that, at least according to, like if you're attempting to disprove evolutionary theory, right? You want to have something that's going to falsify it properly. So what you would need to have is humans, muscelids, right? Cetaceans, felids, whatever, a bunch of different family members or group members, genera, whatever, and line them all up, and then compare it with this DNA barcoding, right? In order to see who's closest to who in sort of how you just mentioned it, but across a very wide range of living organisms. And then it would be quite interesting, and I that's why I would like to see the paper, because I want to see what they concluded, to see if that matches what we find with regard to just comparing coding base pairs, through genomic sequencing, or sort of phylogenies that we get with molecular clocks and things like that. You get what I mean?
Yeah. No, they did test hundreds of thousands of species, so they've got what you want. They're two celebrities. They've got everything you want. They do line up with fish, whales, but remember, we're talking about a species line. So that's how we're looking at. We do have a different way. We do have a way. So when we're looking at base pair sites, you see humans alive today are never more than 2 positions different, exactly the same as Neanderthal and Denisovan. Yet chimpanzees differ by 60 sites, and gorillas are separated from 70 sites from us, placing them into an entirely different family. So if they were same in the species like us, if they were still branched out, that missing link common ancestor would have passed on these because they're germline, like I was saying. So we would be able to see them in the line. And they tested all kinds of things. That original study where they tested 100,000 species, that is tested using DNA barcoding. So they did that as well. And the best thing of all was wolves. They tested wolves, and they looked at are they related to dingoes and foxes, and they were related to all the different types of animals. But yet, when they compared them to bears and badgers and raccoons and things that are supposed to be related to them as well, totally different outside the family.
Right, but you'll forgive me if I say this sounds exactly like what phylogenetics is. I mean, this, to me, like you're saying this, and I'm hearing you, and I'm like, okay, so they're using genetics to group organisms into categories. Right, that's what we've done. That's why we have the caniforms, and that's why we have the primates, and that's why we have, sort of these groups that we have in zoology and when we try to classify organisms. So what I'm hearing here is that this is another means to do so, and that even by this means, we're still ending up with humans incredibly close to the other apes and closer to them than any of the other mammals, and that the apes are also much more closely related to the humans than they are to the other mammals. So I would say it sounds like a very fascinating study. Again, I'd really like to see it. So if you can, send it my way. But I don't understand the point that you're making. I mean, again, maybe it's me. Maybe I'm not understanding it. Or let me try it a different way. Are you trying to say that because we can group them into these categories based off of germline mutations, and I'm imagining that it's probably like a divergence or speciation time that they're sort of calculating, or rather looking at these lineages. You're saying that because we can group them into these different categories, those are the Barrowman groups that creationists should use. Is that correct?
Yeah, sure. A Barrowman is kind of like invented by creationists, so I guess we'll use the term, sure.
Yeah, I mean, I'm just trying to use the right terminology. The terminology that I've seen in creationist literature tends to be created kinds are Barrowmans, right, or Barrowmans. Maybe I'm not pronouncing it correctly. But again, yes, I would agree with you. Organisms are going to fit into neat categories. Neat category. Neat is a very arbitrary term. They're neat enough to draw, nice trees to the best of our ability. But the problem comes with when you compare them to one another, right? Like, my problem is there's no stop point, because the very fact that they could do this comparison and that they could say, well, look how similar humans are to the other hominoids, and look how similar all of the hominoids are to one another when compared to all of the caniforms, which are all very similar to one another, but quite different from the primates. You know what I mean? So again, it sounds to me like this study is, it's giving a sort of a different way to categorize organisms, but it's just confirming the previous way. Sort of like isochron versus other methods of radiometric dating. Like, it's sort of a corroboration, if anything.
Well, the good part about this is that it's not just like a hierarchical pattern, because we predict the same thing, but this one shows those clear genetic boundaries. And you'll see from the researchers what they say. They go, I can't believe this. It's astonishing to them because it's actually not what they predicted. It's outside of their prediction, which is really good, because this is also like I was saying, it's down at the species level. That's what they want. But that selective sweep mechanism that shows that this CO1 gene is passed down throughout the entire family line would actually show the relation of things. And what's really cool about this is it shows that the hierarchical pattern that's in humans is always in humans, and it should be the same that would be in the primate lineage. That's what I'm getting at. Just like you can follow it in the dog back to a fox to a wolf.
Right, but see, I guess I'm getting confused because I feel like you're mixing terms up a bit. So when you say that you're tracing this down through the family lineage, do you mean like the actual category at the family level?
Right, exactly. You can go up to the family level with the barcoding, because
but not the order level, which would be primates or primates.
I would imagine that it's probably too small for that, because it's 650 base pairs, but I'm going to find out as much as I possibly can studying this, so like I can get the video absolutely completely drawn out, and then I'll be able to differentiate of where I can go, because it's used for plants. When it first happened, it was like, oh wow, we can finally identify new plant species, because looking at a plant is extremely hard. Looking at an animal is a little bit easier. You can kind of differentiate the dog kind or the cat kind pretty easily just visually, but a plant, now you're getting really, really tough. So
but even visually, it's a completely bunk method to look at vertebrates and try to compare, or invertebrates, really any member of Animalia, because you've got very, very, very consistent sort of convergent evolution between forms if they live in the same environment. That's kind of what I was saying earlier. That's why you have in areas where there aren't sort of organisms that fill in the cat niche, for example, like in your traditional carnivore, right? Like on the island of Madagascar, you have fossa, right? Which aren't strict. They're not the same as like a lion or a leopard or anything like that. They're entirely unique, but something had to fill the niche. Or the Tasmanian tiger over on Australia when they were still around. Marsupial, very not a canid, definitely not a canid, but looks very much like one. So I'm very much looking forward to your video. I want to see what you have to say, because to me, and again, it could be that I'm totally missing the mark here, and or maybe I'm taking crazy pills, but I feel like what you're saying is that this study is just a different way to confirm the sort of phylogenetic tree that we currently have.
Well, it's one way. It's just one. What I mean by that is you guys want the answer to the phylogeny challenge. So what we're doing is we're combining. We have the DNA barcoding now. We're going to have the taxonomically restrictive orphan genes. We have the statistical tool called heat mapping, which catalogs basically compares the expressed proteins of species, because it proves by visualization methods that you can group species that belong to, physical comparisons by proteins inside of them. So we have that. Then we have zygote fertilization, which obviously is really good, but becomes ethical problems as well when you get into humans and primates. But we can use all these different varieties now to definitely be able to define what a kind is. We can do that with all these varieties rather than just a single heat mapping.
10 minutes left.
Yes, I absolutely look forward to hearing sort of the idea behind that and seeing what you guys come up with. But the phylogeny challenge is Aaron's. It's not really mine. I don't have a name for my challenge because I'm not big enough to have a challenge name. I just kind of it's just the question that I frequently pose.
I think where do you draw the line on phylogeny is basically the same as
it's kind of just limited, right, to primates. It's looking specifically between humans and chimps. And to me, because essentially what it sounds like you were saying, again, this is my best grasp at it, but it sounds like you were saying that via DNA barcoding, they looked at all of the primates and they got all of the primates in a single group, and they found that humans, Denisovans, and Neanderthals were very, very close together, and that the members of genus Pan, so chimps and bonobos, were really, really close together, and that they differed by 60 with humans, and then gorillas differed by 70. And I would guarantee you, if you looked at that paper, it would say that the pongids or the orangutans differed by quite a decent bit more, but not too much. Which again is exactly what we get with genomic comparison. We find that humans and chimpanzees, and as well as bonobos, are one another's closest living relative. I bet you too that if you looked at that study, I don't know if they did this or not, but that if they compared chimpanzees and bonobos to humans and then also compared them to gorillas, that they would have a 60 difference between humans and likely a greater difference between gorillas, because that's what we get with the genomic comparison. And so while I appreciate the idea that this is going to, and I hope certainly that I'm very much look forward to analyzing to the best of my ability what you guys kind of put out as far as the kinds go. But this sounds to me like it's still putting humans with the rest of the hominoids rather, particularly because you mentioned the caniforms. You're talking about all the canids and you're saying, well, this is how we know that the wolves and the dingoes and, the foxes and the dogs are all landing sort of in the same kind. But if you're going to use that methodology, and I would be willing to I'd put $20 on this, mostly because I'm poor. But I would bet that if you use that same methodology, Matt, that you're using to group those canids for kinds for the baraminology purposes, if you apply that to the primates, you're going to end up with humans with the rest of the hominoids.
Well, if that was true, then the dogs would have been lining up exactly with the other bears and the dingoes and the other things that were branched off from their lineage. Because remember, everything goes back in another family.
I mean, what you said was that the canids all lined up together, right? So wolves and dogs, right, are if I remember correctly, wolves and dogs share, if we're looking at just like a basic genomic comparison, they're about the difference between humans and Neanderthals. But I would wager that you would put all the cats together, the cat kind, right? And the difference between a domestic house cat and a lion is about 95.5%, I believe, when you're doing a genomic comparison. I can give you sources on these later, but obviously I don't have it right off the top of my head, which is much greater than the difference between humans and even orangutans. So, that would again put humans in, like, what I'm asking for is an empirical reason to separate humans from the other apes. And I don't think that this provides one. You know what I mean? If you want to create kinds, I would love to see the different kinds. I'm saying that I don't think it's possible, as someone who's versed in anthropology and primatology, I don't think it's possible by any means to use the same methodology for every group of organisms to find your kinds and exclude humans from the apes. You might be able to say, we're using this methodology for all the animals that we don't deem special, in the image of God, and then we're going to use that categorization to separate the humans. But that's not empirical, so it's not science. You know what I mean? So I look forward to your video. I'm interested in seeing how you put humans separate from the other apes.
Sure, yeah. I'll make the video a little bit longer because I did leave out, orphan genes and different things like that, and where they're related. And humans are mostly in the brain, and apes are in a different spot. And I think that because humans have been thrown taxonomically in with primates, that it kind of befuddles everything because they don't even look at the differences. They don't talk about the different ribs, the different how many spinal columns we have, the subcutaneous layer of fat. There's so many different vastness that would have happened in. You would think we would have kept some of the beneficial attributes that primates have physically, but we can get into that later. Another thing that you wanted to talk about one time was radioactive decay. And
yes,
that's fascinating because we're all kind of out of time, but I did want to talk to you about something very fascinating. They believe that stars explode and they create all of the different elements. But what we've actually found is that the prestigious Science magazine, they admit that supernovas cannot make any element over iron, which is, I think, like number 26 on the periodic table. Anyway, that's over 60 elements that cannot be made. But the Proton 21 laboratories in Ukraine, they created the events that replicate Noah's flood by Walt Brown's theory. And guess what happened? They made every single one in a process called Z-pinch. And it occurred quickly, not over millions of years. Instantly, they were able to make, matter of fact, they created more than the 60 elements. There was 3 that they don't even have a name for. And here's the interesting thing. The elements occurred and when they were formed exactly like they are on Earth today. And when they were tested old. So even if the clock, sorry, go ahead. You want to say something?
I want to let you finish. Go ahead. I have quite a bit to say on that. It'll probably have to be a brief conversation. I know we don't have that much time, but I want to let you finish your thought. I interrupted without thinking. Sorry.
That's okay. I just saw you starting to speak. I just don't want to.
No, I jump the gun sometimes. I'm sorry.
But okay, so a guy named Adam Blanco or something, he came up with a process called cold repacking. He found that super heavy elements decay into the other 94 elements, but they also make the unstable one, the radioactive elements. We believe in our model that there was no radioactive elements in the pre-flood world, that they occurred during a flood. So what happens is in this experiment, they wanted to find out like what could happen if this occurred. So they took lightning and they coursed it down, and they did it. They actually did it by accident. So that's pretty cool. And what happened is that exactly the same proportions that these elements exist on Earth are exactly what happened. So that's strong elements that these formed during the flood because their decay rates match exactly what we see when they form right in front of your eyes. Now, let's say you find a clock on the ground and or 100 of them on the ground. You take that and you go, well, they're ticking at this pretty much constant rate. So you can guarantee, well, they're constant. We can guarantee that. So that's the cool part about this because we can extrapolate that back. Like, well, when was it made? Well, you can't really do that. So, we can't tell when a clock was made just based on when it's rotating. Well, that's what we're doing with the elements. We believe that they started out at 0 or at 100%, and they've been decaying by half-lives ever since. But this method has actually extrapolated that that's not the case. They don't have, even if you start out with a 100% parent isotope to decay from, the Proton laboratories showed that new elements are not like this. So that's what's fascinating about this experiment. They've showed our model completely.
Okay, so one, radiometric dating, there are many different methods. I recommend this book to everyone that I get the chance to, particularly those who are religious, it's called The Bible, Rocks, and Time. It's written by 2, I'm not sure if they're old Earth creationists or theistic evolutionists, but they go through it's a dense book. There's like 100 pages just on radiometric dating. And I find it a very handy guide for things like this. You mentioned that I really want to see some of these studies because unless they're quite recent, like as in the past 3 months, and maybe they are, to my knowledge, every time we've seen rocks form, right, on planet Earth, be it in sort of volcanistic conditions, which would, I imagine is most of them, they always start with a heavy bias towards the parent. And when I say heavy, I mean they start with all of the parent and none of the daughter. That's why, isotopic decay or radioactive decay, the law of radioactive decay, is sort of that spontaneous decomposition from parent to daughter. And it's not been violated in meaningful ways. Even the creationists had to admit this, right? So I'm interested to see this Ukraine study, but also I'd like to point out that the method of radiometric dating known as isochron dating assumes nothing about the parent material. And yet it still corroborates methods that do, which would be to say very, very, very old rocks in, by the methods that we're using for older rocks, and so on and so forth for younger ones, and gradient and etc.
Right, but that element was actually created over iron, so that would have been something that also formed during the flood. And by the way, here's an interesting fact. I don't know why I'm throwing this out there, but did you know that silicon-32 and radium-226, they actually shown that they changed from seasonal variation? Isn't that crazy? Their observation of decay rates actually changes when it's cold and warm.
So I think that it's easy to say that, though, because I can also say that certain elements, when you apply whatever, I believe it was electromagnetic fields, that they change their decay rates. But when you actually go in and look at the paper, the decay rate changes by less than 1%.
Yeah, it's almost
which is a hefty amount. And actually the guys at Rate from AIG and Creation Ministries and things like that, they were the ones that were like, yeah, look, we're going to have to assume some exotic solutions. We're going to have to look to miracles and things like that, or assume that we're going to find more about this in the future, because as of now, we got nothing, which is why I'm asking for the dates of this Ukrainian paper that you're talking about, because, and again, feel free to send it in the chat. That way? Yeah, that way. Because I want to see the dates on this and see when it was published, because I would imagine that the geologists like Andrew Snelling, right? Like he's like the go-to creationist geologist, even though in his conventional work, he seems to accept billions of years at least for his, mainstream stuff, which he might have to for the job. But I would say, in addition to that, right, well, backing up, they would know about these papers and would probably have expounded quite a bit on them if they'd been released before the RATE project was disbanded. But moreover, I said this to Bill Morgan in our last discussion, companies all over the world use radiometric dating in order to find natural gases and oils. And they do, this is like a multi-billion-dollar industry that relies on this working. I'm sorry. Go ahead.
I'm thinking I'm thinking that you're saying that I don't believe that the rates are correct. No, they can be 100% correct. That's the fascinating thing. The rates can actually be 100% true, but when they're created, they're already created with different parent-to-daughter isotopes already begun.
Right, but that's why I brought up isochron dating, which makes no assumption about the amount of parent material. So isochron dating it's a relatively recent method, I believe, in the past decade, and it makes exactly zero assumptions about the amount of parent material present at formation. And it still corroborates the dates that we get from traditional radiometric dating. You see what I mean? So I'm struggling to understand how you can make that claim. It sounds to me like, at least if I'm getting this correct in my head, that you're suggesting that, so if there are no radioactive elements prior to Noah's flood, which I don't know what I'm not a physicist, I don't know what a world would look like if it had no unstable isotopes. I imagine that there would be quite a few natural processes impacted by that, but I don't know that for certain because, again, I'm not a physicist. So you're saying that we only get radiometric decay beginning after Noah's flood, and then the elements that we have are only giving the illusion that they are actually very old, because when they were instituted or whatever in Noah's flood, there was actually quite a bit of daughter material already available. Am I correct in that?
Correct, yes.
Okay, so then my question with that would have to be, it's an awfully large coincidence then when we compare sort of our radiometric dating methods to plate tectonics. So the way that the plates move on top of the mantle, right, via convection, it's completely independent to anything regarding physics or radiometric decay, radioactive decay, right? It's literally just big slabs of continent on top of magma, moving at a relatively steady rate. It has like a range, but it's generally like an inch or 0.8 inches, I think, to 1.2 inches per year that the continents are moving apart or together, depending on which one we're looking at. And if you look at the Mid-Atlantic Ridge, right, which is, of course, in the middle of the Atlantic, there's a big seaway there that's, pumped, or big, whatever, volcanic way there that's pumping out new continental shelves all the time through burbling magma, the ring of fire and all that. We all learned it in middle school earth science. But you can make a prediction, right? The rock that's furthest away from this burbling magma crack in the crust is probably going to be older than the magma that's sort of at the center of the crust, because it's coming out, at a relatively steady pace, at least from what we can tell since we've been measuring it. And so it's awfully strange. And of course, we get these rates by having like satellites taking pictures and things like that. It's awfully strange that when we take samples from, very close to this crack in the crust, and again, at the edges of this Mid-Atlantic Ridge, that they corroborate our radiometric dating. When we date the rock, right, via radiometric dating, and when we scale the rock at the edge all the way back using a similar rate of seafloor spreading, those things match. They're entirely independent from one another, and they match. So what you're essentially saying is that God, for whatever reason, decided to have the movement of the continental plates, the continental drift of the planet, match up in stepwise fashion with the radiometric decay rates and measurements that we pick up, just both independently equaling a very ancient Earth, or at least appearing to equal a very ancient Earth, despite having nothing to do with one another. I mean, you realize what an awfully large coincidence that seems like, right?
Well, think about it like this. They have been telling us that Earth's core has 3 main sources of energy, which are potassium, uranium, and thorium. If this is the case, then why don't we find more of it the deeper we go? Clearly, over billions of years, these heavy elements, like uranium, would be concentrated in the core. And if this were the case, then the oldest uranium would be at the core. And as it rises at the mid-Atlantic ridge, then logically, the closer you get to the ridge, the older the dating of the rocks would be. However, the exact opposite is true. The further you get from the ridge, the older the dates become. Now think about it. Over millions of years, uranium would have made its way into the core of the Earth. This would be the oldest uranium, as it would have taken the longest amount of time to get there. Then, it would have pushed its way up through convection at the mid-Atlantic ridge. Therefore, the further you get from the ridge, the younger the uranium would date. Since we find the exact opposite is true, and the further away you get, the older the dates become, this is an invalidation of that theory. So the idea of the slow geologic process and the dates along the mid-Atlantic ridge prove nothing in regards to how old the Earth is. And like I was saying, that this study showed that when these elements were created, they're created instantly, and then all of a sudden there's dissipation, right? They start decaying from their 100% into their weaker elements, into their parent to daughter. So if this is occurring and it's being spread out, by the way, God's not doing this. It's just, these are just elemental forces taking place on Earth, and so we can track those. So if it just because they are dating as though they're old, that's our interpretation of them dating to be old, because we're assuming that all of a sudden, well, it takes millions of years for this process to occur, but this experiment shows them occurring immediately. So that's the cool part about it.
So again, I do really want to see this paper, and I want to see the other two. I don't think you sent it to me.
I have so many links. I'm not even sure which one it is, because as you can see here.
Just send them. I would send me all of them, and I'm more than happy to dig through them.
Okay. I just want to jump in real quick, guys. Sorry. It's been a phenomenal discussion. Everybody's loving it. I'm loving it. We could probably listen to this all night. I do want to respect the debaters' times. Therefore, if you think this is fair, what we can do is kind of end it with, we can start with you, Erika, because you started, I think, in the intro. Give a couple-minute summary. If you want to respond to something that Matt just said regarding the radiometric dating, you can. Just use the 2 minutes or so, however you please. We can do the same with Matt, and then we can call it a night if you think that's fair.
Absolutely, that's totally cool with me.
Beautiful.
Yes. So is that sorry? Is that cool with you, Matt?
Oh yeah, fine. Go ahead.
Yes. So essentially, I'm gonna take my time and I'm gonna respond to what you just said. So again, you're using the Z-pinch method, which I'm going to, just for the sake of argument, assume that is 100% true. I don't think it is. I really want to see the paper, but I'm going to assume that it's true. Even if that is the case, I'm going to reiterate my past point. The rate at which the seafloor is spreading is entirely independent of radiometric dating. That means it has nothing to do with physics. It's just basic, I guess it has to do with physics in that it's like Newtonian physics. It's just like movement, right? It's continents sitting on top of magma, and then the convection currents are what's moving the plates either to one another or apart from one another at roughly an inch per year. This is entirely separate from this Z-pinch that you're suggesting. So there is no reason to suggest that they would be stepwise, and that's why I said we're assuming that God would essentially have to intervene here and say, okay, boom, they're stepwise. Because otherwise you've got this weird situation, like a 1 in a trillion, where the Z-pinch occurred in such a way that it precisely matches up with not just the rate at which the seafloor is spreading apart, but also all of our ice core data, all of the dendrochronology, the coral clocks that we use, none of which have anything to do with radioactive decay, right? We measure coral clocks by growth rate and the size of the reef, the height of the reef. I think the Inuit Atoll is usually used. Same thing with ice cores. It's regular snow rates, or dendrochronology, which is rings. And I think creationists usually like to say sometimes you get double rings, but that's actually not the case. You're more likely to get sort of the opposite of what you would need, right? At least according to Wood Marap, the creationist who has trouble with the bristlecone pines. But again, I feel like you're invoking kind of some funky physics here. That's again, why I want to see the paper for myself. And see when it was from, because I would find it very interesting if it was from the past. Genuinely very interesting if it was from the past 5 years, because obviously I wouldn't have seen it, and the guys at Rate wouldn't have seen it. But if it's even a decade old or 15 years old, you sure would think that the guys at Answers in Genesis or Creation.com would have kind of sunk their teeth into it. And I keep decent tabs on those guys, and I haven't seen it. So to summarize, you have these multiple actually, 4 to 5 independent lines that have, for some reason, all converged on saying the Earth is very ancient. I think that's going to need a miracle invocation for that particular thing. And then 2, you have the guys whose sole mission is to evangelize young Earth creationism, not talking about it. So those are my 2 kind of, but please show me the paper moments. And that's all I have to say.
All right. I guess I'm up. All right. Well, yeah, I mean, we can always jump into that. I think that what is fascinating is that when the people go around the world and they look at these megalithic sites and they start testing them, they find all these discrepancies. That's why people invoke ancient aliens nowadays, because they're finding things that are before humanity even existed. So it's just what's going to happen sometimes, is because they're building these places with elements that were formed in the flood. So I think that's why there's discrepancies. And but I mean, coral clocks, their estimates are all right, but then when they find that the temperature in the ocean fluctuates and the warmer it is, the faster they grow, I mean, it's okay, but it's not a really strong case in my mind. I think that I kind of showed that there's a line in ancestry that can be drawn, and I was shown a little bit. So I'll have a video out on that one coming soon, just to definitively kind of set it in stone. We're going to use more on this coming up, and that'll be good. And the reason you don't find these things in Answers in Genesis are things because they do not like Walt Brown. They don't like his theory, so they don't talk about it. That's why you've never heard of it. I recommend watching a video called by Brian Nichols, and he has it. It's called Where Did Earth's Radiometric Dating Come From? And I'll type it in right here for you. Brian. See how bad I am at debating? I'm just randomly conversating with you. And other than that, we covered a lot. So I don't think that a new anatomical feature arose in some lizard because it decided to become vegetarian, because we can find these cecal valves in other lizard species all around the earth that are geographically isolated. They're in different lizards besides these lizards, and they look exactly the same. They're just a muscle. Epigenetics, I really wanted to get into like showing that all these beneficial mutations that were on in the past, when they talk about, there was a mutation where we found the lactose and it can be tracked to one woman, and then they found that wasn't true. It's just a methylation that happened inside that showed, there's no beneficial mutation at all. And then the bone density was just another methylation event that occurred in somebody that worked out more. All these immunity from HIV, the high altitudes of the Sherpa people, all this information is already set into the genome, and it's expressed differently by the genes that are already inside of us. And evolution needs hundreds of millions of examples of beneficial mutations. And we have these eukaryotic organisms and bacteria that can live hundreds of thousands in generations, and we can irradiate them. We can give them, we can change their diet, we can change their environment, and they cannot do even the most minute steps besides change their fitness. And I know evolution believes that can happen throughout millions of years, but we would love even just the slightest example of something going on like that. And when everything is just being epigenetically switched on and off and genes being lost, I see everything as the exact opposite of what is required for the theory to be true. So I'm more interested in answering questions. So I like answering the phylogeny challenge coming up, that where to draw the line, and that's my next focus. So stay tuned, everybody.
Okay, well, thanks so much, Matt. Thanks so much, Erika. Once again, thanks for giving us your time. As I said earlier, and I'll say it again, it was a great discussion. Time flew by, and the audience had a good time. So, of course, that's the most important part. So, once again, thanks so much for your closings, and we're gonna shut it down. So, Praise, it's up to you, brother.
It should still be noted that the human Y chromosome is still most similar to the chimpanzee Y chromosome than to any other animal. The human Y chromosome is still most similar to the chimpanzee Y chromosome than to any other animal. The human Y chromosome is still most similar to the chimpanzee Y chromosome than to any other animal. The human Y chromosome is still most similar to the chimpanzee Y chromosome than to any other animal. The human Y chromosome is still most similar to the chimpanzee Y chromosome than to any other animal. The human Y chromosome is still most similar to the chimpanzee Y chromosome than to any other animal. The human Y chromosome is still most similar to the chimpanzee Y chromosome than to any other animal. The human Y chromosome is still most similar to the chimpanzee Y chromosome than to any other animal.
Genetic entropy is something that he mentions a lot in this book. Genetic entropy is really, really stupid. But I'm also not a geneticist, and that's not really my world. Yeah, I'll be the first one to admit that I'm not a geneticist.
Again, I'm not a geneticist. I can't say for certain genetics is not my forefront.
Because I'm not a geneticist.
Again, not a geneticist, and I'm not a geneticist, not a biochemist.
I'm not a geneticist.
We should have a conversation about genetic entropy. I've heard you speak to like CRISPR and stuff like that before, and I would be very interested to have that chat. I'm not very, well-versed, as I've said, for like the 20th time. I'm not very well-versed in genetics.
Yes, so here's the thing, and I'm going to use this to segue into genetic entropy because I feel like that's kind of where you're, pulling a little bit, which is fine because I got my notes right here because I don't know enough about genetics.
Again, I cannot emphasize enough how this is not my field. I would, as I've continued to kind of begrudgingly admit throughout the course of this conversation, that genetics really isn't kind of my field. I can talk to you a lot about, other stuff, but genetics isn't it. I don't know about because this isn't my field. Genetics really isn't kind of my field.
I'm not a genetics person, but
we're not going to be touching very much on genetic entropy. I feel very comfortable hand-waving it.
I'm not a geneticist.
That's not my field. I wish I could give you a better answer. I can't give you great answers because I don't know them.
Erika, my biology queen, all in capital letters.
Oh my god.
I don't know about because this isn't my field. I'm not a biochemist. Again, I cannot emphasize enough how this is not my field.
Idiot.
When we see genetic entropy, I'm going to say genetic entropy, and I'm going to do the genetic entropy sign, and we're going to say it's like this, and then I'm going to say skip. So I'm going to be like genetic entropy, skip.
And this is something I drummed up.
This is something I drummed up.
But does evolution have testable?
They claim it to be true.
But can you really test it? You couldn't recreate it. I mean, it would seem like that's the same standard they would have to be held to if that was something.
Exactly.
Okay.
And the way they phrase it, the way they apply it, is maybe the better way to say it, is there are certain expectations they have if evolution were true. In practice, it's a really plastic theory. And I can give examples of evolutionists from the 50s saying, well, we know evolution's been going on for millions of years, and so it's hopeless to find shared DNA among species. It's just been going on for too long. Mistakes have been happening too long. That's a prediction, expectation of evolution. Lo and behold, we find that mice and fruit flies use a similar section of DNA to build their eyes, even though a fruit fly has a compound eye and a mouse has an eye like ours. Well, we've just falsified evolution, haven't we? We've just found the opposite of what evolutionists have expected. No, that's now the textbook evidence for common ancestry. So evolution predicts one thing, the opposite is found. Well, now that's the fulfillment of the prediction of evolution. And these sorts of things happen below the radar in technical journals, and most people don't see it, but this is the reality of evolution. And really, evolution isn't testable. It isn't predictable. If it can explain polar opposite conclusions and both of them be confirmation of evolution, you can't have it both ways, is really what it comes down to. But evolutionists want to have it both ways, and they call those predictions when in fact evolution is an idea that after the fact explains everything. And of course, if you can explain everything after the fact by storytelling, you don't explain anything.
Spartan Theology says, who is your favorite creationist to interact with? Who has the best arguments? And this is really mean, but I don't really know that I don't really know that the arguments can be laid out on like a super coherent tiered list.
I can't give you great answers because I don't know them.
I think most of them are they are on a gradient of bad. I will say that.
But I'm also not a geneticist, and that's not really my world. Yeah, I'll be the first one to admit that I'm not a geneticist.
Again, I'm not a geneticist. Genetics is not my forefront.
Because I'm not a geneticist.
Again, not a geneticist. Then I'm not a geneticist. So I'm not a biochemist.
I'm not a geneticist.
I'm not very well versed in genetics.
Fine, because I got my notes right here, because I don't know enough about genetics.
Again, I cannot emphasize enough how this is not my field. As I continued to kind of begrudgingly admit throughout the course of this conversation that genetics really isn't kind of my field. I can talk to you a lot about, other stuff, but genetics isn't it. I don't know about because this isn't my field. Genetics really isn't kind of my field.
I'm not a geneticist.
That's not my field. I wish I could give you a better answer.
Yeah, that does count as like one of 2 times that I've been outsmarted by what is supposed to be a lesser primate.
There's so much beauty in looking at our living relatives and our cousins, and you look at them. I had the pleasure of getting to go to Gombe Stream National Park in 2015 and see the chimps there, and you make eye contact with them, and there's and this is romantic. I'm romanticizing it, of course, and obviously there's no way of knowing what's actually going on in their heads.
Smitty, will raccoons ever evolve into people?
The survey said.
I hope so. I mean, I was under the impression that they're already like little people. And then I was like, animation takes a long time, and storyboards are really more fun anyways.